I would like to welcome those who have just joined. We'll be starting with day two of the symposium in 10 more minutes. Let's wait for others to join as well. And meanwhile, we begin. Maybe those who did not join yesterday or could not introduce themselves can let us know about their background and their research interests in the chat box. Thank you. Yes, I can see some more participants have joined. Those who have just joined can put about themselves in the chat box and let us know about their educational background and their research interests. We'll be starting with the day two of Research Symposium in a couple of minutes. And also, those who missed out on some of the sessions yesterday, we'll be soon posting each and every speaker talk on our symposium page, the same schedule page where the Zoom link was given. You'll be able to look at each speaker videos over there. So if you missed out on any of these sessions, please refer to the page. We'll be sending out the mail soon and you can go through the talk. And if you have any questions, you can get back to us. We'll get your answers. Hello, everyone. We'll be starting in 2 more minutes. I can see other participants are joining as well. Let them all join. Okay, so I have an announcement to make. Meanwhile, we begin with today's day two of the symposium. The participants who had registered themselves for the symposium, it's important for them to sign up on the OmicsLogic Learn portal. I'll share the link in the chat box. Why is it important? Because as a part of your registration for the symposium, you have an access to the OmicsLogic Learn portal which includes all the courses that we discussed about and also we'll be discussing today. And this access is for a month, so you can go and learn from each and every coursework, sorry, and also have an access to demo account on the T-Bioinfo server platform. Yeah, so that is why it's important for you to have an account on the portal. And once you have an account, please let me know so that I can grant you the access. I can see among us Gunakar sir. We are pleased to have you with us, sir. Hi, Dr. Lewis.
Hello, Sonalika. Hello, everyone. It's nice to be back.
Good evening, Dr. Mohit. Yes, excited to begin with day 2 of the research symposium. Okay. Yeah, so I think we are good to start, sir.
Yes,
So
I think we
after
should
the
start.
huge success, yes, sir. So welcoming you all to day 2 of Omics Research Symposium. Yesterday was a nice, successful day of day 1, where we had amazing sessions and talks by speakers from India, Africa, US, South America. And today we are here to begin with day 2. And today we'll be having again different researchers and academic experts from USA and India. And we listen to our great panelists who are industry professionals from Federation of Asian Biotech Association, FABA, Prema's Life Sciences Private Limited, Google, and Pistol Alliance. So to begin with today's symposium, I think it will be a good start if we could quickly review what we went through yesterday, and then we can go forward with today's talk. So first of all, I would like to invite Dr. Mohit to please give us a quick review about all the talks that we had yesterday, and then we may begin. Over to you, sir.
Thank you, Sonalika. And let me share my screen. All right. Thank you, Sonalika. And hello, everyone. A warm welcome to all of you joining the second day of the Omics Research Symposium. And we are really happy and excited to have you all with us. And in this presentation, I will take you through day 1, where we learn from the experts from India, Africa, US, and Latin America about the changing role of bioinformatics in academia and industry. So let's recap day 1 in case any of you missed out yesterday. So yesterday, when we started our day 1, we started with our journey to our journey in Asia, and how we have been doing these different programs, helping students, addressing the needs of the students and faculty, as well as incorporating and collaborating with industry who are offering cutting edge technological solutions and helping students learn from experts. So in this collaboration, we are trying to work to involve and incorporate pharma industry. So we joined hand with pharma industry and organizations for training, job opportunities, and internships as well. So yesterday, the first panel of discussion, we started with different regions, as I was telling you that we started with India, and we had an academic panel discussing about bioinformatics education, research, and application in India. So the emphasis of Dr. Puniti's talk was about bioeconomy growth, which is between India between years 2015 to 2020, and the projections by the government of India, and to meet growing demands of skilled biologists and bioinformaticians, the courses that are offered by Amity University. So involvement of students in research through term papers, summer internship, major projects, and dissertations. So we learned about a little bit about the structure of the bioinformatics and other courses. So there was also discussion about how industry is playing a role in this by involving industry into student internships like from Serum Institute of India, Novozyme, Biocon, and all of those Prema's Life Sciences. So also we learned from Dr. Rita Sharma. It was an amazing talk where she talked about shrinking boundaries between biology, computational biology, data science, and all these allied fields. She's a botanist who transitioned to computational biology, highlighting several computational biology and bioinformatics initiatives in India. For example, DBT initiative to upskill students through Skill Vigyan. And then she talked about the Vidwan database for identifying collaborators in bioinformatics and computational biology that can be searched through domains, universities. So a lot of resources were discussed during that presentation, and the data-driven approach for precision agriculture technology for improving crop for India was also one of the key topic that she discussed. So after that, we had a wonderful presentation by Dr. Gaurav Ahuja, Assistant Professor, Department of Computational Biology, IIIT Delhi, India. So he was talking about the evolving role of bioinformatics in universities and research labs in India, and taking example from the IIIT Delhi as a case study. And he gave a glimpse into IIIT Computational Biology Department facilities and the vision and the various courses offered from the foundation of biology to statistical computation, from a perspective of curriculums, and from a perspective of training, and from a perspective of anyone learning, actually. So it was a great example, learning through these different academicians, discussing about technology transfer, discussing about liquid biopsy techniques, and different patents, and all of those opportunities that are available. So it was a great session. All of those panelists, there were a lot of questions for them throughout towards the end. It was interactive. And then after learning and getting clarifying to all the participants, getting clarified for the questions they've been asking, we moved to the next session, which is more focused on working on research projects. So we discussed before that a lot of application where these experts discussed about their research and what is possible. So then we discussed as a team here in OmicsLogic that how we are helping the community by working with students and mentors and experts, combining all of them, bringing this OmicsLogic community together to work on opportunities, specialization areas, research areas. And also we discussed about how we have created pipelines for universities to benefit from such a program. So after that, we had an amazing session where students from all around the globe came and presented their work after getting selected through a rigorous evaluation through the process that we have put together. And I would request our team members to maybe share the link to the poster competition or the poster presentations that that many students presented during the session, and all the judges were impressed by the participants' poster presentation. So I'll even ask many faculty and mentor who are joining to take a look at some of those research that have been done via the research fellows and also from participants around the globe, as well as for students who are starting their journey to take to look at these examples from different students who were able to achieve this and were able to present and complete work. So after that, we moved to our panel in Africa and the programs in Africa. It was a very exciting discussion that led to different kind of work that has been going on in Africa. So we discussed the considerable growth of bioinformatics in Africa, talked about the OmicsLogic programs in Africa, and then the university partnership and shared view by our team in Africa, Mr. Adi Kunle and Mr. Ayan Fay. And then we moved to bioinformatics research in Africa through the lenses of these experts working in very renowned universities and companies and network, representing a lot of specialized people and groups. So in these sessions, when we are discussing about the education and bioinformatics and data science and application, and then how we are utilizing all of this for healthcare and very specialized kind of problems related to the demographics. So we discussed about the various innovative bioinformatics research happening in Africa and impact it has on the world, and talked about how bioinformatics has become an indispensable tool in tackling various serious threats faced by the whole world, including food security, climate change, healthcare challenges. So highlighting the significance of transdisciplinary approach in bioinformatics by Professor Charles, who is a distinguished professor of microbiology and has an experience of over 30 years and over 300 publications. So it was followed by another amazing session from Dr. Virena Ras, representing H3A BioNet, and their entire project, this huge project that we discussed during that session, where the major goal is to increase the number of qualified graduates in bioinformatics, and then on-site workshops, train the trainer opportunities, internships, and the live online training hackathons, all of that. And then she discussed about developing multiple delivery mode training models, foundation of local classroom across Africa, providing training coordinators, adapting models for advanced training, virtual classrooms, all of that what H3A BioNet is planning and implementing by collaborating and also by utilizing the expertise from these trained experts such as Dr. Virena Ras. So it was followed by a panel discussion. I mean, all of this connecting the dots like bioinformatics education and training, and then the role of industry and the future opportunities, and then what are the different ways to evolve and also integrate some of these elements and keep on doing this, because it's such an evolving field that it is difficult to keep up with the advancements and also train people to do these new technologies which are relevant for the industry, which are relevant for healthcare, and the need for the trainer and the need for the faculty to adapt. And this was all about India, Asia, Africa, and then we moved to our to US, where our US team and Mr. Ilya Brodsky presented and talked about current challenges in bioinformatics training in terms of dynamic technology, lack of processing power. What are the different things that we have been facing as challenges? Where this is like it could be for curating data, creating these pipelines and workflows, and lack of practical knowledge. So how do we address those challenges? We discussed about bioinformatics SOS, so standardization, organization, and systematization. And then we also discussed about the incorporation of Bloom's taxonomy. In Bloom's taxonomy, if we looked at factual knowledge, conceptual knowledge, apply, analyze, and then understand and remember. So we discussed and looked at the pyramid resources, and then we discussed about different OmicsLogic resources like training modules, curated data sets and tools, and the T-Bioinfo platform, and the coding, no coding bioinformatics paths. And then it was also about the hybrid approach where we are incorporating asynchronous and then different learning models to make sure that everyone is equipped and have the resources to do this. So we discussed also about collaborations and partnerships with NEPI network, Puerto Rico, Louisiana, and also we have had Laura with us, taking us through this session, who is an OmicsLogic mentor representing our US team. So after that, we have this opportunity. So in the beginning, we are learning about the different education systems in bioinformatics, biotechnology, and different allied disciplines, understanding that how they work for students and what are the challenges that these experienced professors, mentors have faced, and their suggestions and their those valuable insights that could help us, bring this community together and integrate those elements. So Dr. Sona Vasudevan Professor Sona Vasudevan from Georgetown University Medical Center discussed about system medicine as an aspect of precision medicine, and the rapid explosion of omics data and these new educational programs introduced the system medicine master's degree program that is taught at the university about their different core courses, elective courses, research exposure, and then how they include analysis of big data, NGS, single cell, machine learning. So briefly discussed about the career opportunities after the course is in academia, and also encouraging students to reach out and learn more about it. So after that, we had this great opportunity to learn about strategic projects and programs by Google Cloud US. So the main focus on Google Cloud for research purpose, and mentioned about the opportunities. She mentioned about Alice. She mentioned about the opportunities for learning Google Cloud skills, boost for students and IT admins, Google computing and free modules, and then the credit courses and all of that. So two types of community at Google Cloud. So research community and the research innovator community, and shared about the stories of Google Cloud research innovators from 2021 cohort. So there's a lot of things to learn from this session that you can take a look later. So then we had the opportunity to discuss and learn by Dr. Adelaide Rhodes, who had 20 years of experience focusing on the fact that she haven't had a good experience in bioinformatics, or she had like any experience in bioinformatics recently, and then she learned about bioinformatics through OmicsLogic programs, and now she is mentoring and helping a lot of students through to learn about bioinformatics and to apply bioinformatics through the program that she's teaching. So then we moved to the Latin America group panel, and we wanted to discuss and know about the situations, comparing different models, as I was telling you before, that it is very important to understand that what is going on. So we have people from different areas, geographies, where challenges are different for students, the challenges are different for faculty. So addressing and understanding that is a key part of this entire symposium. So here we had the opportunity to know this by Dr. George Papas. So he shared his insights on the topics of big data analysis, data which is always only useful if we turn them into knowledge, and then the deep knowledge about the background in biology and theory to practice. How do we understand about this computational tools and programming languages, and how he himself learned and learned through without any formal training, and been able to teach this as a computational biology, and also to be able to do research. And also he discussed about the limited opportunities in Brazil, due to which students usually go abroad to find careers in this field, and how things can change in a very short period of time. based on the data that we are generating and the information that we are knowing and the value that it is creating. So this was followed by another talk. So if there were many talks that happened after the regular time in many time zones, such as in Asia and Africa and India and other places. So here are some of the discussions. I wanted to summarize all of this in little depth because I thought that it would be useful if you have an opportunity to know about these specific areas and what we discussed, and also to understand what to look for if you want to go back and see what was covered. So in this session, Dr. Gabriel talked about the identification and mechanism. He was talking about the circulating tumor cell for studying metastasis, and then discussed about the identification and mechanism of how NR4A1 regulates expression for immediate early gene suppressing replication stress in cancer. And then he shared his experience working as a lead bioinformatician for a project in a pharma setting. So I think it's a great session to know about how things work in a pharma setting when you're leading a group. And then we had the opportunity to get to know about Rosalind innovation that aims to promote science in youth through experimental STEM programs, and challenges of bioinformatics education in Costa Rica, bioinformatics and understanding the session in the context of the syllabus and how it is divided, like databases, management, managing biological information, genetics and evolution, and phylogenetics, and how these different topics have been taught, and how they are helping in education and learning approaches, which has been seen as a very satisfactory for among students who have gone through this learning. So after that, we started discussing about wet lab student investigating the role of STK38 in a signaling pathway. So here Mr. Piero discussed about the situation with the pandemic, and he had to work from home, transition to study the topic using computational tools. So how one can transition, how one had to transition from wet lab knowledge to dry lab because of the situation, because of the lack of resources, and further, like once because of this difficult situation, you tend to you have to do it. There's no alternative. You tend to find really the interest that was lacking. So he found his interest in this field, and then he did a second internship in Cleveland Clinic studying protein engineering. He's currently working as an astrobiology bioengineer at a project at IRMA, and he's taking classes on synthetic biology and computational biology in Spanish, as not many people speak or read English in Peru. So yeah, many challenges and inspiring stories that led to this long group discussion. That was the end of our first day. That this entire group discussion was amazing because it shed lights on different aspects, like umbrella program initiative for creating equality in STEM, and then the purpose of how do we pool new talents, and how do we use Python and research and R and research in computational biology. There are scientific internship programs and global fair network. And then Dr. Gio Poliano Chavez discussed about the computational biology programs to understand human diseases, highlighted the problems of access of bioinformatics education, and briefly talked about the promising field of liquid biopsies as biomarkers. Here, after that, Miss Victoria Maria talked about the use of synthetic biology to solve the pressing issue of leaching in Brazil. And there were many panel question and answer. So discussing about where is this gap between academia and students wishing to join research. And then Dr. Porugra expressed his views that it is tempting to think that journey towards research is linear. However, it is misconception. Anyone can plug in, basically. So this was a lot of help. This was a lot of help for anyone who started their journey or who are even thinking about just to getting on board and getting started or do something and then build on it. So if you were to focus on a single job, what would it be? So these are some of the things that we discussed in this session, a wonderful session, and this is how we concluded day one. And I'm really excited to start another day, the second day and the last day perhaps of this amazing opportunity to discuss and meet people and to talk. So we are back and with yet another day. Today, we will hear about the perspective of different researchers and academic experts from the US, from India. Also, we will listen to great panelists who are industry professionals from pharma, from Prema's Life Sciences, Google, and Pistola Alliance. So today, we will start our first talk with our keynote speaker, Dr. Rudy Saxena, who is currently a co-founder of the startup biotech company, Regene Innovation. He's helping students at pharma and Pine Biotech as a mentor. So with this, I will like to pass the podium to Sonalika to introduce Dr. Saxena and take you to the first session and another amazing day at this Omics Research Symposium. Another warm welcome, and I hope that you will get an opportunity to write your questions in the chat box so that we can address them as we get time. Thank you so much.
Thank you so much, Dr. Mohit. And I'm pleased to invite Dr. Rudy Saxena for his talk on omics technology in target selection and drug discovery process. I would like to take the pleasure of introducing him. Dr. Rudy Saxena is currently a co-founder of the startup biotech company Regene Innovations. He has held executive and leadership positions at Parker Davidson and Arbor Heterogeneity in Atlanta as VP Drug Discovery, Dr. Reddy's Laboratory as Chief Scientific Officer, US and India, and Carius Therapeutics CEO. He has a PhD in biochemistry from Memorial University and postdoctoral training at Columbia University. He was associated with the team at Park Davis, Pfizer, that discovered Liptor and atorvastatin, the largest selling peak sales of $17 billion drug in the pharma business. He led teams that brought several drug candidates from an idea into the clinic. And we are glad to hear from you, sir. Over to you.
Thank you very much, Sonalika, and thank you, Mohit. It's a pleasure to be here. Let me now try to play the slides myself. I have a 50-50 track record. Sometimes I'm able to do it, sometimes I'm not. I still haven't quite figured that out, but let me try it. Can you guys see my screen? Oh no, I have to do a screen share, is it?
No, sir. Yes.
Okay. How do I, okay, one second. You'll have to let me do it. It's not letting me do it right now. So it says one participant.
Can you try now?
Okay. Okay. Can you see it now?
No, sir, we can't see it. If you click on the share screen icon, it will ask you this window or a specific slide deck that you would like to share. You can click on it.
Voila, can you see it now?
Yes, it's coming up.
Okay,
yes, sir,
thank you. You know, you learn something new every day, so it's always a good thing. Let me now play. All right, so now you can see it, right?
Yes, sir.
Okay, thank you. Yeah, once again, thank you, Pine Bio, for a very kind invitation and introduction. As Mohit said, I'm going to tell you about omics technologies and specifically their applications in target selection and really all of the drug discovery process. So I will speak for about 25 minutes or so, and if there are questions, I don't know what the format is, but if there are questions, I'll be happy to take that. So So briefly, I'll talk about, I think I should introduce drug discovery to this audience because I'm not so sure what your backgrounds are. So I think I'll go ahead and do a brief introduction on what it takes to discover a new medicine. Then I will plug into that the role of omics, and clearly omics have a huge role in discovery of new medicines or drugs. And specifically, I want to focus on proteomics and its role in drug discovery. Right off the bat, I can tell you that 90% of the drugs that we have today have a protein target. In other words, if we did not have proteins, we would not have had a drug. So that's how important proteins are in drug discovery. And as a result of that, proteomics has certainly assumed a very important role in discovery of new medicines. So this is to those of you who may not be aware, but discovery of a new drug is a very long process. And when I say very long, it takes anywhere from 12 to 15 years. The reason is as follows. If you look at the bottom, you will see that in the purple color starting, there's three phases to it. The first phase is what we call in vitro and in vivo testing. In that, basically, you start with suppose you want to go after Alzheimer's. What you have to do is figure out what is your target. So for example, in Alzheimer's, there is something called tau. Tau is a protein that gets deposited in the brain, as a result of which it's neurotoxic, and then that causes Alzheimer's. So the target for Alzheimer's drug discovery could be the tau protein. And there's a couple of approaches. Either you can block its synthesis or you can block its function. Either way, you can approach it. So that's how it starts. It starts with an idea of what target you want to go after. And then once you do that, as I will tell you in the next slide, there's some additional things you have to do. But from there, it moves on to early discovery, in which the chemists start to make drugs that might actually block the function of tau, whichever way you want. And then you have to select the best compound that becomes what's called a lead drug or a lead compound. And then you have to move into preclinical testing. Up until now, what I just talked about is all in vitro. And then as we move into preclinical, it's mostly in vivo work, starting with demonstrating that the drug you have chosen actually works in an animal model of Alzheimer's. And there's many animal models of Alzheimer's that you can do. And if it looks good, then you have to move into toxicity testing. Remember, a drug should not only just be effective, but it also be safe. In fact, I would say more than 50 to 60% of the drugs fail, not because they are not active or efficacious, but they're also toxic. So you want a drug that is safe, which is not toxic, as well as efficacious. Once you find that gem, you then move it into clinical development. This is in humans. You have three phases: phase 1, phase 2, phase 3. Phase 1 is typically in healthy volunteers, and then phase 2 in a small population of Alzheimer's. Phase 3 is in a very large population of Alzheimer's patients. And oftentimes you also have to compare it with what else is out there in the market. So all of this takes about the preclinical phase. Up to the preclinical phase, it might take about 5 to 6 years. And the rest of it really is clinical development, which means all the clinical trials. And something like Alzheimer's, which actually progresses very slowly over a period of years, it might take about 5 to 10 years to do the clinical development part. But something like COVID-19, as we'll talk about it, is an infection, and that could, you could develop a drug there much faster than Alzheimer's. Once you successfully demonstrate that the drug is safe in humans as well as is efficacious for the disease, then you have to submit what's called a new drug application to the FDA, US FDA. They'll take about a year to review all of the data, and this is thousands of pages of data that you have to submit, and eventually then you get then you get approval. So it's about 10 to 15 years. It's generally $500 million, and the success rate is very, very low. You might start with 10,000 drugs that you look at, but only one is successful. So the probability of success is extremely low. So it's As you can see, it's a very complicated process, time consuming, high risk, and takes a lot of money as well. So this is not for, somebody who is weak-hearted. So drug discovery, now you know why new drugs, when they come on the market, might sound expensive. And the reason for that is so much of effort and money has to go in by the company to produce a new drug, right? So this is a basic format of how new drugs are discovered. You need to know this before I tell you about how proteomics are important in this process. So the number one step that you have to have, as we already talked about in drug discovery and development, is you have to select a target. And I gave the example, you'll see in the green box, it's a strategic research decision you have to make. So the target could be tau. It could have been, amyloid also, or there are multiple targets in the Alzheimer's area you can go after. So you have to actually choose one. And then you have to validate it. So what validation actually means is if you manipulate the expression of the target protein, and if you have to demonstrate its relationship with the disease. So for example, tau, if it goes up, it causes Alzheimer's. So you might take an animal model, overexpress tau protein in the brain and demonstrate that you start to see Alzheimer's-like symptoms, right? So that's target validation. So before you put any money, time, and effort, you have to validate the target to the satisfaction of the company that you are working at. Once that is done, then you have to set up some assays, which are basically in vitro assays, to be able to say that if I add a drug, tau function is inhibited or not. That process, at that point, chemists come into play and you generate drug compounds that are called hits. Then you deal lead identification. Eventually, then you have to test in animals, in which you have to look at ADME PK, which is basically once you give the drug to the animal and eventually to humans, you want to know that it gets to the brain or not, right? Because that's where it has to act. So that process is called ADME PK. And then in clinical trials, you have to kind of repeat a lot of what you did in the preclinical animal studies. You have to kind of duplicate that there. So as you can see, it's all depends with the target that you select. If you did not select the right target, which is robustly involved with the disease process, you are going to fail. And the longer it takes for you to fail, the more expensive it gets. Clinical trials, for example, are about 80% of the cost. So off of $500 million, if you fail in clinical trials, then you probably lost about $400 million. It happens. Like I said, the odds of success are quite low. So the target selection, the protein that you want to go after, and its association with the disease has to be very robustly demonstrated and convincing. So just showing something in a petri dish and saying, I think this is going to work, is simply not enough, because you're setting yourself for a failure. So everything starts with selecting the right target. And usually, as I'll tell you, targets are 99% of the time they're proteins. So how do omics come into play here? Before we had the tools of the omics, like the genomics, transcriptomics, proteomics, and metabolomics, it was a little bit of a hit and miss, but now with these very sophisticated tools, you can actually understand the disease better. So to be able to select the right target, right, you should understand the disease process very well. Which step in Alzheimer's is tau coming into play? Does it come into play before the disease sets in or after the disease has already set in, right? Because once the disease has set in, then there is no point in going after tau. So you want to choose something that's upfront and upstream in the process. And it takes a lot of gene expression, proteomic analysis, and all of that. So not only in target selection, as I will tell you, does omics come into play, but also now our ability to understand disease processes with all these wonderful omics tools that are listed here has dramatically increased the likelihood of success of a drug. If you don't have an understanding of the disease, you will not be successful. It is as simple as that. So any tool that you can use to improve your likelihood of disease understanding, the better off you are there. So not just in drug discovery, in target selection, as I'll say, but this understanding the disease itself, all of the omics tools actually have a fundamental role to play. This is just reiterating what I said. I've broken it down more into the language of drug discovery. At the very bottom, you'll see, starting from lead discovery, target selection, and all of that. We just talked about every step of these have some form of omics being used. Sometimes it's genomics, sometimes it's transcriptomics, proteomics, or metabolomics. So at every step of drug discovery, even in clinical trials, when you are actually dosing patients with the potential drug, you can get their blood samples and do an array of omics technologies and try and better understand if the drug is working or not. So I think in summary for this slide, what I want to say is there is not one step in the drug discovery process in which we do not use some form of omics or not. So omics have really, really become very important in our quest for new medicines and drugs. So of all those that I talked about, you have the genomics, proteomics, and all of that. I think it's very safe to say that proteomics probably have the central role to play in that. That's simply because the target, the basis of designing a drug discovery program is selecting the right protein and understanding it in many different ways. Its function, its validation, what happens to it if a drug binds, all of that. So proteins, to my mind, are the perhaps, and proteomics thereby, are perhaps the most important tools that we have in drug discovery today. Again, I'm trying to tell you where proteomics plays a role. Obviously, target identification and validation. The target is a protein, so it's a no-brainer you'll need. Also in lead discovery and optimization, I'll tell you a little bit more later on about that. And molecular docking becomes very important as the scientists choose which is the right drug to move forward with, right? And then the in vitro and in vivo studies, even to understand the drug's toxicity and efficacy, you want to look at the drug might be affecting the protein that you want, which is tau, for example, but it may be acting on something else that's fundamentally important, some other protein that looks like tau, and which is very important in day-to-day function, for example, in the liver, for example, and the drug might be acting on that too. So that's where proteomics comes into play in understanding what else is the drug doing besides what it is supposed to do. Is it doing something harmful or not? And in the clinical trials, I already talked, the number one thing that proteomics we use for in the clinical trials is the biomarkers. We use the biomarkers to select patients, to select how much dose to drug with, and finally to know whether they're responding to the patients are responding to the treatment or not. So within all the omics tools that are available to us in a drug discovery program that is targeted against a protein, I would say proteomics really is the fundamental backbone of how we move forward in drug discovery. This is just reiterating the theme. So one, you might ask, why is there so much protein? Why is there so much focus on the protein? And I'll come to that. But one of the things is not all genes produce proteins, right? Certain genes may never produce any proteins. So it's not that simple to go after a gene target. The way to go after a gene target would be to go after a transcription factor, for example. Then you know that there is going to be some protein expression. So proteins really are fundamentally the backbone of drug discovery. Upstream things like RNA and DNA, transcription and translation are slowly becoming important, but to the best of my knowledge, 90+% of the drugs today focus on protein, and that's why proteomics becomes such an important tool. So what makes a protein a target, right? A protein could be an enzyme. A lot of the kinase protein, tyrosine kinase, serine threonine kinases, these are all wonderful targets. Enzymes actually make beautiful targets simply because they have an active site, and using proteomics, you can understand, and X-ray crystal structure, you can understand what that active site is, what are the amino acids involved, and you can design a drug. So enzymes make wonderful protein targets. Structural proteins, sometimes like dystrophin in Duchenne's muscular disease. Dystrophin is a structural protein. It's very rare, but sometimes targets can structural protein. Receptor, and we'll talk about SARS-CoV-2, where the whole field will focus on disrupting the ACE2 binding to the spike protein, right? So that's a receptor ligand type of thing, and it's very easy to antagonize. And lastly, as I said, sometimes this is happening more in the inflammation field, a transcription factor that might actually regulate the expression of a certain protein might become a target. So these are typically some of the categories of protein targets that the industry likes to go after. How do we select a target? Now you know what a target is. It's got a protein with all the properties I talked. How do you select the one you want to go after? Ideally, you want to select a target that is the only protein in that. So if you select a kinase target, for example, right? The problem with kinase, although we have been successful, is there are probably 50 plus kinases. So first of all, you have to go after the right one and then show that's highly specific for that one. So because to avoid all that, we like to go after a single protein, for example. Tau is a great example. There is no other protein like tau. So going after tau is a no-brainer for that reason. The other, and this is the validation I talked about, it has to be directly associated with causing the disease. It cannot be a phenomenology or it cannot be anecdotal. It has to be directly, which means if I upregulate that, the disease should get worse. If I inhibit the function, the disease should actually get much better. So that's how it is, that you want a target that is directly related. The other thing that you want is that you want your target only to be expressed in the disease organ. And that is done to actually limit the side effects you might get. Again, going back to the tau example, tau is only expressed in the brain. It's never expressed in the muscle or a kidney or there. So if you manipulate tau, it's unlikely that you're going to affect other organs. So expression of your target protein only in the disease organ is a wonderful thing to have. The other interesting thing when we select targets, which is that we like to inhibit the function. That's why I said enzymes make beautiful targets, because there's an active site. I can design a drug that'll go bind to it, inhibit its function. On the other side is trying to increase the activity of an enzyme. It's very difficult. It has to be done through an allosteric mechanism, not through the active site generally. And we don't like that because those are much harder to design. So we prefer to have things to be inhibited, not to upregulate their function, but downregulate. The last thing that, and this is a really nice thing to have, is we like to have protein targets that are actually on the cell surface. So receptor ligands make fantastic targets also for that reason. Why is that? The reason is, in that case, the drug doesn't have to get into the cells or into the organ. You know, the organ, especially for Alzheimer's, there's something called a blood-brain barrier, and it doesn't let drugs to get into the brain at all. So oftentimes you don't have drugs that can get into the cell. But if you have a cell surface target, it's a whole lot easier. So these are some of the simple criteria, and omics, proteomics specifically, comes into hugely into deciding some of these target selection as well. Okay, so how do drugs interact with protein targets? So here's an example I'm showing you with the family of enzymes called tank erases. And first of all, now if the crystal structure of the target is available, it makes our job very much easier because we know what is the active site, and we can then take that active site with all the different amino acids. We'll talk about it more, how it forms a pocket and all that. But virtually in silico, we can actually then select compounds that are already there that might fit into that pocket or not. So upfront itself, we could probably screen 50,000, 100,000 in silico compounds that can fit into the pocket of the active site of the enzyme, and then only work on those. Out of those, maybe only 100 might actually fit very well. So your job is now shrunk down from having to deal with 100,000 compounds to about dealing with 100 compounds. So having a crystal structure is a very important and usually the most useful thing you can have when you have selected your target. Okay, I want to now move on to how we apply, how proteomics has been applied to a disease that now we are all familiar with, which is SARS-CoV-2 driven COVID. So what are the key features of SARS-CoV-2? On the very left-hand side, you will see everybody has heard of spike protein. So this is the virus, and the way it engages with the host cells, human host cells, is through a protein called spike protein. How does it engage with the host cells? How does it get into the respiratory system? How does it get into the lungs? Is through a receptor called ACE2 that you see. So if you look at the second box, there's the SARS-CoV-2 with the spike protein sticking out, and then you have ACE2 receptor. This is a counter receptor protein that's present on a lot of different cells in the body. The endothelium, the lung is pretty much ubiquitous. So therefore, although the focus of COVID has been on the lung, you will know now that long COVID, which is the effects of COVID that come a few months after the COVID infection, can be affecting the kidney, the heart, because they all have ACE2 there. So the virus can enter all of those organs. So the virus binds to the ACE2 receptor, and as shown on the third box, that's how it gains entry into the cells. And it creates a mayhem from there. As you know, the cytokine storm, the inflammation, and as well as the thrombosis that sets in. So a key feature of SARS-CoV-2 infection is driven by two proteins. One is spike protein on the SARS-CoV-2 virus, and the other is the human ACE2 receptor. So as you can imagine, most of the targets for COVID are actually related to either ACE2 or the spike protein. On the left-hand side, you will see strategies that target ACE2 receptors. Strategy one could be to create soluble forms of the spike protein. Within the spike protein, as you'll see, there's a specific domain called receptor binding domain that engages with ACE2. So one strategy that is being attempted is to design soluble RBD. So this will bind to ACE2, so the actual virus cannot bind. You can also actually make soluble ACE2, for example. So the ACE2 soluble simply means it's floating around. It's not bound to the virus or not bound to any organ. So it's floating around in the blood. It'll see the virus, it'll bind it, as a result of which the virus, by using both these strategies, as is shown on the second figure there, you can see that there, that the virus cannot engage with ACE2. And basically, if the virus cannot enter the human cell, it's dead. There's nothing it can do. Vice versa, you can also have strategies to attack the RBD. And this, which is the domain on the spike protein that's important in binding the virus to ACE2. And RBD has become the #1 target protein or target protein region for COVID-19. Whether it's vaccines, whether it's diagnostics, whether it's new drugs, everybody is focusing on RBD, including the virus. The virus is also focusing in RBD. And when I say that, what it's doing is it's mutating the RBD region to gain superiority, so try and escape from the host defense mechanism. So the whole world of SARS-CoV-2 COVID is actually focusing on this region called RBD, which lies within spike protein. Very interesting portion of that protein, this RBDs, and I'll tell you why. So this is just to show you the structure of spike protein. So spike protein is actually sits on the surface of the virus. It's a part of it is into the virus, but most of it is outside, so that it can bind to go and grab ACE2. As you can see here, the spike protein has actually been broken down into spike protein S1, spike protein S2, spike protein S1. The S1 is really where the RBD, you can see that in the green. RBD lies there. It's on the outside of the cells of the virus particles, and so that it can go and grab onto ACE2. All right. The other parts are, there's also an enzyme too, which is called Tempress. I mean, I'm giving you a short form that allows, once the ACE2 is bound to by RBD of the spike protein, then these rest of the ancillary proteins come into play and make sure the virus can successfully get itself into it. Right. So RBD is really the star of the movie. If somebody were to make a movie on SARS-CoV-2/COVID, RBD would be the hero, I think. Yeah, so this is just to reiterate that, again, in the orange, you will see the virus, and then you see the spikes coming out. That's a spike protein. And on the human cell, you'll see ACE2. And I just want to reiterate, everything we are doing today to combat SARS-CoV-2/COVID is targeting this protein, whether it is vaccines. All of the vaccine, either using a nucleic acid approach or using a protein subunit approach, the antigen that's going to be used in the vaccines is RBD. There's nothing else. Some people use full spike protein, but a lot of them use RBD. The idea is, if you either vaccinate with RBD or produce RBD in human body in small amounts, you start to have neutralizing antibodies that come into play, which will stop the binding of the virus to ACE2. Same with diagnostics. There are many diagnostics now, which is the rapid antigen test or the RAT test you've heard. What are they detecting? They are detecting spike protein. And the drugs. I mean, there are many drugs that some of them we have worked on and published are also focusing on that. So RBD interaction with ACE2 is fundamentally the target for everything that's going on SARS-CoV-2. So why is RBD so important or so interesting? Well, I already said it's important because that's what makes the virus enter into the host cell. Beyond that, the virus is very smart. Once you have developed a vaccine against the original strain, right? What happens is that once you have the original strain, once you have the original strain, and you find a vaccine to it, the antibodies to the original strain RBD will actually neutralize it. So the virus has started to mutate itself in the RBD region. Originally we had the alpha, then you have beta, gamma, delta, and now Omicron. The one big change in all of this are that mutations are occurring. So the virus has figured out that if I mutate, change some amino acids in this area, then I might be able to escape the neutralizing antibodies that's being produced by these humans in response to vaccines. So it's trying to constantly outsmart the humans by doing that. So not only does it escape the vaccine when it actually changes or mutates certain amino acids, all of the mutations that have a detrimental effect are in the RBD region. To the best of our knowledge, every single one of them, Delta, Omicron, Gamma, they are all in the RBD region. And some of them make the virus bind tighter to the ACE receptor, as a result of which they can enter, but they're more transmissible. Some of the mutations make them lose the neutralizing activity, and I'll tell you about that too. So just remember, the take home from this slide is RBD mutations are fundamental to how the virus has been moving forward all these times. All right, so one of the things you will see is you may have had the word called escape mutations. There's a big problem. What are escape mutations? Escape mutations are nothing but mutations in the RBD of the virus that actually cause the mutated virus not to be neutralized by existing antibodies. So just to show you in the box here, we created vaccine against the wild type RBD, right? And that we were able to neutralize. The virus mutated itself. It created something called B1.351. When this happened, the neutralizing activity slightly decreased. So delta actually slightly decreased. The neutralizing activity that was generated against the original wild type didn't fit very well with the delta, for example. And so not all the neutralizing could happen, so the virus could escape. Actually, things got even worse with Omega now. There is about 39 mutations plus in the RBD, and they seem to be escaping all antibodies produced by the vaccines to the wild type. So you can see that having small changes in the RBD, substitution of certain amino acids, gives a huge upper hand to the virus in escaping, and that's why they're called escape mutations. Escaping the neutralizing antibodies that we are producing. So sometimes what happens is we are giving ourselves a booster. I mean, recently, in the hope that maybe having higher titers of antibodies might stop the Omicron. We don't know how it's going to happen, but Omicron is clearly is not being neutralized by most of the vaccine-related antibodies that have been created. So the virus won there. So far, that has won. If with the wild type and delta, we are very happy because the vaccines work pretty much nicely. Omicron, we have started to see some issues. So why is the vaccine success depend on protein antibody interaction? So again, having a crystal structure or structure of these, the RBD domain, as well as the antibodies becomes very important. What I'm showing you here in the green is the RBD. And then I'm showing you, breaking blowing that up a little bit, is that there are different epitopes. Epitopes are just regions of a protein to which antibodies bind. And because of the mutations that have happened, the epitopes are changing. When you mutate a protein, the amino acid change, right? The structure also changes. Inevitable that the structure will change. Once the structure changes, the original epitopes to which you designed your vaccine might be hidden or might be lost or might no longer be recognizable. So that's why, this interaction between the antibody and the vaccine pretty much dictates the success of your vaccine, right? So if there is too many mutations in the antigen and your antibodies cannot dock very nicely, that vaccine is going to fail. I want to move on and tell you about how omics and protein drug interactions are important and form the basis of small molecule drugs also. I talked a lot about vaccine, but even in designing small molecule drugs, the interaction of a drug and protein is very important. Here's an example. So this is the structure of a drug. And what is important here is how will the drug fit and bind to the protein in a region of your interest. And usually the interaction between a drug and a protein are either hydrophobic, which means that there are hydrophobic amino acids there, and your drug might have some hydrophobicity, so there'll be an interaction there. The other types of interaction, hydrophilic, there might be charged amino acids on the, for example, an enzyme in the active site, and the drug will have to have charged amino acids and interaction there. So there's hydrogen bonding, there's electrostatic, hydrophilic, hydrophobic. All of these are important in the high affinity binding of a drug to the protein target. So a medicinal chemist, once he has a structure of the amino acid, he will try and tweak the drugs to see if he can fit into that pocket. So not just in vaccines, but the protein structure and proteomics are playing a very fundamental role in us designing drugs as well, to make sure the drugs fits very tightly with the protein target, tau, for example. I also want to tell you that omics are helping us understanding. So one is designing vaccine, the other is designing drugs, selecting target. I mean, all across the value chain of drug discovery, they're playing a very big role. The other big role that omics are playing is trying to better understand the mechanism by which the drug will work. Why do we need to know that? Why do I care about what the mechanism? The reason to understand the mechanism by which a drug works is then you can predict what kind of side effects it might have. So if there is a kinase inhibition is the mechanism, right? We know that there is 50 plus kinases. So it's very possible that your drug might actually bind to more than one kinase and might give you side effects. So knowing the mechanism of action is a good way of predicting the adverse or the side effect profile. And omics have really played. For example, here you can see that this is a Huntington disease cell model shown on the right hand. You throw the compound in, and there are a variety of proteomic technologies you can see to see two sets of experiments can be done. One is what set of genes are being upregulated or downregulated by the compound, right? So that gives you some information. The other is where exactly is it binding and what else is it binding? So there is a lot of exciting things that are coming in, understanding of mechanism of action of drugs since the omics had arrived. There is a saying now that a lot of the drugs that were approved in the 60s and 70s, when we didn't have these beautiful tools, would not have been approved today because they just have too many side effects. Aspirin, as an example, today, if it were to be launched, we know it does so many other things that you don't need it to do, including things like gastrointestinal bleeding and all. So it's only today or in the recent past that we've understood that drugs of the past have many other things to do, and that has only become possible because of our ability to use genomics, proteomics, and metabolomics, I would say. I think it's a very, very safe statement to make. So in summary, I think
I would like
omics,
to
this is my last slide.
Oh, thank you so much, sir.
Omics have rapidly become the heart of drug discovery. There's no question. Protein targets are the basis of 90+% of the drugs. As you can see, mutations in the protein targets can cause dramatic changes, escape mutations. And proteomics is pretty much useful in target selection, drug selection, mechanisms of action. So long story short, long live omics. Because of omics, I think the likelihood of success of drug discovery is going to dramatically increase. So thank you very much for your time. I don't know if I exceeded by a couple of minutes or so. I don't know. Yeah, I did, but don't kill me for that, please. I'm done.
It was definitely a wonderful talk. We have a lot of questions. I'll mail you out, sir, and then we'll get back to our participants with those questions.
Absolutely, no problem at all. Yeah. All right. Thank you, Mohit. It was fun being here. Thank you, Sonalika. Yeah.
Thank you, sir. Okay,
Thank you.
It's time we invite our next speaker, And our next speaker is Mr. Rajavarman Kitu, and he'll be highlighting and telling us about Ngs technology infrastructure in India and its application. To talk about Mr. Rajavarman, he has completed his master's in bioinformatics from Shanmugh Arts, Science, Technology and Research Academy. His expertise lies in bioinformatics, computational biology, genomics, NGS, comparative genomics, cancer genomics, and he has also worked as a senior technical officer, clinical bioinformatician at Kokila Bhai Dhirubhai Ambani Hospital. And we are pleased to have you with us, sir. Over to you.
I'd like to thank Biotech for the given chance. Can you just confirm if you can hear me clearly, plus you can see my screen?
Yes, sir, we can hear you clearly, and also we can see your screen.
Yeah, sure. Pardon me for the noise. I'm somewhere in the transit. Yes, so far I think we have covered. I just listened the previous talk. I think we went very deep in the COVID-sick. Now we are going to switch the gear. So currently we're going to see what is the infrastructure generally needed for the NGS. So I'm just comparing NGS to C. C in the sense the SEAC. So since there is so much of varieties, there's so much of things to explore. Uh, we are going to surf in the sea now, right? Literally the ocean. Let's see how it goes forward. So the main thing which you need to know is Ngs size. When I say size, it means the file which come out of any given machine is basically different, very different. The file format as well as the individual file sizes, starting from MB to GB. So in that case, the size really matters when it comes in place, right? Second one is which machine you're going to handle and what are the applications you do. And the third one is the checklist for readiness. When we say you're going to do the NGS, what are things you need to have in place? So let's go into detail. As I told before, the smaller organism generally gives in MBs, and this, well, let's say human data, you can go still 3.3 for 1x. So in that case, when we consider for 30x or 1000x, it's going to be bigger and bigger. So this is not just to scare you, rather than to make sure this is really the big data, which is a buzzword, which is happening in the IT industry. It's literally happening in the biology as well, in the genomics field. So it's up to the depth. It's up to us to decide how much depth we are going and how big the data is. Again, the second thing which we need to know very clearly is the data flow. When I say data flow, it means how frequently we are going to run the machine. In the sense, what are the samples we are going to load, how many samples we are going to load, how frequently we are going to operate the machine, right? Second one is deep sequencing. As I told, how much depth we are going. Is it like 50x, 100x, or 250x, 500x, or it's more than that? Even sometimes people go for even 10,000x. Based on the question which we are asking, the scientific question we are asking, the depth can go like probably vary, right? And the last one, which is very important, is what kind of panel you're going to run. Is it just a basic panel with a limited number of genes, probably the targeted genes, or the full exome, or the whole genome, or whole exome kind of data, right? So these three things has to be anyway first should be understood. And the next important thing is what are the things we are going to archive. So when I say archive, basically, for any given English data set, we'll be having a raw data, process data, and we'll be going for archive. Apart from that, in the clinical setup, we might even need to do the reanalysis, or we need to review the data set. So what do you mean by reanalysis? Suppose down the line, so there are steps where we'll be going for annotation for the given variant. So that will be changing with respect to time for certain number of genes, right? So those kind of cases where reanalysis is important. Based on that, even the therapy recommendation might vary, not in the bigger level, but in the slight deviations can be possible. So in that case, reanalysis is one of the key factors. So we need to be knowing what are the things will be permanently archived and what are the files, or probably what are the details we need to have always like in ready, so that we need to go for reanalysis. Apart from when I say review, it means say 5 years you have did some analysis. You want to recollect in the 5 years what are the variants like very commonly occurred from the given data set. And probably you can even find the trendings, in the sense recent 2 years. What are the new variants, or what are the recent findings are from the given data set. So those kind of repurpose related analysis, this module, like the second part, has to be considered. So in that case, when we really go for the data management, we need to know what are the regular exercise and what are the repurposes exercise, right? And which mission. So as we know in the market, there are, when I say market, in this market, there are very huge varieties of machines are available. Based on the needs and the application, we need to be sure about which one we are choosing and whether it is solving all of the questions or not. It is useful to solve all the questions which we ask, right? The first is purpose, as I told. Whether are you asking a basic question or intermediate or advanced, so whether the given technology will help us to answer this one. The second one is sample load. How, as I told before, how frequently you're operating the machine, how much load are you having, like the data load which you're going to have it or handle it. Last one is the capacity, whether are we having high throughput machine or low throughput machine, right? So these are the key points which has to be checked beforehand. I was keep on speaking about the missions. So when I say missions, it means, as you can see the varieties, starting from 1.2 GB to 6,000 GB. So this is the capacity for the single run when we fully utilize the machine to a full extent, right? So as I told before, These are all the, let's say, low throughput, and this is a mid throughput, and this is a high end, right? And these, where do we use a low throughput machine into? For sequencing the smaller organisms, be it bacterial, virus, fungus, right? Any given smaller organism, except the human, for the whole genome sequencing, it can be used. So particularly for smaller organisms, this sequencing machine is being used, right? And apart from this, if the sample number is less, and we need to have, let's say, as I told before, 0.2 GB, 1.2 GB, right? So those kind of smaller amount of data, if you want to let's say, generate, these kind of facilities fair enough, right? When we are going for bigger projects, say population studies or cohort related analysis, those kind of bigger studies, if you want to do, probably we need to have this kind of hierarchy. Even including the COVID sequencing data set, we need to have these kind of missions in place, right? For example, when it comes to human, whole genome sequencing and whole exome sequencing, right? And apart from that, when the sample load is higher, even though it's a small organism, but your sample load is very higher. So in that case, probably you'll be using this kind of sequences, right? So this is prone to give high throughput. Let's see what are the applications generally we get out of these machines. Of course, these machines can be used in all kinds of setup, clinical, academic, and the research. Currently, what we are seeing is in the clinic and research, it's been widely used. Academy, it's slowly coming up. So we'd like to see more the missions which we discussed in the probably the forthcoming futures, right? Application wise, if you see, as I told before, you name it any given application of genomics, it can be performed with the machines which we discussed. Whole genome sequencing, whole exome sequencing, methylation, single cell, HLA, PGD, PGS, comprehensive panel, targeted panels, RNA. So you name it, any kind of application, everything can be done, right? So, uh, okay, we know about the background now. Can we list out what are the key points to do that we should be having to proceed? So the first one is security. Why? Because there are very sensitive data is being used in the NGS, probably the medical data sets, where we don't reveal much details everywhere. Rather, all the patient detail will be converted in the particular ID, and that will be processed all over. In that case, even the data access will not be given to everybody, since there is a HIPAA accredited certifications are given to the informatics tools. Security is the first concern, right? Second one is user friendliness. Since it's not like everyone is master in the informatics to do the analysis and give the reporting. So in that case, the friendliness is user friendliness is very important. Third one is basically audit trials of file access. Why? When it comes the clinical setup specifically, which file is being accessed by whom at what time, that will be traced out, because that's very important thing to back, not only the backtrace, even to there are multi levels of screening will be done to release the report. Even those kind of places, this audit trial is very important, right? And the fourth point is basically manpower. As I told, establishing the full facility, starting from scratch to a reporting solution, is it basically needs a really good number of manpower who is very much skilled. And apart from that, we need to have a good battery backup for the setup, like say to have an infrastructure, and the room temperature should be monitored for 365 days in a year, and dedicated network is needed. Apart from that, good internet speed is needed. Why these are the things are needed? Since we are speaking about 6,000 GB data set from one particular run, you can imagine how long it takes to transfer the data set, process the analysis, and then archiving it. So in that case, these are the basic needs should be in place, right? So say I'm very new, I'm from academic. Since I can't go for all kind of thing. I have only simple laptop. Can I do the NGS analysis? Yes, you can do. Probably if you see there are softwares like Galaxy, and there are even some pipelines which is ready-made available, which can be used using even Windows. But typically, if you want to do in-house without sharing data with any of the third party, probably outsiders, in that case, this is the computation which is expected. Like this is a RAM, this language processor, probably storage is 50 TB, and Linux OS. It's very difficult to get as a workstation. Probably as a server linked with the workstation is good to go with. So In that case, to start with complicated, to answer the complicated question, this kind of computation is expected, right? So I got the facility now. I got the setup now in place. What are all the basic things I need to check to make sure my data is good enough to do the analysis? First thing you need to know is coverage. Like whatever the panels I sequence, I have got all the data set in place. Second one is depth. Like whatever the depth I estimated, have I reached the depth? In the sense, how many x depth have you reached? Right? Reads. There are cases where in RNA kind of data set, you have to know how many reads have been generated, whether those are all enough to proceed further. And the third one is mission, as I told, which mission should I process to get these kind of data sets? And sample number, as I told before, how many samples can occupy in one particular batch? If I put this many samples in this particular batch, how much of coverage and depth will I get? So those kind of pre-calculation estimation should be well defined so that the expected results are in place. And files to preserve, as I told before, which are the things should be archived permanently, which are the things should be in the front desk so that anytime you can access. That part should be very much well defined in SOPs. And finally, the process. So before even process, I would say there has to be a testing phase, or probably the beta versions should be there in place to make sure, yes, every individual process is going well without any disturbance, right? Uh, when it comes to research setup, probably open source tool if you plan to use, how does it goes? This is how the overall workflow goes. The literature survey will be conducted based on whatever the tools which is widely being used, and the same tools will be downloaded, and then we'll be standardizing based on the data set and the question which we ask. So when I say standardize, it means fine tuning the parameters. When I say validate, it means same kind of study is being observed from the PubMed publications, and then replicating the same work so that to make sure, yes, my tool is meeting the standards which has been already published. So that is a validation. And finally, the pipeline. Again, every individual application which we discuss have its own pipeline and the steps. Every steps will be validated with the relevant parameter, and then we'll be going for the final product, right? So this is a protocol which has been followed for in-house development. So in that case, the complication is really very high until otherwise you are not skilled. So in that case, there are alternative solutions which is there in market, which is basically the softwares are already feeded in, the parameters are already tweaked, and it's very easy to use. In that case, cloud solutions are available which can be used. Apart from that, there are some servers which comes with FPGA card where all the programmings are already feeded, validated, and standardized. The moment you probably it's been installed from the day one, it can be used. So I'm not going into these two topics much. Why? Because we are mostly going to speak about infrastructure. With that, I'm just closing this talk. If you have any questions, I'd like to take it forward.
Thank you so much, Mr. Rajavarman, for your presentation. If you have any questions for sir, please put them in the chat box and we'll be able to get your questions answered. And also those participants who are asking about the recordings, yes, we'll be sharing with you the recordings very soon. For that, you need to fill out the registration form so that we have your email IDs at least where we can share the recordings with you all. So we'll just put the registration form the list in the chat box, and those who have not registered themselves, they can fill the form. Okay, so sir, I wanted to ask the question, what are the career opportunities with Prema's life sciences?
Okay, currently, since you took the name Prema's life sciences, I would say there are who are PhD and postdoc passed out, they have opportunity for product managers, as I can see, right? Again, not in the informatics perspective, overall, probably the coming up with the new assay, validating studies. So there are multiple openings out there where probably we even, since we are covering all kind of segments in the sense, we train the people in the analysis. We train the wet lab people for preparing the libraries and loading the samples. Plus, we have a specific marketing team who takes care of probably all the marketing activities and the sales team. So in that case, we have opening almost in all the departments at this given point. So if you're asking specifically informatics, I don't. There might be a junior level intern opportunity might be available, but not a full-time job. But with other segments, there are full-time jobs are there at this time.
Okay, sir. Thank you so much. And the next question is, what could be the possible online resources for NGS-based identification of microbial variety?
Okay, so when you say the database is already in place for almost all the model organisms, the sequencing data, which the process data as well as the sequencing data is in place. Particularly when you say smaller organism, I can think about two things quickly, which is MTB and COVID. COVID data is anyway it spits out with GSIDE actually. At the same time, even NCBI SRA has, to name it any kind of application, everything is already in place. So this is two resources which I can think about quickly.
Right, sir. And is Linux a must for NGS bioinformatics? What are your thoughts on it?
Again,
Is Linux, yes, sir. The question is Linux a must for NGS bioinformatics?
Okay, is Linux is important? Okay. So I would say Linux is even, I think the voice is breaking. No issues. So the Linux is important. I don't think so it is important because they are, since as I told the last slide, the cloud computing solutions which are coming up or which are in the market currently doesn't need any skills at all. So it's based on which solution you are looking for. If you are like say if we are skilled in the, let's say probably the scripting as well as the Linux commands, then probably there are certain tools which is again the FPGA card related tools, or probably developing their own pipeline can be done. If not, then there are cloud solutions which is alternative for people who are very new to informatics or not aware of informatics. So we have both kind of solutions available in here.
Right, sir. Thank you so much, sir. And the other questions we then be taking up via mail, sir. Thank you so much, sir, for your talk and answering the questions. Yes. With this,
Thanks a lot for the event.
Good. Thank you, sir. It's a pleasure having you with us. Now, with this, let's move ahead with our next speaker, Dr. Vivek Das. Dr. Vivek Das will be enlightening us with his knowledge on role of bioinformatics in the present era of high-throughput biology, digital health, and drug discovery. And let me introduce Dr. Vivek. He was born and raised in India in the city of Joy, Kolkata. Vivek Das, doctor, has been quite the wanderer, having lived in various parts of the country and the world, from having a bachelor's degree in biotechnology from Bangalore University, master's in bioinformatics from BIT, Mesra, Ranchi, IT programmer analyst job he did in Cognizant, Kolkata. He holds a PhD and a postdoc in cancer computational biology from IEO, Milan, and industry postdoc at Novo Nordisk, Seattle. Dr. Vivek's professional career has a global footprint. Presently, Dr. Vivek's passion brought him to Denmark, where he is a computational biology senior research scientist at Bioinformatics and Data Mining Department in Novo Nordisk. He is involved in biomarker and target discovery in chronic diseases using in silico approaches. His present work involves crunching big data from bulk, single-cell multi-omics, along with phenotypic measurements that need careful integration of interdisciplinary knowledge from biology, computer science, mathematics, and statistics. He also explores the application of advanced statistical machine learning and deep learning models in high-dimensional single-cell multi-omics for particular problem scopes given the data bandwidth as a part of some of his curiosity-driven research activities. And last but not the least, his hobbies include science communication, remote mentoring, blogging, traveling, functional training, hiking, and reading. And for more, you can follow him on his LinkedIn profile. I'm excited to hear to your talk, sir. We all are indeed. Over to you.
Thank you. Thanks for the kind introduction. Can you hear me?
Yes, sir, we can hear you.
Yes. So I'll try to share my screen. Can you see my slide deck?
Yes, we can see a slide deck.
Okay. Thank you, everyone, and thanks for having me here for the day 2 of the Omics Research Symposium under the Data and Life Science Getting Important, Making Impact. Today, I will speak about the role of bioinformatics in present era of high-throughput biology, digital health, and drug discovery, and what kind of work I've been doing at Novo Nordisk, and some of the gigs that I've been doing outside as a part of my curiosity-driven research. So before going ahead, I just want to present what's the perspective of how do people see bioinformaticians and computational biologists. And there are different aspects, a different perspective how people see us. It's sometimes very amazing, very funny, and I would encourage everyone to actually think like how bioinformatics is perceived in today's world. It's a very exciting field actually, and you must have all seen how actually everybody has explained what its role is. So I will just put some flavors of how I see it and how I've been using it. I basically am this person who is basically doing stuff with data. These data are generated in-house from different therapy areas, different biological scientists, or from public level resources. And then I use algorithms or methods that are already developed or we are developing in-house to crunch these data in a way that we can make novel discoveries out of it. That can help us to realize early drug targets or even biomarkers for onset of the disease or even disease progression. But the overall goal is to actually feed them into these 4 cornerstones of medicine, which is prediction, prevention, personalization, and participation. This is the 4 corners that I think bioinformatics is slowly emerging as a leading interdisciplinary field. Moving ahead, I would first like to give an overview of what I do as a scientist in our department, Novo Nordisk. We are a big pool of 30 plus people now across multiple sites at Novo Nordisk. I belong to the bioinformatics and data mining team, and we stand on these 4 pillars. We serve multiple therapy area R&Ds. We serve different research technologies where we provide different bioinformatics solutions. We also serve, providing solutions to understand mechanism of actions by using leveraging the potential of sequencing in clinical trials. We are trying to understand where clinical sequencing can help to get better understanding of mechanistic studies in clinical trials. We have some new trials that we are using the role of sequencing, more precisely single-cell sequencing. And we're also, working in translational medicine. But what we do basically in all these 4 pillars are early drug target identification, building analytical workflows for target evaluation that can be, a very fantastic target that comes from literature, from particular chronic disease areas, or data visualization, because as a part of bioinformatics, we also often try to, provide solutions where biologists can, figure out the gene names, how they are changing across disease cohorts or single cell maps and so on and so forth. And then we do a lot of biomarker identification work.
Dr. Vivek?
Yes.
Dr. Vivek, I'm so sorry to interrupt in between, but your voice is not that clear. Can you please check for your mic?
Sorry. I'll try to increase my volume. Can you hear me now?
Yes, I can hear you now. Okay.
Okay, I apologize for that. So, yeah, great, thanks. So what we basically do is we use various public and internal omics data, example, blood transcriptomics, proteomics, metabolomics, genetics, epigenomics, or single cell omics taken all together, analyze them mostly from human and also in vitro in vivo settings to derive biological insights. I have highlighted the part human is because we follow an approach in our pipeline which is more human-centric. What that does mean is we start from human data, we derive insights, we find specific targets or biomarkers, and then we try to understand what setting of different in vivo and in vitro can help us to reach the clinical phase. So that's why we start with human data, and we have a very scalable pipeline across in a human-centric manner. Now, I will go more into the details of the talk. The outline is divided in 3 parts. What is bioinformatics in time of high throughput biology and digital health? Why is bioinformatics important in drug discovery? And what is the future state and aspirations of modern medicine? Now, these are all my own personal perspectives and opinion based on my experience. And so it might not always align with everyone, but this is how I see the field and how I see the role of bioinformatics in a pharmaceutical setting and beyond. So what is bioinformatics in the time of high throughput biology? Now, modern medicine and biology has gone a tremendous improvement and evolution. I would say it's a revolution. In current age, where we have so much of high throughput, technologies, where we can measure different modalities from, we can do imaging, we can understand tissue cellular composition, we can do genotyping. We can actually also leverage the powerful omics like genomics, transcriptomics, proteomics, not only from organs, but our tissues or cells, but also in vitro settings or in vivo settings. All these taken together, when we sequence them, we can actually build up a map or network at a cellular level, right? And these, when put into sequencing machines, can actually build a huge information complex of multi-omics information that can help us to tease out the different molecular features, which when integrated properly, can help us to understand the disease progression or the kind of disease drivers. And what this gives us actually to help us to understand how does the central dogma of molecular biology work. I consider that as the hottest science in town, basically, because it helps us to understand the basic foundation of the entire molecular journey. And this is still the key to understand any development or disease biology. And sequencing, high throughput sequencing of different types of omics can actually do this. What it does is basically it tries to accrue all this information from different organs or tissues or cells from the human system, accrue or let's say, build or achieve the information from each type of different molecules together, stitch them with the information that we get from the clinical notes or the imaging, put them together with suitable models or methods. But in the end, it's trying to derive the relationship between the genotype and phenotype. And once we get this information, we can actually better understand a disease footprint. Now, why is bioinformatics important in drug discovery? Now, medicine over the years have been undergoing some paradigm shift. What does paradigm shift mean here? We know that one-size-fits-all theory in medicine doesn't work. Not every therapy works for everyone. Some have benefits, some don't, some even have adverse events. So we have been seeing a change from stratified to a more precision medicine. And what precision medicine is actually ensuring is trying to say we can deliver the right intervention to the right patient at the right time. And how this can be done? This can be done with individual patient level information that can actually take information from genomics and other omics, club them with the different information of lifestyles, their preferences, their health records, and medical history, all taken together so that we can deliver a very tailored therapeutics to each patient. And this is the dream where the medicine is actually going. And most of our industry are also trying to understand how we can actually implement this in our system when we are trying to generate new drugs or new therapeutics. And another part is, over the years, we have also learned that the drug development journey is actually very, very slow and expensive. A lot of candidates fail. Not every drugs have great effects. Most are marginal. So this poses problems, right? Because it's a huge investment. So for us, it's very important now that we need to have much more clear and effective solutions. And how can we do that? With the help of sequencing or omics, when we talk about bioinformatics comes from the fact that we are able to sequence or have omics information from different layers of the molecular biology, right? And each of these plates has its own specific role to contribute to any kind of biomarker and drug target discovery in any kind of disease understanding or developmental biology understanding, as you can see from this slide, taken together with any kind of clinical information. Now, when we take together all these information, we have to be also very efficient on what models and methods we are using, right? If we are talking that we want to develop biomarkers or drug targets. Now, this whole thing is called the era of omics medicine. And I find it very enriching that over the years, due to how the field has emerged, how our scholars in the academic field has helped us, this has become now very important component also in the pharmaceutical industry. And we are now joining this journey as well. Some of the applications of omic-driven medicine where bioinformatics feed into is population genetics and genomics. Some of you might heard about, single nucleotide polymorphism or genome-wide association studies that falls into this. Then developmental biology learning or disease biology learning using different kind of multiomics of organs, tissues, and cells, longitudinal studies or cross-sectional multiomics, and then early drug discovery and therapeutics. I have highlighted some of the disease areas that we work in at Novo. Ones that are bold are the ones where I contribute and I work, and I give my scientific expertise. However, all these are great, but these are all mostly done in a population of cells, population that was not having the information of cellular resolution. These lack that cell level information because we were always looking into in a more holistic manner where all the effect of these cells were actually averaged out. And human body is complex, as we know, as we are learning now of some new technologies that have emerged in the last few years, that our system has actually trillions of cells, and some share commonalities and some do not. And that's when the era of the single cell technologies emerged, right? And one of the exciting example of this was this new preprint that came out from the consortium of the Human Lung Cell Atlas, where they are able to now create and map a big human cell atlas, human cell lung atlas around that maps around over 2 million cells from 444 individuals. Now, this is a massive task that has been done by some brilliant scientists from the in the field that has used bioinformatics, that have used biology, that have used interdisciplinary computer science, mathematics, and statistics and machine learning all together to understand what are the different cell types that are involved in any disease setting when we are looking into the lungs. What are the cell types that are affected by variants? What are the effects of age, smoking, and BMI, and what's not? So this is one of the biggest tasks that came out recently, and this is a wonderful example of the single cell technology. However, these kind of high throughput work is also very complex because it builds a huge information complex. It runs us into the problem of dimensionality curse, overfitting, any kind of linear nonlinear structure, and lack of generalizability. Why? Because we are talking about matrix, like big tables, where big tables is composed of multiple dimensions. These dimensions comes from features that can be your genes, proteins, metabolites, and across your observations. Now, observations in the bulk setting is your patients, animal samples. But when you think of single cells, it's each cell coming from all these information, right? So that's a huge information complex. And this is often very challenging. And this is where bioinformatics is challenging, right? And I have discussed about it in a systematic review in 2020, where I tried to stress that how multiomics field work should be done. What are the caveats? What are the pros? What are the cons? And what do we need when we want to embark on this journey? And then in single cell field also, there are amazing paper out there which also speaks about what are the different challenges in these pertaining to the data science. Because when we talk about these high dimensional interdisciplinary field, we know that it comes with its own caveats. And these caveats, some of them can be data sparsity, data heterogeneity, non-flexibility of the statistical frameworks, then missingness of the data, and then integration. We can tackle some of these, but they're not that easy. How we can is if we can build tools that are well benchmarked or systematically checked with other competing tools, we can solve some of these problems. But there's a lot of work that needs to be done. Not every problems have been tackled. Some of them that we have mentioned in these papers, but not everything. So there's a lot of scope of research out there, and bioinformatics has to play a lot of role in this. Now here, what I'm trying to say is, apart from the work I do as a part of a scientist for the company I work for, Novo Nordisk, I also sometimes do work with certain students back in India, where I put my brains in some curiosity-driven projects. And as a part of these projects, I think about problems in method development for bulk multiomics integration, as you can see here on the left. Then I'm very fascinated by the concept of single cell technologies and how high throughput biology in forms of single cell is such a big data in terms of the dimensionality. And this often begs me to question, what are the best way to actually squeeze those high dimensional data in a small latent representation so that it's human readable, human can understand and derive insights from it. And that's where the concepts of dimension reduction methods and clustering kicks in. And then when we look into these single cell, we have to have those single cell in specific clusters, and this cluster seems to have certain identity. And these are not very easy tasks, because when you're talking about labeling those clusters with certain identity where a group of cells comes together, which means that we are trying to say that that identity belongs to a specific biology, and we call them as cell type labels. And I've been also been thinking around those problems for quite some time, mostly on the mathematical side, like how we can get better at those cell type labelings. So these are some of the curiosity-driven problems that I've been also working as a part of my side projects. Two of them I've highlighted, and I will share a bit of those work in the next slides. One is how to better integrate multiomics data by improving feature extraction issue. And can we improve single cell annotation using a better embedding method? The first one is based on multiomics. Here in 2020, I teamed up with a few researchers back in India, where I tried to understand, can I estimate gene expression function from DNA methylation, which is an epigenomic information, and copy number variation at protein-coding region. And we came up with a method of feature extraction. We thought that this is an issue of feature extraction at high dimension. We came up with a method that involves deep datizing autoencoder and multilayer perceptron, and we took together, we were able to see if we integrate this information from DNA methylation and copy number, can we estimate mRNA? And if we can estimate this mRNA, how good is that estimate from the real data? And we used liver hepatocellular cancer carcinoma data from the Cancer Genome Atlas that had over 400 patients' data. And it was exciting to see that when we were able to actually build a model, we were able to predict the observed RNA values as good as the real ones. But that does not end the story, right? When you build a model, it's also important how good your model is when it's being tested with every other existing models that are out there. And when we did that, we benchmarked systematically a different regularization hyperparameter, how our model is working. We found that our model was actually working as one of the top models. We did in 3 different variants, and one of the variants of our models was working, came out as one of the top with the least error. So this was very encouraging for me to think how multiomics as a feature extraction issue can be actually improved. Next, I wanted to stress a bit on my single cell work that I've been doing, how I'm thinking about the field and where the problem scopes are. Same, but before that, I want to say that single cell field is massive. There's a lot of work one can do. It's beyond what we did with bulk omics or bulk multiomics. We can get information both at cell level and at gene level, which was usually we often did at gene level when we were looking into always the multiomics, right? We did differential expression, gene set, gene regulatory networks, but now we can add another layer of information. We can actually understand what is the trajectory of a disease, what is the different states of the disease, what cell states are involved, what is the composition of those, and what are the clusters that we can derive from when we are actually sequencing these cell populations. I am particularly interested in 2 problems at this moment, where I'm trying to think single cell problem scopes based on clustering and cell cluster annotation. And recently, one of the work that I have worked with 2 researchers back in India got accepted. It will be out in the next week online, where I thought of the cell type annotation using single cell data as a problem of embedding. What I thought was, like, given we have these huge data matrices of cell and gene, what if I think of this problem of cell type labeling or clustering or annotation, whatever you may say, more as a problem of cell embedding and clustering. So we came up with an approach where we tried to use non-negative metric factorization to reduce the dimension of the data of these cell costume matrices across different samples, and then try to understand if we can then additionally use recursive k-means to cluster the data, and we can actually define the cell types from limited labels that we already have from the data. And in fact, this we tested on like 6 or 7 to 8 different data sets that are publicly available thanks to the research community. And when we did any kind of accuracy and performance, we saw that our method was working very well. Interestingly, what we also did was we randomly then missed out the real labels that were available with the data to understand how well there are cluster similarities. And we found that our methods was actually having a very high cluster similarity, as you can see from the ARI indexes for each of these data sets. Now, again, as I always say, I'm a big fan of systematically benchmarking. I always think that why will someone use my method if I'm not actually looking at its performance with any other methods? So I took a step back, and then I tried to understand, well, now given that we have seen that it is performing well, how does this perform with existing semi-supervised method or unsupervised method? And here there are some representative images where I was able to see that our method was actually performing very well in more than 5 data sets out of 6 when compared to another semi-supervised method that was recently published called Color, as you can see from the accuracy plots. And then when I compared the cluster similarity using against an unsupervised method called Rbd, which is very popular in the single cell field, I was able to find that at least for 50% of the data set, our method was as good as Rbd. And but it was good across almost all the data sets, apart from one from the other semi-supervised method. Then additionally, we also actually tested the method for intra and intercluster dispersion with 2 data sets that were downloaded from the European Bioinformatics Institute server, where there were real missing annotation. When I say real missing annotation, means these data sets had a cloud of cells which did not have actually any kind of cell type information. So we wanted to see whether we can redefine using our method those data sets, and can we actually get cell clusters out of it. And when we did that, we found that our method was actually outperforming the Rbd at that time. So this was very encouraging for us, that looking of the cell type problem as a problem of embedding and clustering, we were able to come with a very efficient, simple, scalable method. So here I'm now progressing to the end part of my slide. The last slides are future state of aspirations of modern medicine. I see that medicine is actually going interdisciplinary in terms of cell-based intercepting. Why? Because now we have a huge explosion of single cell data, and single cell multi-omics or single cell data means a lot of advanced statistical and machine learning methods, because the data dimension is huge. And this also means we are also getting better at understanding orthogonal, experimental disease models. But this is a great future for us, because now we are trying to go in a high dimensional era of medicine, where we are trying to understand the disease trajectory from healthy to different type of disease, be it neurological, cancer, or chronic. But we can actually understand one cell at a time how the disease is evolving. And we can actually detect early, which can actually understand the medicine, the molecular mechanisms, and actually, get better new drug targets. And these can all happen if we can efficiently use bioinformatics. So here I come to my last slide. I hope I was able to convey how bioinformatics is actually, helping the modern medicine and biology. All these informations that we are trying to accrue these days using molecular, spatial, molecular, histological, anatomical. This is a huge abundance of data. These are, high dimensional data. But the overall goal is to take all these different modalities of data together, build up integrated, cell phenotypic map that can be multi-scale, multimodal, which can actually help us to understand the underlying phenotype of the cell. And when we understand the phenotype of the cell, we can better understand the states that can be the epigenomic states, the cellular interaction, the tissue organization, and the identification of sometimes new novel cell type, which eventually can help us to understand disease mechanisms, interactions, signatures, and new biomarkers, and overall can help in designing leaner clinical trials, which can improve personalized medicine and eventually also help us building, better improved drugs. So this is how I see the field of bioinformatics contributing to the modern medicine. And I hope I was able to share some of the thoughts with you all. And I thank everyone who has been instrumental in providing and guiding me with the scientific knowledge. I thank my department for supporting me and my collaborators. And I hope you guys had a good time. And this was a very nice conference. I really enjoyed everything about it. So I hope you had also some curiosity-driven questions. So feel free to ask, and I'm happy to take more questions.
Thank you so much, sir, for the presentation. It was very crisp and short and giving an overall view about how these different types of techniques are important and what bioinformatics role is in all of these studies. We have a question by Jasprit Kaur. She's asking, what is the first step to begin with while learning bioinformatics and data mining? What basic skills are needed to start with, and please mention accessible sources.
So I would say that my field, my growth has been, I started as a biologist, and then I went on to computer science. I always see that it's important to pick up soft skills, so programming and a bit of mathematics. The way the field is progressing, one would need a bit of more understanding of mathematics. I would always say to pick up programming and mathematics or statistics a bit more if you want to venture in the modern bioinformatics. And the biology is important, but that you can learn over the line. There are some resources I can share. It's if I get an email, there are some very available free resources that is there from Swiss Institute of Biomathematics. I keep sharing some resources on my LinkedIn profile as well. So I would be happy to share them offline to the group here.
Right, so we also have a plethora of resources, and we'll be able to share as well. Yeah, okay. Thank you so much, sir. And the next question is, what are the applications in digital health and how are they being employed in the process of drug discovery? What are your thoughts about it?
Yes. That's a great question. I didn't touch much on the digital health, but what I'm trying to portray is when we consider digital health, the digital health can be, wearables, measurements from the wearables, blood pressures, right? And these can be done from your smartwatches. And these are often your snapshot, and they are these days taken also into account when we're talking to your doctors. Now, how it will be used in a day-to-day, it's still an open research problem. FDA has not yet approved everything. We don't know how it is going to be approved, but having a blood pressure, ECG, EKG from your wearables, that's already a great data point for you to improve your lifestyle. So you can always improve your lifestyle on that front. In bioinformatics, it's still very early, but there are some trials going on. So we will learn more about it in future. At this moment, I would just say that in measuring blood pressure, it is some observational way of doing it. So yeah, that's the way I would see it.
Right. So our next question is, can you brief on the translational potential of single-cell omics and how categorizing the heterogeneity of cells will help in targeting the tumor as a whole?
Well, that's a great question. So when we talk about trials, it has to be, so single cell as a diagnostic is still very early. There are some trials going on, but what we are doing is we're trying to understand, can single cell help us to understand the mechanism of action of certain drugs? So there, single cell is used, quite being used right now, where we're trying to understand the role of different, semaglutides and all those things, mostly in the chronic setting. What cell picks up, because the effect size is pretty low, right? That's where the single cell is actually helping a lot. Now, cell type problem is actually cell type heterogeneity in terms of tumor is a very interesting problem, because it's an uncontrolled growth of the proliferation of the stuff. I think there are some very interesting papers that came out recently in drug tolerance. I think we should check on that. I don't remember the name of the author right now, but what they're trying to say is your single cell is actually trying to tell you, when you're giving in tumor patients, particular solid tumor, a particular drug, how the drug is responding and what cells are responding and what cells are not responding. I think this is one of the fascinating things to understand, whether the drug is actually being treated by some small cells that are responding to it, and they are actually reversing the disease. So this is how these things can help. But these are still very open research scope problems. They're not entirely solved, because, as I said, the data is very, very complex and highly abundant. So it's not something that is already solved. We are still trying to tackle these kind of massive problems as an open research problem. But there are, leaps and bounds of work that are already going on.
Right, so I hope the ones asking the questions, your questions are clear. And also, we'll be able to share with you some of the resources that you can go through to learn more about single cell. And as the question was put up, like how you can characterize the heterogeneity of cells, targeting the tumor as a whole. So we'll be able to answer that as well in more greater detail. And thanking you once again, Dr. Vivek, for your talk. It was an amazing talk, and it was nice to have you with us.
Thank you.
Now we would like to, thank you, sir. Now we would like to proceed ahead with the next talk by Ms. Anka Maria. She'll be talking about collaborate to innovate, about lab of the future and women in STEM. To introduce Anka, she has more than 10 years of international management experience, creating and leading cross-functional teams, working with large corporates and SMEs. She has led the introduction of new products and services across 140 markets. In her current role as a consultant at the Pistra Alliance, she is a strategic team lead for improving the efficiency and effectiveness of R&D. Her role includes leading communities of interest in life science and healthcare R&D for more than 150 member companies, including the Pistra Alliance Lab of the Future community. She also leads the Pistra Alliance's new Women in STEM Leadership Pilot Program and is responsible for developing other non-competitive cross-industry initiatives where members of the Pistra Alliance can collaborate to solve common challenges together. So pleased to have you with us, Ms. Anka. Are you here? I can't.
Yes. Can you hear me?
Yes, I can hear you.
Thank you very much for the introduction. Thank you very much also for the invitation. Could you see my screen? Can you see my screen?
Yes, we can see your screen.
So again, thank you for the invitation. I'm really happy to be here. My name is Anka, and before I start this presentation, I would really want to tell you some things about me, about my educational background and career background. I studied biotechnical engineering. I have a master's degree in the medical engineering from the Polytechnica University of Bucharest and an MBA degree from the EU Executive Academy in Vienna. I must confess that although the university helped me a lot in forming my mindset and in developing the logical approach when it comes to solving different problems, I never worked in the area I thought that I'm preparing for as an engineer. What I did after graduation, I actively searched to join a multinational company from the pharma industry because I had the desire of having a job that for me was meaningful in terms of helping the people in need, but in the same time I had the dream of becoming the type of the business person that travels from places to places and meets different people and cultures. So I wanted this international exposure, and I really wanted to meet and work with people from different cultures. So I climbed in my career from a project management and marketing, starting with Romania. Then I moved to responsibilities on Central Eastern Europe, and then I went at the European level responsibility in less than 7 years within the same company. Then I moved to a small medium enterprise for another number of years, where I got global responsibilities and where I built the project management department. And then I became an independent consultant. I joined the Pistoia Alliance as a strategic film lead, as you presented earlier. And in my current role, I see myself as a connector. I connect people, I connect their ideas for more than 200 member companies of the Pistoia Alliance, all across the R&D ecosystem. I facilitate collaborations between these companies in order to address and to solve common challenges in a non-competitive way. In this session, I'm going to present 2 of the currently running initiatives. One is called the Lab of the Future, which is a community of interest. And the other one is the Women in STEM, which is a program that we are currently piloting. Do you see my next slide?
Yes, we can see
that you see the next slide. Okay, thank you very much. So before going into the presentation, I'd like to tell you a few words about the Pistoia Alliance for those of you that maybe didn't have the chance to work with this organization. It's a not-for-profit organization. It gathers under its umbrella more than 200 member companies, including the 16 out of the 20 pharma companies, top 20 pharma companies. And the mission is to lower the barriers to innovation in life science and in healthcare R&D through pre-competitive collaboration. This means that we bring at the same table different companies that might be competitors in the business environment, but we facilitate a collaboration between them for a common purpose. The COVID-19 changed our lives in many, many ways, including in the way we work. More companies became more permissive to remote working, and the cross-industry collaborations also increased. We saw a high acceleration of digital transformations across the healthcare industry, and the benefits of these industry-wise collaborations are very well known, and more and more organizations we see that are leveraging a legal framework that not-for-profit organizations can facilitate in order to collaborate with different organizations, research-based organizations, maybe digital technology players, also patient groups, for example. Both of the examples that I'm going to show you are global initiatives, which means that we involve people around the world to support each other, to share and to learn and to address certain challenges, but also to look for solutions together in the benefit of the whole industry. And when you will go on the Pistoia Alliance website, you can see a list of projects and a list of communities of interest. And I highlighted just some of them on the right side of the screen. Among all these initiatives, we also have the Lab of the Future community, which has a mission of driving a pre-competitive approach to building the next generation of laboratory environment. And we are looking from a perspective of data standards, processes, instruments, and knowledge sharing. And the members of this community are presenting use cases, are sharing the challenges and best practices, but they are also exploring different ideas and initiate projects that can help support the lab transformation in the digitalization pathways. Now, one of my roles here is to furthermore develop this community and the engagement between the participants. So we are looking at short, medium, long-term perspectives, and we organize dedicated workshops, webinars, projects, and community discussions, looking at the improvements that can be done in supporting these lab transformations, no matter how far an organization is in this process, in the digital journey. So we gathered a good mix of industry experts from the pharma, CROs, tech suppliers, academia also, and other types of organizations with wide diversity of roles that are working in the ecosystem of R&D labs. On the right side, you see a highlight from the second half of the year when we organized different webinars. Almost half of the participants came from pharma companies, but we also had other organizations joining, and also almost half of the participants are having leadership roles, which means that they are decision makers and they can have an influence also within the organization, but also for the industry-wise project that we want to develop, for example. Now, how will the lab of the future look like, and what does it really mean? There are many general predictions. People are talking about having a smart lab or an automated and fully remote AI-powered lab, cloud labs working hand in hand with scientists, scientists working completely remotely without being confined within the four walls of a lab and so on. But there are different types of labs, and there isn't one path that can fit all. And it's very interesting to see how these laboratories are evolving. What is their digitalization speed, their particular vision on how their future will look like. And even though there are some differences, we can all agree that there are a lot of common mechanisms, interfaces, terminology, technology, and so on. And they do have some similar milestones and objectives that they want to achieve in this transformational journey. Let me show you. One of the common visions for all type of labs is that they will make use of the automation and digitalization. So the digital infrastructure needs to take into account the large volume and variety of data that is being produced. Some later on challenges can be avoided if the organizations have data centricity in mind when implementing the new systems and workflows and automations. And one of the general challenges that all labs are facing is how to capture this large amount of data that is being produced, including the contextual data, in a fair format, which means to be findable, to be accessible, to be interoperable and reusable. And how to redesign the current paper-based workflow in case the organization is still recording the experiments, let's say on paper and not on some electronic system, in order to fit the purpose, right? How to extract the data from obsolete device. As an example of industry initiative to enable a fair, aligned, and comprehensive semantic capture and translation of data is the SEED project that you can see on the right side of the screen. SEED comes from semantic enrichment of ELN data, and ELN is the electronic lab notebook. This project developed a new standard assay ontology in the phase 1, and now aims at filling important ontology gaps of interest for ELNs that are not currently covered in the well-formed public ontologies. Another challenge related with the digitalization is how to standardize the practices across the industry in a way that the data can be properly captured. And to be able to automate something, you need to describe the process. And there is a huge gap in the data standards in the automation area. And in Q3, in Q3 last year, we asked the community to highlight some of their biggest barriers in the digitalization process. And we identified as top 2 barriers the fact that the equipment is unable to communicate with one another and that there are no common data formats. So the participants proposed as next step to create some sessions on the different industry standards and their practical applications. And this is what we did. We created a series of webinars where we discussed about the adoption of open standards and why multiple standards are necessary condition for digitalization. We then saw the need of mapping all the process communication and data standards that are being used by the Lab of the Future community in an easy to understand format. So now what you see on the right side, the standards mapping across the R&D value chain, this is a current initiative, and we are gathering ideas on how this mapping should look like. It can either be an infographic type or it can be a decisional tree that will help the user to figure out which standard is relevant for their work and for their lab environment. Now, some standards exist and we can do the mapping of all of them, but others need to be created where there are gaps. And this can be done in 2 ways. One way is from bottom up. For example, coming from the research need of the organization to have some communication or process or data standards to fit the purpose, or it can be developed from top down, which means from the compliance, from the regulatory side. And as an example of an industry-wise initiative developed by the Pistoia Alliance and its members, we have the Helm notation, which is the hierarchical editing language for macromolecules, which is a system of standardized naming for complex biomolecules. It's widely adopted. It's used for representing therapeutic large molecule in the bioregistration systems. Helm is now an FDA recognized submission format, which also received an award of best practice from the bio-IT world. Here is another example of initiative that can help standardize the data across the industry. This is called the IDMP Ontology project. The IDMP comes from identification of medicinal products, and the terminology is being used in different domains, as you can see on the columns that are marked in blue, in the development, in the production, in regulation, authorization, and so on. The project is aiming at creating the ontology that will line top-down the IDMP ISO standards to the in-house standards of the organizations to reduce the current implementation inconsistencies at the organization level. And in this way, it can save time and costs. On the right side, you can see another example of a regulatory initiative, which is called In Vitro Pharmacology Project, where the FDA invited the Pistoia Alliance to help develop the data standards for in vitro pharmacology bioassay. The project just started, and the aim is to accelerate the submission process by defining the ontologies that will enable better data reuse, interpretation, and understanding. And all these examples and top-down standards that are being developed will be seen at the laboratory level in a period of time. But from a process perspective, these top-down standards are very efficient because they created a need of everybody to accept the same ontologies and data standards. Now, the Pistoia Alliance has also other communities of interest that are connected to the Lab of the Future, and we create also collaborations and partnership between these communities. On the left side, you can see the data governance community. This is managed by my colleague, John Weiss. In the middle, you see the AI/ML community of interest. This is managed by my colleague, Vladimir Makarov. And on the right side, you see another example of community, which is the UX community, managed by my colleague, Paula De Matos. So if we take one by one with the data governance community, we created a series of standards webinars. data governance is essential to enable digital transformation and to advance innovation. And this growing community already has more than 30 companies that bring together opinion leaders to identify best practices, common obstacles, and some potential solutions that we can all work together. The AI community of interest is aiming at discovering and promoting the best ways to use these new promising technologies, to apply them to solve the current challenges, and to educate the companies through presentations and discussions. And when implementing tools and software, AI, ML, an important aspect is to consider the change management and particularly the user adoption. So the dedicated user experience community, which is formed by practitioners from the pharmaceutical, healthcare, and software industries, is delivering frameworks and guides and toolkits to enable the businesses to adopt UX principles and methods. And their most recent initiative is to create concepts and ideas that will help the lab of the future community. Last few years, we saw rapid growth in terms of the number of new pre-competitive collaborations between the pharma companies and AI providers and software providers, and in general, the organizations from the R&D ecosystem. And in 2022, digital transformation continues to accelerate, and the pharmaceutical industry is seeing the benefits of non-competitive collaborations. And at the beginning, when the companies are learning how to assess their organization and the potential for digital transformations, we do encourage them to join different non-competitive platforms and groups to learn from the experience of the other companies. And same for the organizations that are the digital journey, because it's useful to contribute into and to help each other by sharing the use cases and best practices and lessons learned. And in this way, the organizations together can share both the risks and the benefits of the deliverables. Now, if you are not part of an organization yet, if you are a student or an early graduate, I do encourage you to join the communities of interest. Most of the discussions are open. You can join without any registration cost, and you can be more in contact with what is happening in the industry. I would say a recommendation to stay informed, to subscribe to different newsletters about your area of interest, to join different webinars and discussions, and very important to be proactive. So for example, if you see a challenge, discuss it with others, maybe look for some solutions. And if you see a challenge and a solution, you can join different working groups. You can reach out to different not-for-profit organizations that have the means and the contacts to validate the concept and to bring the right stakeholders at the table. I want to show you how at the Pistoia Alliance we create new projects. We are constantly developing new projects, and the process starts with the ideation phase, where the pre-competitive project idea is being raised. The idea can come from anybody. Then the idea is being validated with the community in terms of the benefits and the interest. And once there is a buy-in for that particular idea, it then starts to take a shape into a project that has a clear problem statement and the industry benefit. The outcome can be a member-funded project or a program. It can be a new formed community of experts. And this is the exact same path that we went on for creating the Women in STEM pilot program that I'm about to present you. So firstly, why is the Women in STEM topic important for so many organizations? You might not see a gender imbalance at the university or in your early career levels, but the gender imbalance in the top executive roles is a well-documented case, unfortunately. So studies showed that women are more likely to leave the STEM career pipeline than men, leading to a wider gap at the senior level So women account for only 3% of the CEOs in this field. And this is why the Pistoia Alliance has chosen to focus this pilot scheme on the leadership roles, because we believe that we believe in the top-down changes and in creating more role models for the future generations. And this was the process. We started in April last year at the Pistoia Alliance conference, where we had the Women in STEM panel with some very interesting discussions that continued even after the end of the conference. And by the way, some of the most interesting discussions happen before and after the presentations. So if you're a student or an early graduate at the conference, don't limit to the presentations, to listening to the presentations. Try to network and join the conversations that happen during the breaks or in the organized networking events and the poster sessions and so on. You can get a lot of valuable information from there and connect with the right people. So going back to how we started this program, in April last year, we received the feedback from the participants that really made us think of the different programs that we can initiate to help the women who want to start or grow into their STEM career. So in Q3, we gathered more interested parties and we asked the participants, what do they think are the biggest barriers for women in STEM? And the lack of childcare, maternity policies, depending on also the locations, but also the absence of female role models were mentioned as the barriers. But by far the greatest seemed to be the workplace culture. And we didn't go into more depth at that time, but we knew that the workplace culture is highly influenced by the leadership and the strategic organizational directions and the management. So what was also interesting in Q3 was the feedback that came from one of the only 2 men who joined the call. He experienced for the first time the feeling of being in the minority. And this feedback confirmed that in the Women in STEM program, all genders need to be involved. We agreed that any diversity effort needs to incorporate the intersectionality, which is the acknowledgement that everyone has their own unique experience when it comes to discrimination, and that gender can make people feel marginalized. And that is why we wanted to make sure that everybody is well represented and that men are included in all aspects of the program. In Q4, we started the different ideas on how the program should look like. And the most appealing idea to our members was the one to create a cross-company training and networking program, which is what we are piloting now with top managers from the pharma companies. The current pilot program brings together 40 participants from 12 countries, from 3 continents for a period of 6 months. We started in January, we will finish in June this year. We currently have 17 companies involved in this pilot stage, and each group of 10 participants includes 2 men as minority. The group meetings are held weekly. We have accredited trainer and coach, and we also have monthly networking sessions. Each group includes diversity in terms of the gender, race, location, role function. Almost half of the participants are from the US, the other half is mostly from Europe and China. And the Pistoia Alliance brought together leaders with different areas of expertise because we wanted to see different perspectives. And these are limited but not included, are including but not limited to the drugs research, development, data science, industry regulation, drug safety, and so on. And just to tell you that this Women in STEM pilot program, it's a first step in a wider diversity and inclusion in STEM leadership initiative. And we are aiming to run a series of future programs as of September this year, using the feedback and the learnings from this pilot program. We would like to form a community of leaders who share the same values and that want to contribute to the change in the overall industry, either by directly changing or by promoting equality in their organizations. If you want to find out more about the Pistoia Alliance and how you can get engaged, please do reach out to us on the website. And again, for students and early graduates, join the industry discussions, stay informed, ask questions, and be proactive. Reach out to us. Thank you very much.
Thank you so much, Anka, for such a wonderful presentation. I have a question. How do competing big pharma companies collaborate in the Pistoia Alliance projects?
Well, we facilitate a non-competitive environment. So, for example, we bring together projects or ideas or solutions that if the companies are working together, there's a benefit for the whole industry. There is no competitive advantage, right? So that's why we can bring different companies that even if, for example, in the day-to-day business environment, they are in competition, we bring them together with projects and initiatives that all of them can benefit. Because imagine that if, for example, a company has a big challenge, one thing is to try to solve the challenge by its own, which means investing resources, and resources can be hiring a lot of people to try to solve that issue, spending the money, the time, and so on. Or they can share the risk, right, and the resources with other companies that are facing similar challenges and try to solve them together.
Okay, got it. Yeah, thank you for answering the question. And the next question is, what is the challenge that Big Pharma is trying to solve with bioinformatics?
Well, I think it's also related with the data standards, and actually when it comes to challenges from the industry, we always ask the participants, and we want to understand what is the challenge and what we can do for that challenge, right?
Thank you. And do we have any more questions? Okay, we have one more question. What are some of the challenges faced in the current role as a strategic team lead, and how do you handle those challenges?
Well, the challenges that I see are mostly related with ensuring that we have all the right stakeholders at the table. So, for example, if somebody comes to me with an idea or a challenge and says, look, I need more people that would be willing to work together on solving this issue, right? For me, one of the challenges is, okay, I do have a lot of contacts. Who is the best person to come and work on this particular project? Or validating that a challenge that one company is facing, it's actually a challenge that more companies are facing. Because many times it happens that a company comes with a challenge that is not very sure if other companies are facing similar one. So then this comes the validation process that I'm doing, going and talking with different companies, seeing if that challenge is similar, and if we have an idea on a solution that we can work together to solve it.
Okay, got it. And our last question, Maham is asking, how can we join Women in STEM program?
So currently the Women in STEM is for the people that are in the top executive roles in the pharma, because the way we started this program, we saw that at the entry level, so it depends on where you are, right? If you are at the entry level in a career, there are similar programs for entry level, right? What we saw the need of doing the same in the top executive to make sure that the people that join at the entry level can have the same chances to advance in the career, right? So our program is targeted at leadership roles. And you can join by going on the website of the Pistoia Alliance, and you will see there under the projects, the program, and you can register either to join, either to be informed or with the newsletter information.
Okay, perfect. I hope, Maham, you have your answer and you can join. Yeah, thank you so much. Now let's move ahead with our next talk. So next talk is quite interesting as it's a special panel of researchers talk. So I'll just quickly introduce our panel of researchers. We have thus Dr. Julia Panna, and she'll be talking about analysis of single-cell transcriptome data that enables detailed understanding of multicellular tissue in health and disease. And then we have with us Mr. Avi Titevsky. He'll be talking about containerized pipeline as a stable and secure solution to bioinformatics analysis tools. And Dr. Eliza Veta will be talking about analysis of 16S rRNA metagenomics data and specially resolved transcriptomics. And last but not the least, Dr. Vadim will be highlighting manifold learning, T-bioinfo sections, and applications for the analysis of biological processes. So we have the recordings by Mr. Avi Titevsky and Dr. Vadim.
So, Anita, I will actually give the introduction. Thank you.
Perfect.
Okay, and everyone else who's going to participate, you can start your video. So I think we heard a very nice overview yesterday about different areas of bioinformatics that are very important to many people. We heard about the microbiome. We also heard about how important it is not only to have the data, but to analyze it and be interested in it. And so what are some of the questions that people are asking from such data sets? In today's sessions, we heard also about the single cell transcriptomics and other types of single cell technologies. And we heard about how rapidly this whole field is developing. What are some of the challenges with such big data? So this session that will be handled by the presenters from the Tauber Bioinformatics Research Center, I think kind of addresses a lot of these questions. So one of the questions that you might have is, as a new user who's new to bioinformatics, who's new to a lot of these methods, how do I use them with understanding? And how can I still apply them, even though I might not know a lot of the settings that need to be adjusted? We'll also hear about how integration of multi-omics data, in a sense, spatial single cell technologies, can be used in a simple interface, and how important they are for the future of some of these technologies that we've been discussing so far. And I think an important part of this as well, and we kind of started talking about this last in our previous day, how can you, once you understand the pipeline and once you understand what it does, how can you deploy it in an effective way? So there are a lot of technical challenges around pipelines, how to resolve technical issues, how to set up the environment, how to make sure that you have everything installed. And so the last presentation will also address how you can containerize, deploy these pipelines, and then provide an interactive interface for someone to explore the data in an effective way. So with this brief introduction, I'll pass it on to Dr. Julia Panna. She's the Associate Director of the Tauber Bioinformatics Research Center, and she's in charge of kind of the vision behind the platform that is being developed there, the T-bioinfo platform that we use extensively in all of our training. So over to you, Julia.
Thank you very much for the introduction. And we are really, really excited to be here. And thank you for the invitation. This is a great opportunity for us to show our work, which is mostly behind the scenes. So let me maybe share my screen. All right. Great. So we at Tauber Bioinformatics Research Center are a team of biologists, bioinformaticians, mathematicians, and computer scientists. And we developed this T-bioinfo platform where we implement open source and sometimes our own algorithms. And today we want to share with you some of these new sections that we have been working on. So the new sections that we will be talking about are manifold utility section, and I will go over some of the manifold learning techniques and what we can learn from them. And then Vadim, who is our software engineer, will walk you through the pipeline that we have been implementing for the last couple of months, actually. Then we will move to, actually, we decided in the last minute to skip the single-cell RNA-seq, and I hope that you will have the video next time. But we will jump right to spatial single-cell analysis. It's a new technology that allows us to measure the gene expression and at the same time know where the cells that are expressing gene profiles are located in the tissue. And then we will kind of move gears and we will speak about metagenomics analysis. Lisa Tararenko, who is our bioinformatician, will speak about both the spatial single-cell analysis and the metagenomics analysis. And finally, Avi, who is our software engineer, he will talk about the importance of containers in bioinformatics. So let me start with data, actually. And before we begin speaking about the manifold learning itself, we can talk about biological data in general. So before, biological data sets were quite small, and you could use simple statistics to investigate the data sets. But today, these days are probably almost gone, and we are living in a data-centric biology days. So our technologies allow us to collect huge amount of data about biological phenomena. For example, we can study the behavior of animal groups, relying on new tracking technologies such as biologgers, remote sensing. Also, for example, in the field of behavioral studies, we have new technologies such as fMRI, which allow researchers to quantify the behavior of neurons and their interactions across time. Other examples of current biological data sets include biopsy images and transcriptomic studies on the level of individual cells. And another example could be DNA sequencing, which allows to trace the virus population dynamics. And other huge data sets in biology and in medicine. So manifold is a topological space that is locally, so near each point, resembles Euclidean space that we're all familiar with. So if we think about manifold learning very intuitively, what we're trying to do is we're trying to unfold the manifold and position all points of this manifold on a low dimensional, usually two-dimensional space, so that the distance between nearby points is somehow preserved. And this procedure of unfolding the manifold helps us understand the patterns in the original data set. And actually the aim of this exercise is to uncover some hidden variables that can successfully expose the underlying structure of the data. And most frequently, these patterns are either discrete clusters, groups, or continuous gradients or trajectories. So all manifold learning techniques use three basic steps. First, we are trying to find neighbors, nodes, objects in our original space, highly dimensional space, that are somehow close or similar to each other. Next, we would like to measure distance between the neighboring points. And this measure of distance can be measured in a different way. So for example, we can measure Euclidean distance, or we can measure geodesic distance, Or we can use some other distance measures, such as time in a random walk. And there are others. So different algorithms in manifold learning actually use different distance measures, and that's how they are different. And finally, using partial eigenvalues decomposition, we will try to put this highly dimensional data graph into low dimensional space where steps 1 and 2 are preserved. So there are several popular approaches for embedding the original data points into Euclidean space of low dimensions, usually 2 dimensions, and for determining the underlying patterns in the data. And probably you're all familiar with principal component analysis, which is a linear dimensionality reduction technique, and it preserves the Euclidean distance between the points in the original space. The next highly useful dimensionality reduction technique is called isomap, and it seeks such mapping on the low dimensions that the geodesic distance now is preserved. The global geodesic distance of the manifold is preserved. Next would be locally linear embedding. And this local linear embedding seeks the lower dimensional projection of the data, which preserves distances within local neighborhoods. So it can be thought of as a series of local principal component analyses, which are then globally compared. The next one would be eigenmap or spectral embedding. And it attempts to preserve the local geometry by using the Laplacian operator. So in other words, calculating the change in gradient or the curvature across the manifold. Another approach is called diffusion maps. And this approach uses a very different way of measuring distance. It uses probabilistic measure of distance. The time it takes the random walk algorithm to find to go through the neighborhood graph. And finally, we will talk a little bit about UMAP, which is short for Uniform Manifold Approximation and Projection. And this algorithm uses entropy as a measure of distance. So we will show all of these approaches using 3 examples. Some examples are synthetic and other are biological, sometimes very simple and sometimes a little bit more sophisticated. So the first data set that we are going to talk about is a famous iris data set, which actually consists of 150 iris flowers, which belong to 3 different species, Setosa, versicolor, and virginica. And each flower in this data set is described by 4 measurements: sepal length, sepal width, petal length, and petal width. So, for example, if we plot this data set using 2 out of the 4 original dimensions, or we call them sometimes features, without using any dimensionality reduction techniques, we will see that one of the groups, Setosa, is always separated from the other 2 groups. So here on the graph, you can see that I put the groups on the 2 dimensions, sepal length and sepal width, and the blue group is separated. And the other 2 groups are mixed together. But this is only in 2 features. What would happen if we are trying to conserve all 4 features of the original data set? Well, the first thing we could do is just try to do the PCA. And PCA captures all features of the original data set, but it reduces the dimensions from 4 original dimensions to 2. And it preserves the Euclidean distance between the objects, so between my dots. And we see here that the Setosa flower is separated. The blue group is separated from the other 2 groups. However, the other 2 groups are not separated. And if we would color them in gray, you would see that it's almost impossible to separate these 2. So even though PCA works quite well for the blue group of flowers, let's see what other algorithms can give us and how we can try and understand the relationship between the flowers, between our data, based on the embeddings that we will see. So for example, if we use diffusion map, we will see, which uses the measure of time as distance. After the constructing of the graph, a random walk procedure is used to determine the distance or the time traveled between nodes on the manifold. And this technique is actually very sensitive to outliers because they are far away from other nodes on the graph, and you sort of get stuck there, and it takes a long time to move to the next node. And this is actually exactly what we see in the embedding. The blue group, which was very separate from the other groups, converges to one point and is very far away from the other ones. But also, interestingly, we see another small group of outliers, the orange outliers. So sometimes diffusion maps are a very good approach to find your outliers in your original data set. What about other ones? Here you see the embedding of the data when only two groups of viruses, the ones that weren't able to separate on the PCA, are present. And we see that spectral or eigenmap give us two trajectories, while UMAP gives the data, divides the data into two clusters. And both of these algorithms actually have a number of mixed up samples. Here is the in spectral and also in UMAP. So if we look at the on the spectral, at the part of the data set that was very separated, we can actually study these trajectories and how they depend on the X and Y axis using, for example, linear regression. And we would find that the blue trajectory, so the versicolor group, can be described by change across the X and Y axis. And the dependence there is a dependence of the location of each dot to the sepal length and petal width. And this means that the larger the sepal length and the petal width, the more to the right and to the bottom the iris flower actually is located on this graph. On the orange trajectory is dependent on three features: sepal length, sepal width, and petal length. And here, the larger are all three features, the more to the right and to the top the flower is located on this graph. Sorry, I forgot to put my Swiss roll animation, but Swiss roll is this figure, which is actually located in the three-dimensional space, and you can turn it around. So just to demonstrate this, we wanted to demonstrate this is a synthetic, totally synthetic, actually example of the data where the colors don't mean anything. It is just to demonstrate some properties of the embedding. So what you see on the right is the PCA embedding of this three-dimensional, if you can imagine it moving around. Here, if you could imagine this moving around like this. Oh, it's not working. Yeah, it is. Okay. So if you could imagine it just moving around, this would be the Swiss roll. For example, in the PCA, it makes a projection on the two-dimensional space and retains the Euclidean distance between the points. So for example, if we look at the blue points and the pink points, on the PCA, we see them as positioned very close to each other. And the Euclidean distance, so the distance in space between these points, actually was preserved by the algorithms, by the PCA algorithm. However, it did not preserve the geodesic space between those points, because we would expect, actually, if we were to unroll this roll, the Swiss roll, we would expect the pink points and the blue points to be very far away from each other. So PCA is not able to give us this structure of the manifold. But if we look at the isomap, and this is the isomap measures its distance as geodesic distance, here is exactly what we see. We see that the blue points and the pink points are very, very far away. What happens if we look at the same embedding, the same data set embedding by LLE or local linear embedding? And here we see that the local distances are preserved so that the nodes that were close to each other in the original data set are still close to each other, but the global geodesic distance is not so well preserved. For example, the dark blue and the light blue nodes are closer in the LLE embedding than they are in the isomap embedding. And now another example, biological example that is more close to real life would be the next example, where we have a data set of expression of 77 proteins in the brain of wild type, control, and trisomy model mice. And if we just project this data set on a two-dimensional space using PCA, this data is actually well separated. We see that the expression of proteins in the brains of control mice, the blue dots, is different from the expression in trisomy model mice. And already this result can be interesting, and we can explore which proteins are involved in the separation of the samples. But maybe we can find additional patterns in this data by using nonlinear embedding techniques. For example, examining the isomap embedding, which is based on the geodesic distance and not Euclidean as is PCA, we will find two trajectories or changes from one state to another. And if we investigate these trajectories using, for example, again, linear regression, we will discover that there is a dependence between the position of the blue object, wild type mice on the x-axis, and the expression of the protein called NR2A. So the higher the expression of this protein NR2A, the farther to the right we see the sample in this embedding. NR2A protein is a subunit of NMDA receptor and is linked to neuronal plasticity, to learning, and to memory formation. So you can imagine these mice furthest to the right having a lot of higher expression of this protein. If we will investigate the changes across the y-axis, we will find that they are dependent on the expression of another protein of a phosphorylated PKCG protein. And this protein kinase is expressed solely in the brain and in the spinal cord, localized to the neurons. And it has been demonstrated that this protein is involved in important neuronal functions such as long-term potentiation, long-term depression. And it is also linked to neurodevelopmental and neurological disorders such as autism and schizophrenia. So the higher the expression of this phosphorylated protein, the more to the higher is the sample of our mice. So isomap gave us an additional way to look at the data. Even though PCA separated the samples, here we find a very different trajectory and how they are related to the original proteins. Another embedding could be done with spectral embedding, and it also gave trajectories that could also be explored. On which proteins are they dependent? If we would look at the diffusion maps, it gives us another interesting pattern, namely that the control samples are all clustered together, so they are very similar to each other. And the trisomy mouse model is actually some kind of a gradient, a trajectory. So it changes across the y-axis. And we can also, again, study this trajectory and what it depends on. So if we look at UMAP, it is very different, and unlike the other methods that we have been looking at, UMAP, which is based on the notion of entropy, tries to make round clusters and separate these clusters as much as possible from each other. So again, we see that the control mice and the trisomy mice are very separately from each other, but instead of giving us trajectories, we see tight clusters. So the question here with manifold learning, I think the question shouldn't be which embedding is best, but rather our question should be which embedding or a combination of embeddings is useful for trying to elucidate hidden data structures and dependencies. So by using many algorithms and trying to look at our data from different perspectives, using different embedding methods, we can discover different dependencies and different structures of the data. So now maybe Vadim will show you how we implemented some of these algorithms on the platform and how you can use and play around with your own data. Vadim, maybe, or Ilya? I'm not sure what's going on. Vadim, maybe you can show us just or
Hello,
great.
my name is Vadim, and I would like to demonstrate how to visualize our complicated data easily. I show my screen. I'm glad to show you Tower bioinfo web platform start page. Firstly, we need to go to data integration and manifold and modeling zone when we choose the manifold utilities section. Now we are on the page where we can process our datasets with one of manifold learning approaches, which we talked about earlier, such as isomap, LE, spectral, UMAP, diffusion maps, and other. Here we can select the type of dataset. It can be object features table or distance matrix for graph, which will be used as pre-computed graph for manifold method. Let's get object features table for mice expression dataset and upload it to the platform. If we already know about classes of our objects, we can upload table with this information too. Or if you don't know about it, we can skip this step. Okay, we are ready to choose the approach, manifold learning. Let's start try UMAP approach. And this page shows us a small description of method and its parameters. Advanced users can change all of these parameters. Each parameter has description which we get by hovering the mouse or icon near parameter. Newbies in manifold learning can save default parameters and move to the next step with press save button. Now I'm filling the name of our pipeline. For example, test map mice 10. Okay. Now our simple pipeline is ready to start, and let's run it. We need to wait for a little time for the pipeline to finish. While data processing, we can look at input data format. In this data set, each row is expression values of 70 proteins. The classes of each object written in the following format. Column with objects ID and classes. Let's return to our pipeline. When pipeline will be finished, the email will be sent on my email. Okay, ready. Let's click download output files, and we can choose a PNG picture of our data manifold embedding visualization, or you can choose this TXT file with the points coordinates. Let's check a visualization. Great, UMAP approach works. We can see dividing of samples into two clusters, control samples and trisomy samples. Okay, it is all. Julia can.
Thank you very much.
You continue. Thank you.
Thank you very much, Vadim. So now we will have Lisa, who will present to us the spatial transcriptomics. Lisa?
Yeah, hi everyone. Can
Great.
you see my screen?
Yes.
Okay, great. Okay, so yeah, I'll be speaking about GWATA tool, the tool that is used to analyze spatially resolved transcriptomics, the method that was named the method of the year by Nature. Well, it was a great breakthrough when single cell was developed. Compared to bulk DNA, it allowed us to specifically study separate cells. But when we analyze data using single cell, we end up with something like this, just cells in an artificial dimensions. In this case, it's UMAP, but we never really know where exactly in our tissue these cells are located. And with spatial transcriptomics, finally, we have this opportunity to map the cells to our tissue. Like here, this example, this is the kidney, and this is normal kidney, injured kidney, and another injured kidney. And we can see that, for example, our cortex is comprised of a mixture of different cells that are located here, the glomerular or the collecting ducts. And our other cells, for example, proximal tubule cells, this third segment is located here. It's pretty much all the outer medulla is comprised only of this type of cell. But other than organs, we also can look at, for example, biofilm populations. This is the clustering of different functions that cells, individual cells in the colony do. And using spatial transcriptomics, it was possible to map them to the specific positions in the colony, which allowed to identify some hotspots for functions. For example, here we can see that this hotspot does the denitrification. Here we can see it's hotspot for high oxidative stress and so on. Of course, it's not the only application of spatial transcriptomics. It's used a lot in medicine, for example, in studies of cancer, because tumor is very heterogeneous and there are different clones. Some parts are evolving faster, and of course, we can see some differences in expression. Also, it's very useful to identify the invasive margin of the tumor and actually how the cells in this cancer interact with normal cells. And it was possible to build a bunch of expression atlases for healthy organs or the ones in disease. Also in molecular biology, it allowed to localize the individual molecules and of course study their expression inside the cell, for example, in cytosol or in nucleus. It also helped to understand better the chromatin organization and cell development. So there are different experimental platforms to study spatial single cell. We can separate them into microscopy-based methods like SECFISH, OSMFISH, or MERFISH, all the hybridization-based methods, and sequencing-based methods, for example, division. The difference is that for microscopy-based methods, since it's based on hybridization, we have a single cell or even single molecule resolution. And thus we can identify their locations if it's in cytosol or in nucleus or somewhere else. But we're restricted to only known genes and we cannot study any novel ones. And we also can't analyze too many genes at the same time because we're restricted in number of colors, or they can just basically overlap and we won't see exactly where this molecule is. Sequencing-based methods are easier and cheaper, and they don't really need a lot of pictures made on microscope to study the locations. And we also can study novel genes that we don't know the sequence of, but it's not always a single cell resolution. And we will be focusing on vision. That is exactly not single cell resolution, but basically how vision works. We have a plate like this, and the capture area that is comprised of small spots. And each spot already has the barcodes, the spatial barcodes that will tell us where exactly this position is located. So we place our tissue above the capture area, and we treat it so the mRNA will ligate to our barcodes. And then we just proceed to sequence, and we get a sequence like this. So after sequencing, we end up with our matrix where we have gene counts per cell and cell coordinates. And of course, we can get some pictures to work with as well. So with the data I just showed, we can start our pipeline, and spatial transcriptomics is actually very similar at the beginning. It's very similar to single cell analysis. It also has the pre-processing steps, the dimension reduction, different clustering algorithms, and different algorithms for cluster-specific marker gene identification, cluster annotation. But Giotto also allows us to check, to play with clusters, to separate them or to merge them based on their similarity, and run enrichment. And this module, the spatial analysis, is what is really different and interesting for us. It starts with building a spatial network, and then we can find spatially variable genes that have some spatial pattern and study co-expression and cell neighborhood. So I will focus only on this part. So we already did all the pre-processing and single cell analysis and end up with our artificial dimensions. In this case, it's UMAP. And we color them with latent clustering. In this case, we have 11 clusters. This is kidney sample. And what is different from single cell at this moment is that we already can map our cells on our tissue. And we see here that, for example, clusters 6 and 7 comprise the inner medulla, and classes 2 and 9 comprise the outer medulla, and the rest of the classes comprise the cortex, and it's really a mixture of cells. What we can see here is, for example, we can plot how many genes are expressed by which part of our tissue, and we see that medulla is expressing a lot more transcript than the cortex. So the first step in spatial analysis is to plot a spatial grid or network that will allow us to calculate the relationships between these parts. In case of grid, this is the expression will be summarized inside the grid. And in case of network, it will remain as a single cell or single spot resolution. And this allows us to run spatial gene search. And I wanted to show you an example of two genes, NAPSA and GPX3, which are both marker genes of proximal tubules. But you can see on these plots that they actually have completely different spatial expression patterns, right? We can see GPX3 expressed in the cortex and not expressed in the medulla, and NAPSA is expressed in outer medulla. So comparing it to already known cell types in the kidney, we can see that actually GPX3 is a marker gene for proximal tubules, but segments 1 and 2, which are located in the cortex. And NAPSA is more correlating with proximal tubules, the segment 3, which is located here in the outer medulla. So spatial analysis can help us understand marker genes better. Also, the next thing is spatial co-expression. So this actually allows us to identify the metagenes, a group of genes that are sharing same patterns of expression. And for example, for this data set, 8 metagenes were identified, and here we can see the correlation between these 8 metagenes. And here are the expression for the metagenes on our tissue. And you can see that they all have different patterns of expression. Some are specific to inner medulla, some are specific to outer medulla, some are specific, well, not quite, because here's a mixture of cells really, but they're specific to the cortex. And we can go from the metagene level to single gene level if we want to. And one more, maybe the most interesting thing, is how the cells interact. And in this case, it shows how really, well, the first step is to find the interaction changed genes. So the genes that actually change when their expression changes when there's interaction with different cells. And based on these genes, we can find cell types that are interacting within this cluster or with other clusters. And we can see, for example, that clusters 4, 5, 8, 1, and 3, they all are here. They aren't really correlating even to each other very well. And for example, cluster 10 and 11, these are small ones. They're located here and here. They have very strong interaction inside the cluster and not so much with other clusters. Well, they interact with each other as well. So a spatial single cell really gives us a lot of opportunities to study pretty much anywhere we can apply single cell. We can try applying spatial single cell, and it will give us more information at where exactly this function is happening, and a lot of information about expression patterns, spatial expression patterns. So that's it on the GOTO. If you have any questions, I think you can type them in the QA section if there is one here. And I will move on to amplicon sequencing, right, Julia?
Yes, thank you very much for this introduction to the spatial.
I think we do have. I think we do have a couple of questions if we can answer them.
Okay. Yeah.
Yeah, so the first question is, how do you select between various manifold methods when analyzing complex data, such as spatial single cell?
So I think, yeah.
Yeah, sorry.
You want to answer? Take that, Lisa, sorry. You can take that.
No, I mean, how I choose, I just look at the data, I guess, and see if it makes sense or not.
I would also suggest that you can use, there is no right embedding. So and I think it's an important issue where it's an important thing to remember, that you're not trying, each of those embeddings is going to be somehow right and somehow wrong. It will preserve some of the hidden structure of the data, but it will lose some other hidden structure. So by using many different methods, you can find different, sometimes it's trajectories that you're interested in, sometimes it's clusters that you're interested in. So, so we always try to recommend using as many methods as you can to look at the data from different sides. So also such complicated data as single cell or spatial single cell, the UMAP is actually most, if you look at all the articles, you will see that most of the people use UMAP or TSNE, but there are many, many, many more embedding methods available, and we recommend to really look through at least a few, because some of them, they're based on different things, and then they also will reveal different underlying structures of the data. So I hope that helps.
Great, thank you both. Another question, will spatial single-cell RNA-seq make single-cell RNA-seq obsolete, without imaging data obsolete?
Can you please specify what obsolete is? Sorry.
Yeah, so will single cell RNA-seq without imaging become irrelevant with the introduction of spatial single cell?
Well, I think that there are options when we don't really need spatial imaging or we can't really capture it. Well, looking at the pipeline, yeah, it looks like all the steps we do the same as single cell, so we can replace all the single cell projects with spatial single cell. And I think that's a good idea, but still, I guess there are options when we can't really use spatial single cell, so we can continue working with only just regular single cell.
Also, if I could add to that, if you are using Visium, then as Lisa has mentioned, you are not actually capturing each individual cell. You are capturing a small well that has a few cells in it, right, Lisa? So probably at least for now, single cell is still the resolution of the single cell RNA-seq is probably better than at least this Visium. And another important thing for science is a matter of resources. Spatial single cell is a very expensive technique. We cannot imagine that all of our studies will become all of a sudden only single cell and only spatial single cell. It will be just too expensive to do science this way. So for a well-identified project, we hear prices around $100,000, $200,000. So not every lab can actually afford this.
Actually, one more thing, Julia made a good point that Visium is not single cell resolution. There are studies when just regular single cell and spatial single cell are combined together. So you get the spatial information, but at the same time, you can kind of get the single cell resolution. So yeah, I think single cell won't go anywhere. We'll use it.
And if to add a little bit to that, a lot of people ask us if now we have this technology that allows us to do single cell, will bulk RNA-seq be obsolete? And the answer is probably related, probably not. There are more and more computational ways of combining single cell data, which is very sparse. So you have a lot of genes that appear to be not expressed at all, but in reality, they are expressed. So the data is very sparse, and you combine it with your bulk RNA, which is much more stable, but has this average cell that is never actually exist. And you do something like deconvolution and use other computational techniques to try to connect those two together. So I think it's the same kind of question.
Great, thank you so much. All right. Now I
just
can
a minute.
share.
I think now we will have Quine. And Lisa,
yes, I was going to show the recorded presentation. Is that correct?
No.
No. It's a different one. Maybe yours is the next one. Sorry. Can you see my screen?
Yeah.
Okay, yeah. I'll speak about our platform section for CHIME. CHIME is a tool that analyzes metagenomics data, the culture-independent microbial communities. What we basically learn about them is composition and function of these communities. And it can be applied in a bunch of areas like agriculture, where you can study soil properties or any bacteria-driven processes. In healthcare, where we can study microbiomes of different organs, or ecology, where biosensors are developed using metagenomics, and biodegradation of plastic is also a bacteria-driven process. And of course, biotechnology, the nutrition produced or pharmaceuticals or biofuel, everything is done thanks to bacteria. So basically, when we sample our data and we extract DNA and then we want to amplify it, we can go two ways. We can either go whole genome sequencing way or target sequencing way. And in case we want only to sequence the target, 16S ribosomal RNA is the region that mostly is targeted in bacteria because it has both conserved and hypervariable regions. Actually, there are 9, just I didn't have a picture good enough for to include it. But these hypervariable regions help us differentiate between different species. So 16S or target sequencing or amplicon sequencing, it's cheap compared to whole genome sequencing. It's pretty standard. We already know all the primers, so it's easier to conduct. But it's restricted to previously known and annotated taxa, and sometimes it's not enough to differentiate between the samples. Also, we only get the compositional information. We don't know any function that are done here. Whole genome sequencing allows us to study novel organisms, and we can assemble the reads in, and we can run functional genomics and understand what function this organism does. It's also more information to differentiate between the taxa, but there are a bunch of problems to whole genome sequencing of bacteria because it's expensive, it's hard to understand what is contamination, what is not, and so on. So on the platform, we present 3 modules, pre-processing that consists of filtering and denoising algorithms. Then we go on to taxonomy with silver classification, filtering our classified sequences, and conducting phylogenetic trees. And then we can move on to metrics to calculate for alpha and beta diversity. So at the very beginning, we start with the quality control, and it's very important not to include regions of low quality because they will interfere with all the downstream analysis with our abundances, with our phylogenetic tree and taxonomy and everything. So you have to understand where you want to trim your reads like this, so you will not include low quality nucleotides in your downstream analysis. So after filtering the replication, we come to denoising or clustering techniques. So the idea is that when we first sample our data, we have one sequence per species or something like that. But when we run amplification, there are bound to be some errors, and we get small fractions of sequences that are slightly different from our original one. And when we sequence, there are even more errors and more unique sequences. And so we end up with a plethora of unique sequences, and clustering algorithms would look for the most abundant sequence and assign it as a center and cluster everything that is 97% similar in one cluster. Well, there are a few problems with this approach. And first is that not all the species are 97% similar. Some are a lot more similar. And researchers came up with another approach, denoising, which was taking into account different models of errors and sequencing quality calls to try and correct the errors, the mistakes, and end up with haplotypes. But these haplotypes are 100% similar. So after denoising or clustering, for paired end reads, there is also option to merge, well, not an option, it's an obligatory step to merge the reads. Since even for targeted sequencing, we can pick different size, different regions, and the size of region may vary, and sometimes the reads are separated like this, sometimes they overlap, and for example, DADA2 treats them a bit differently. In case they overlap, they only take the part that overlaps. In case they overhang, it will take one of the reads. And if they're separated, it will concatenate the reads together. So when we sampled our data, extracted DNA, amplified it, and did denoising, we end up with a table like this, where we have counts of each unique representative sequence per sample. And here we can ask ourselves three main questions. First is, how similar or different the things are in one sample? And that will be alpha diversity. Second question is, how similar or different are the samples between each other? And that will be beta diversity. And the third is, what specifically leads to differences between the samples? And that will be taxonomic profiling. We'll start with the third one. So for taxonomy analysis, it is strongly advised to use the same primers that you used for your data. So the same region will be extracted from the database. Here we use Silva database that is being updated and curated. And you can see the mostly used primers here and primer sequences. So you just take the sequence, add it to the platform, forward and reverse primers, and they will extract the exact regions from the database and run classification. So it will look for if the sequence is in the Silva database, and it will assign taxonomic unit to the sequences. And you will end up with a bar plot showing what bacteria are there in your samples. If you want some of them removed, you can exclude them. Just type the correct name of the taxa, or if maybe you want to include just several samples, you can do the same. And you can also group the samples based on some categorical columns in your metadata. For example, I was interested in body sites to see if samples from gut were any different from palms and tongue. So after taxonomy analysis, we go on to phylogenetic tree. So this is this will show how similar our representative sequences are, and it starts from multiple sequence alignment, and then there are a few algorithms are available to build the tree, fast tree, and IQ tree. This is mainly used for core metrics, but you can also extract the tree and visualize it in, for example, Fig Tree or any other tool that helps visualize phylogenetic tree files. So after that, we have, well, looking at this table, the artificial, I just came up with it to show. Looking at this table, we see that total counts for each samples are different. And in case we can compare sample 4 and sample 1, they're pretty close in total counts. We can't really compare sample 3 with others, right? So because they have different sampling depth, and we can't really run any reliable statistics on something like this. So one of the ways out is to subsample the frequencies so that the total count will be the same for all the samples, like here. So we subsample all the table and change the table so that the total will be the same. This is not ideal because we'll lose information. Sometimes we even lose whole samples if their sampling depth is lower than what would peak. But this is a good way to normalize your samples, so the statistics will be reliable. But how actually should we pick the value? Well, for example, how did I pick 100? We should be looking at our samples and their sampling depth. For example, here, we can see that some samples have sampling depths lower than 1,000. Some have sampling depth a bit less than 2,000. some are here, but the majority are having bigger sampling depth. Also, we can see that the number of features are quite stable starting from here. So it looks like this region looks appropriate to take the sampling depth. But it's also very meaningful to look at your groups and if the samples in your groups of interest are still there. For example, if I take sampling depth of 2000, my right palm sample group will be will consist of only 2 or 3 samples. But if I take sampling depth 1000, my right palm group will be okay. I can still work with it.
Excuse me, I'm sorry to interrupt. We're a little bit running over time, and I know you still have a few slides. Would you be able to maybe wrap it up in a couple of minutes because we have some others on the schedule? Thank you.
Yeah. So yeah, the only thing that is left is core metrics, basically calculating all the metrics for alpha and beta diversity using the sampling depth that we just identified. And it will give us an idea. So the alpha diversity will give us an idea how things are inside the sample, basically telling us how rich and even our samples with different indices, Shannon index, Filo index. And beta diversity will give us an understanding how samples are between each other, plotting PCA based on different distances like Jakarta or Bay-Curtis. And we also can run Prema test on a column that we are interested in. In this case, it was body size. And I got really good p-value, meaning that this group has very different beta diversity statistics. That's it. Thank you very much for your attention. And yeah, here I just mentioning some studies that were done using amplicon and whole genome sequencing of metagenomics. Thank you.
Thank you very much for that presentation. That was great. So in the interest of time, I think we have a recorded video from Mr. Avi Tidovetsky, and we are going to actually share that on our YouTube channel, the link to that video on our YouTube channel, and move forward. And the next speaker is also going to be a recording. So it is my pleasure to introduce Dr. Tony Jia. Dr. Jia obtained a BS degree with honors from Caltech in chemistry and business, economics, and management, and in 2010, and PhDs from Harvard Chemistry in 2012 and 2016, respectively. Since 2017, he has been a researcher at the Earth Life Science Institute at the Tokyo Institute of Technology. And in 2019, he became a specially appointed assistant professor and lab manager at ELSI, an affiliated researcher at Blue Marble Space Institute of Sciences. His current research interests include origins of life, astrobiology, and primitive compartments. So here is Dr. Jia's recording on the origin and evolution of biopolymer and non-biopolymer based compartments.
Good morning, good afternoon, and good evening to everyone. Thank you for joining the symposium and thank you for joining today's talk. Unfortunately, I'm really sorry, but I can't be there in person. Unfortunately, the way time zones work, it's not so great, but I hope that this video presentation will be interesting and exciting to you. And of course, at the very end, I'm happy to take questions by text, by email, by chat, or any other way. So today I'll be talking about the origin and evolution of biopolymer and non-biopolymer based compartments. So first of all, I'd like to start with a little bit of a self-introduction. So I actually did my bachelor's degree at Caltech, where I studied chemistry and business economics and management. And part of my research involved studying chloroplast protein expression in crystallography, as well as using mass spectrometry to study pulmonary surfactant lipids. Next, I moved to Harvard where I did my PhD, and I studied mostly RNA. I studied RNA sequencing technologies, specifically used for non-coding RNA quantification. And I also studied ways which RNA could polymerize and replicate without using enzymes, so-called non-enzymatic RNA polymerization. Because I had so much experience working with all of these different biomolecules, lipids, proteins, nucleic acids, I got really interested in how exactly those biomolecules emerged and evolved in the first place. And that led me to my current position. So since 2017, I've been a researcher at the Earth Life Science Institute at Tokyo Tech Institute of Technology. And also since 2019, I've been an affiliated scientist at Blue Marble Space Institute of Science. And since then, at ELSI, I've been working on a variety of different topics related to origin of life and astrobiology, but also related to other interesting topics such as DNA liquid crystals or continuing with some RNA sequencing technology work. So before I get into today's main part of the talk, I'm going to turn off my camera in case there's, in case the portrait interferes or blocks some of the content. So today I'd like to go through 4 different, minor topics. So in case you aren't familiar with what exactly is astrobiology, especially in the eyes of a pre-biotic chemist, I want to introduce and give maybe a little bit of a review about primitive cells and what they might look like, and then give 2 short case studies in origins of life research featuring different polymers. One is from a biopolymer-based background, one is from a non-biopolymer-based background. So hopefully you can see the similarities and differences of those sequences and those systems that we study. And finally, I want to introduce some opportunities for students and researchers to participate in astrobiology research, both at ELSI, my current affiliation, as well as outside of ELSI. So what exactly is astrobiology? I think many people have different concepts of what exactly is astrobiology. For example, if you take a look at the word astrobiology, you would think that it's the intersection of astronomy and life science. And basically it is. Astrobiology takes concepts from astronomy, takes concepts from life science, and what astrobiology is encompasses many different things. So what I think, and from a prebiotic chemist perspective, what I think astrobiology research encompasses includes, for example, the active search for life in space. This is, searching for current life or former life, and often this is through missions. For example, in Japan, there's a Hayabusa 2 mission, which went to get samples from the asteroid called Ryugu. It recently returned to Earth a couple years ago, and they're studying those samples right now. And in a few years, JAXA will be sending a mission to one of the Martian moons and to study kind of the structure of the Martian moons and bring those samples back. So that's one aspect. Another aspect is searching for habitable planets in space. So for example, searching for exoplanets, probing the habitability of extraterrestrial bodies. And this can be done, for example, through space-based telescopes like the James Webb Space Telescope shown on the left, or through these radio telescopes that are on Earth. And this is ALMA on the right. Next, we also have the active search for the origin of planets, Earth, and life on Earth. And so this includes the search for organisms that live in extreme environments, which might provide a window into what life was like back on early Earth, as well as research on planetary and geological formation. And this also involves studying, areas of the Earth which might simulate areas that one could find on other planets. And these 3 last topics are something my research has a hand in. So the search for how to make synthetic living systems in the lab, how primitive chemicals assembled into primitive life, and finally, how this primitive life evolved into modern life. So this is the topics that I'm personally researching. It's part of this, subfield of astrobiology, which is origins of life field. And it's a really small part of the field, but extensive collaboration is really essential in order to really understand aspects of the origin of life, and especially collaboration with different types of researchers, and even researchers or those with an engineering background or people from non-scientific backgrounds is really essential. So coming from kind of a biochemistry or a biology background, I'm sure many of you are really interested in how the cell works, what the structure is. And so, this is a simplistic cartoon of the cell, but essentially it's built up of many complex parts, and researchers have really had a lot of success understanding many of the structures and what they do and what they're made to do, or maybe during diseases when they have trouble. And we've learned a lot about the cell in the last 20, 30, 50 years, but there's still many unanswered questions. And one of the big unanswered questions is, what did the first cells look like? How did these first cells function? And some researchers believe that something like a more primitive type cell existed on early Earth. Some people call this a protocell. And maybe this included some type of compartment boundary, some type of replicating genetic system, housed, catalysts like enzymes or primitive enzymes, and then also could participate in evolution, growth, and division. And so one way to think about origins of life research is we want to understand how the chemicals on early Earth in the prebiotic soup, if you will, assembled into these primitive compartments or protocells, and how these protocells functioned and eventually evolved into modern cells. And specifically, if we think about this, a non-biomolecule or non-biopolymer-based prebiotic soup, how could this eventually assemble and evolve into a biomolecule or a biopolymer-based modern cell is a really interesting and big question. And in order to do this, one way is to try to see or understand what this primitive cell looked like, what it could do. And we can approach this in two ways, going from the bottom up, building a primitive compartment using these non-biomolecules available on early Earth through prebiotic chemistry, or we can go from the top down, taking what we know about biology and kind of working backwards to see what minimized functions or what minimized types of cells could have existed. So what I like to say is we want to build this primitive compartment from both directions. And so let's say going from top down, I think this is something that more of you in this audience are familiar with. Let's see how this primitive cell would have looked like if we work backwards from a modern cell. And forgive the simplicity of this cartoon as well. This is one of the processes we see in the modern cell. It's, as you know, the central dogma. And these are composed of different processes made up of biopolymers and biomolecules and catalyzed by biomolecules. So what we want to know is on early Earth or in an early cell, which of these processes were still essential. So for example, it's very likely that proteins did not exist on early Earth. They're too large. Their folding is too difficult to have achieved. And also it's very likely that a DNA couldn't have existed really on an early Earth. So this leads many to postulate that the first cells on early Earth contain RNA. And this is specifically leads to a origin of life hypothesis, which suggests that RNA is the first and most important biomolecule of life. And this hypothesis is called the RNA world hypothesis, and it postulates that because RNA can store and pass down genetic information, because it's similar in structure to DNA, and also because RNA can fold into specific structures that can catalyze chemical processes in the form of ribozymes, similar to proteins, that RNA was the first and most important biomolecule on early Earth. And while there's been a lot of research in this field and a lot of progress has been made, there's still a number of outstanding questions in the RNA world. For example, how the RNA could be synthesized, how it polymerizes, how it's prevented from hydrolyzing. There's many questions that researchers are answering to this day. And there are also hypotheses that RNA wasn't the only biomolecule that existed on early Earth. For example, some researchers have postulated that DNA and RNA could have coexisted on early Earth, maybe in a hybrid form. And based on the structure of the ribosome as we know it today, it's also been postulated that perhaps RNA and peptides co-evolved So instead of a purely RNA world, you might have had something like an RNA peptide or an RNA protein world. And so what all of this is just saying is that we're not exactly sure exactly what was in this early cell. And it's plausible that all of these or even more types of molecules existed in or around these cells. So let's assume from now on that we have access to a number of different types of biomolecules for building this primitive cell. The next thing I want to focus on is this boundary. So this boundary is really important and defines a cell from other cells. Specifically, it makes the cell defined as a compartment. And compartments are really essential to origins of life. Compartments segregate and concentrate reactants. They promote chemical reactions, promote evolution, prevent fusion of genetic materials, many different important functions on early Earth. And modern cells, they're made up mostly of a phospholipid membrane. That's the boundary. It's been postulated that perhaps early cells that are membrane bound were made up of fatty acids rather than phospholipids because they're much easier to synthesize using prebiotic chemistry and what's available back on early Earth due to there being only one chain rather than two. And so one further question is that can you actually make a boundary that's even simpler than fatty acids that doesn't even require that type of bilayer? And so what we're interested in is seeing whether such types of compartments could have existed and whether they could form or function as early cells or protocells. So one way is through liquid-liquid phase separation. For those who are familiar with this, you might have heard about this. If you haven't, it's actually one way to form droplets in solution. These droplets are membraneless, but they're still liquid, and they provide compartmentalization without a membrane. They allow exchange of molecules between phases, and in fact, they're present in modern biology. So there's a phenomenon called biological phase separation, which results in formation of membraneless organelles in the cell. These organelles, there's some of them are called, for example, nuage granules or stress granules. Maybe chromatin is even considered one of these. And these are membraneless organelles in the cell that are really important for controlling biological processes. And it's been postulated that because most cells or almost all cells have this type of phase separation inside to make these membraneless organelles, that perhaps they're an ancient invention and that phase separation was also used to make compartments on early Earth. And so here, let me move on to a couple case studies of origins of life research. And let me start with the biopolymer case. So this is a case study on primitive liquid crystal coacervates. So you might have heard of liquid crystals before. They're present in TVs or displays. And in fact, you can actually make liquid crystals out of DNA. So this is in collaboration with Tommaso Fraccia, who's a researcher at ESPCI in Paris. And so his previous research showed that if you took short DNA oligomers that were self-complementary, such as this sequence shown here, they can actually form these duplexes that aggregate end to end. They stack together and they form these long rods. And these rods will bundle together. And after drying, after increasing the concentration of the DNA significantly, the structure that results is this columnar liquid crystal structure shown in this polarization microscopy image on the right. So these structures are somewhat liquid. They have liquid and solid characteristics. And you can visualize them through polarization microscopy because of their supramolecular chirality. So we postulated that perhaps it would be interesting if, for example, a primitive compartment, like one of those phase-separated systems, could actually house one of these liquid crystal structures because they've shown the ability to affect electrochemistry or regiochemistry or regioselectivity. So what we did was we knew that, when nucleic acids like DNA and when peptides, like cationic peptides, are mixed and bind together, they actually can form these liquid-liquid phase separated structures. So we took PLL, polylysine, about 250 residues long, polydisperse, mixed it with high concentrations of this DNA oligomer. And we were able to find conditions in which these structures, these nice membraneless droplets or coacervates, co-localize with this DNA crystal, liquid crystal structure shown on the bottom through the polarization microscopy. So we believe that this DNA liquid crystal structure is formed through this end-to-end stacking interaction of DNA. And we actually found that these structures, the co-localized structure, the liquid crystal coacervate, only exists in a very narrow range of salinity. For example, when we varied the salinity, we found that at low concentrations of sodium chloride, precipitation occurred, whereas at higher concentrations of sodium chloride, only droplets formed without liquid crystal characteristics, or no droplets could form at all. And we believe that this is because the ionic strength of the solution is really important for this interaction. If there's lower amounts of salt, the peptide DNA binding interaction may be too strong, forming precipitation or solid-like particles. Whereas at higher salt concentrations, it makes these structures, these interactions between the peptide and the DNA weaker, suggesting that, you know, when the peptide and DNA can no longer interact, for example, at 1 molar sodium chloride, these droplets can't form as well. We next wanted to test our hypothesis as to whether this end-to-end interaction is indeed what's forming these crystal structures. So we did the same experiment, but with a slightly different DNA sequence. This time we included a 2T base overhang so that this stacking process could not occur. And we found that through all of the different conditions we tested, in no single case did we find liquid crystal structures. We did find many conditions where these droplets could form, these phase-separated droplets could form, but liquid crystals could not be observed. And this shows that this end-to-end stacking interaction is really essential for these liquid crystals to form. We then next wanted to study the what happens if we subjected these liquid crystal coacervates to processes or environmental processes that might have existed on earlier. So one of them is dehydration. It's been proposed that dehydration could be one way to effect different chemical reactions on early Earth. You'll see this in the second part as well. And so we subjected these systems to drying. And so what you see here is that throughout the drying process, you can see these structures go from these solid-like structures transitioning to these liquid crystal coacervate-like structures to just a simple coacervate and then to a disassembled structure. I'll play this video again. And we believe this is the case because similar to the salinity titration, dehydrating the solution actually up-concentrates the sodium chloride in the solution perhaps, which suggests that this phase transition follows a similar phase transition as what we observed in the sodium chloride titration case. Next, we started again and we wanted to see the reaction of when we heated these samples. So we actually heated these samples up to about 60 degrees and we can see a transition from this solid-like structure to a liquid crystal coacervate structure. Furthermore, further heating resulted in disassembly of these liquid crystal structures, resulting only in these non-liquid crystal coacervates. And then upon decreasing the temperature, we noticed that at 40 degrees on the way down, these liquid crystal coacervates started to pop up again. And we saw a few different types of liquid crystal coacervates, but also we saw that the population was also heterogeneous. Some of them were still typical droplets without liquid crystal characteristics. Some of them had liquid crystals inside. And we found that upon decreasing at further to about 10 degrees, that almost all of these droplets reconstituted this liquid crystal character. If we compare between these before and after heating, the structures are pretty different. And we think that this is because this heating process is basically akin to an annealing process. And of course, annealing is used to allow in DNA and RNA case to allow the strands to bind completely to find the most stable thermodynamic conformation. And we believe that in this case, perhaps the right figure here is this is what the most stable conformation is for this system. And so we believe that what's happening is we have these liquid crystal structures here with increasing heat and salinity. You have these long DNA rods in teal and these red strands of peptides bound together. The rods, the DNA rods, are in this packed hexagonal pattern. Upon increasing heat and salinity, these rods start to come together, come apart, causing some structural change. And then the rods start to disassemble these into, destack. And so you lose this liquid crystal characteristic, but you retain the droplets. And then further increasing heat and salinity results in disassembly of everything, and you result in no droplets. So for those who are interested in learning more details, some of these studies are published. You can see one here and one here as well. I'd highly encourage you to take a look if you're interested in the details. Next, I'd like to talk about our other side, non-biopolymers side of organs of life research. And here we focus on membraneless polyester droplets. So here we're building from the bottom up, and you see we're interested in building this primitive compartment from the available chemicals on early Earth. And it's actually known that there's many ways by which biomolecules could have formed on early Earth, through meteorites or atmospheric discharge. But we also find that together with biomolecules, that there's also many non-biomolecules synthesized at the same time. For example, Murchison meteorite has tons and tons of molecules inside. And if you analyze a Miller-Urey reaction or atmospheric discharge reaction products, you also get tons and tons of different molecules. Not all of these are biomolecules. This suggests that non-biomolecules were just as or even more abundant than biomolecules on early Earth. So we're interested in understanding how biomolecules could catalyze, how non-biomolecules could have catalyzed life's emergence. The system we like to use are alpha hydroxy acids. They're monomers similar in structure to amino acids, but with an amine group that's replaced by a hydroxyl group. And what's great about these is that through simple drying, these monomers can form polyester polymers, shown on the bottom here. So we wanted to create these polymers, these polyesters, and see if they could, assemble into something useful for the origin of life. So we first took monomer solutions and dried them to form polyesters. And here's some mass spec data, and you can see that in all cases, the monomers were able to polymerize into polymers. Let's zoom in again. This is one of the monomers, lactic acid. So you see that the products are polydispersed. They're not a single product. So you make many different lengths of polyesters. And so you also form this gel-like structure. So this was observed in almost all cases where all of the polymer gel structures were formed through by these polydispersed polyester products. So we took these gels and we dissolved them in aqueous solution. And what we found was that droplets could form. They were turbid, and we could see these nice spherical droplets in solution for almost all of these structures. We actually took these droplets and we acided them a bit further. So we first found that these droplets could encapsulate functional superfold GFP. We also found that these droplets could catalyze or scaffold the assembly of some type of lipid structures around the droplets. And finally, we found that RNA could segregate into and function in the presence of these droplets. So one of the next questions we tried to pursue was that, only one type of polyester droplet we found could compartmentalize RNA, but these are all neutral droplets that we studied. Can other polyester droplets or droplets made from other compositions compartmentalize RNA? And we hypothesized that if we included a basic alpha hydroxy acid, something that could be positively charged in solution, the droplets that contain this positive residue could allow the RNA with a negative backbone to segregate more strongly than before. And indeed, we incorporated some percentage of these cationic residues. We still saw droplets, but we found that increasing the amount of cationic residues actually resulted in dissociation of the droplets. And we actually believe that, for example, at really high concentrations of the cationic residue, it's very likely that the droplets are too polar and too soluble in water, or they're too charged and too soluble in water, so they don't form the droplets. But at lower percentages of the cationic residue, we still see droplets, and it's very likely that these droplets are still cationic. So it's likely that the polyester formation is governed by the total charge of this mixture. And in fact, we did find the ability for these new novel, cationic droplets to segregate RNA. You can see here on the bottom that the fluorescent RNA is really strongly segregated into these droplets here. And we found that even increasing the amount of charged monomers into a neutral polyester by only about 20% allowed polyesters, which originally when in the neutral form couldn't segregate any RNA, to be able to segregate some RNA here shown in the middle and the right. So even just a small amount can change these structures. And so again, if you're interested in learning more details about this study or these studies, some of them have been published. I would highly encourage you to read some of these papers here for more details. So I'd like to, of course, thank my funding sources. A lot of these funding sources came from Japan, but also one from the French Embassy in Japan. I'd like to thank some of my collaborators that have worked on this project with me before, especially Irina, Jim, Kuhan, and Tommaso, both at ELSI and outside of ELSI. And I'd like to thank other collaborators at ELSI and Tokyo Tech, especially Chen and Rihanna, who recently have been working with me on some other really exciting projects related to polyesters. And also a lot of my collaborators around the world for all these projects as well. It's been a great experience and I hope to continue working with everyone. So finally, I'd like to give a few, programs that you might be interested in to get into astrobiology research. So Blue Marble Space Institute of Science has a young researcher program over the summer. If you're studying in university or recently graduated and are interested to do a virtual project over the summer, you can take a look. There are different projects of different types, and some are research-based, some are outreach-based, some are communication-based. And so you can take a look at the website shown here. My main affiliation, ELSI, we're a research institute located in Tokyo, and we study the origin and evolution of Earth, planets, and life on Earth. And here's some other pictures here. We've recently started a 5-year integrated master's PhD program, and this program is open to both domestic and international applicants, and you will be fully funded. If you're interested, the next deadline for fall 2022 enrollment is mid-April. You should visit this website here. Hopefully you'll be interested in this. And finally, last year, I was part of the organizing team for a conference called AbGradCon, which is Astrobiology Graduate Conference. It's an annual conference for students and postdocs in or looking to get into astrobiology, and it's organized by students and postdocs. And it's a really great experience, especially if you've never done a formal astrobiology or formal presentation. It's a really good experience and opportunity. Graham actually also was one of the other speakers of this session, was an organizer back in the day as well. So the next edition is online. It's going to be held in August, and the abstract deadline is May 15th. So if you're interested, please visit the website and the new team of organizers. They've been doing a really great job, and I highly encourage you to attend that. So thank you all for your time and attention. Thanks again to the organizers for giving me this opportunity. I'm sorry again that I couldn't be there live with you, but I'm very happy to take questions either by email or if you want to leave a comment on the YouTube, I'm sure we can be in touch. So thank you again, and I hope you enjoy the rest of this meeting.
Okay, great. So now we will continue forward. So it is my pleasure to welcome our next speaker, Dr. Fatih Karuya. Dr. Karuya is a scientist affiliated with Blue Marble Space Institute of Science within the Astrobiology and Space Sciences Division at NASA Ames Research Center. Dr. Karuya conducts research related to astrobiology, space biology, and human spaceflight. In particular, he is interested in life beyond Earth and how biological systems respond to a space environment, understanding and mitigating the effects of long-duration spaceflight on astronauts' health, and developing high-throughput technology for in situ space research. Furthermore, Dr. Fatih Kurua is also a project scientist, and to date, he has successfully implemented 18 investigations using model organisms or humans aboard the International Space Station. To contribute to his research with associated space exploration, and until recently, Dr. Kurua was a research professor at the University of California, San Francisco, and the NASA Space Biology Non-Rodent Portfolio Lead Scientist. For the latter, he oversaw a broad portfolio of investigations conducted aboard the ISS and managed by the NASA Space Biology Program. Dr. Kurua received a PhD in biology from the University of Houston, a master's degree in space studies from the International Space University, a master's degree in space technologies from Louis Pasteur University, and a master's degree in physics and a bachelor's degree in physics and chemistry. So it is now my pleasure to welcome Dr. Fatih Kurua.
Hi, how are you? Good morning, good afternoon, or good evening, whatever you are. Thanks again for the introduction. Let me share my screen. Are you able to see my slides?
Yes, we are.
Okay, great. So again, thanks for having me. It is my great pleasure to be able to spend some time. As you can see, it will be a very short period of time, and so I will not have time to go into detail. So the intent of this presentation is just to tease you out a little bit about space biology and more importantly about the different type of ongoing studies and the need of talented scientists like you that will need to be involved in the next few years or even decades to try to help human spaceflight and space exploration. So let me start with giving you a brief outline of my presentation. So as I previously mentioned, it will be just like a 20-minute talk, so I will not go into too much details. But I just want to emphasize that currently we are living under exciting times, and so I will be spending a little bit time related to that. And then I will dig into describing a little bit, because I will assume that many of you don't have a strong background related to human spaceflight or space biology in particular, and therefore I will spend a little bit of time trying to describe a little bit all the different type of space environments and what is the impact of the lack of gravity, increased level of radiation on humans. And then we kind of transition with giving you an example of a study that have been conducted on board the International Space Station, and in particular the Microbial Tracking 2 study, which was looking at the microbiome of the environment, as well as from the astronauts. And then kind of transition from what we have learned so far, for example, in low Earth orbit, and how we extrapolate by going beyond low Earth orbit and, for example, going back to the moon, and hopefully soon to Mars. And then I will emphasize the fact that there is a need for having space biotechnologies on board habitat or spacecraft, and that would be the only way for us to be able to sustain a safe space exploration. And so let me start briefly by mentioning that we are living under exciting times, and as you are well aware, between now and 2025, a lot of people are currently working and trying to implement the Artemis program, and the idea there is to have the first woman on the lunar surface by 2025, and that will start with an habitat orbiting around the moon, the Gateway, and hopefully then we will be implementing habitats and surface exploration around the moon. And so, but that is only one step forward towards hopefully exploration of Mars. And here is a roadmap of NASA by continuing performing studies in low Earth orbit on board the International Space Station. Then, as I just mentioned, with some lunar activity, and hopefully within the next decade also, then having exploration of Mars. And I think that we all have an important role to play in those new types of robotics and human exploration. When we deal with the space environment, obviously it is an environment which is very different from what we are experiencing here on Earth. And so it's composed of a near vacuum or vacuum, depending on your locations, extreme temperature variation, lack of gravity, and so going from microgravity to partial gravity if you are on the moon or on Mars. And then obviously one of the main limiting factors is space radiation, and depending on your location, you have different type of environments. When we transition to the impact of that environment on the human physiology, as you can see, the effect of microgravity in particular on the human physiology is important and are functions of the mission durations. And here I'm laying out some references related to the impact of the space environment on different organs or physiological system. And so just to kind of tease you out, let's talk about the central nervous system. And so studies have shown that actually there are nervous tubular problems and cognitive alterations in regards to the cardiac system. There are heart atrophy, cardiac arrhythmia, decrease of plasma volume and blood cell mass, and then orthostatic intolerance upon return to Earth. In regards to the immune system, I mean, as you're probably well aware, the astronauts' immune system is, and I guess we have a tendency to call it immunocompromised. So the crew are immunocompromised. And so that means that actually they have a lot of problems related with the immune system, and that leads to, for example, alterations in natural killer cells, T cells activity. There is a lower phagocytosis and oxidative burst capacity from neutrophiles. There is a reduced antimicrobial function of monocytes. And then also there are changes in the distributions of interferons and cytokines production. And then also some issues related to viral shedding, and so there's an increased frequencies of viral sheddings when crew are in space. Then there's bone loss density, which is also obviously very important, and the rate goes from 1% for the lumbar spine to 1.5% for the hips and the femur per month. Obviously, muscle atrophy because of a lack of gravity. So there's a decrease between 5 to 10% of muscle size and over 20% of muscle strength for a 6-month mission. And then there's a new issues related to the health of the astronaut, which is called spaceflight associated neuroocular syndrome, which results actually in optic disc edema and also some issues related to the optic nerves. And so some of the crew comes back to the ground with some visual impairment. And so certainly this is an issue. And then on top of that, as I just mentioned, I mean, radiation could have some additive or synergistic effects. So as I just mentioned, I've been involved in a few projects and I've been lucky to do so. And so I would call those astrobiomes, and so microbiome of the space environment. And I will be talking about project that I've been involved with. And so I've been involved with the Microbial Tracking 1 studies, which was recurrent studies sampling different area from the International Space Station. And today I will be talking mainly briefly about Microbial Tracking 2, which was a combination of taking samples from the environment as well as taking samples from 4 crew members. But as you can see, NASA is not just the only entity conducting those type of studies. There have been also similar studies coming from European space agencies as well as JAXA. So Microbial Tracking 2 was an interesting study, and actually that was the first human-related space biology study that was sponsored by the Space Biology Program. And so the study was looking at, as I mentioned, taking samples from the environment. So we were swabbing different surfaces, dining table, toilet, exercise equipment, and so forth. And then we were also taking samples from the crew. So we were taking samples from their mouth, from the nose, and from different skin areas, so like forearms, and the navel, and behind the ears, and so forth. And so this is a picture from the International Space Station. As I mentioned, there was a sampling that took place mainly from the United States orbital segments. And so what I mean by that, those are the main elements from international partners as well as from the US. And so here's a snapshots about all those different locations. And again, I'm not going to go into details, but different samples. So here you can see the toilet. So we took samples from the toilet, we took samples from the exercise machines. Here you see the dining table. So we also took samples from there. And again, that was combination of environmental sampling as well as taking samples from the crew. So obviously these days, so after collecting samples from the station and then those are returned to the ground, and then obviously we use high throughput methods to be able to characterize the microbiome environment. And so here's a snapshots looking at the relative abundance. So the top 12 genera are here depicted here. And so for each rows correspond to one crew member. So as you recall, there was 4 different crew members. And so we took, again, samples from the ear, the nostrils, the skin, which was combined from different locations, the mouth and saliva. And so again here, different crew members, some of which were operating at the same time, which was also interesting. And then actually there was also from crew members from different countries, which was also interesting to see. And for example, from a saliva perspective, so as you can see, the changes when we take samples prior to the mission, during the mission, and after the mission, as you can see, there's a lot of changes depending on the time period and the locations. And what was interesting also in this case, for example, when we're trying to perform what we call non-metric multidimensional scaling and principal coordinates analysis, where we were comparing samples location by crew members. And as you can see, there's a good correlations when we look at, for example, the saliva and the mouth. We could very easily identify them by crew members as opposed to other area which was more mixed within the samples. So here's just like giving you a brief synopsis of the type of studies that can be conducted where we collect samples from the environment as well as from the crew using high throughput methods. And so again, I don't have the time to go into details, but what was interesting to see is that when we took the samples from the crew, samples coming from the skin, nostrils, and the ear were much more similar to the surfaces as opposed to those samples coming from the mouth and the saliva, and in some fashion it makes sense. There was a strong correlation when the crew member was taking surface samples with the microbiome of the astronauts. Interestingly speaking, when we were looking at samples coming from the saliva, there was a lot of changes in relative abundance from those samples. Similarly, there was an increase in antimicrobial resistance from the saliva samples. And then again, there was a lot of changes related to the saliva, but those seems to come back to normal after the crew returned to Earth. So when we extrapolate from what we have learned from low Earth orbit to moving forward to going back to the moon, for example, and going to Mars, as you can see, I mean, up to now, you know, we have activities on the stations that are the maximum, roughly, you know, over a little bit over a year. And certainly when we went to the moon some time ago, it was only for like up to 2 weeks. And so certainly now by having those different type of explorations and going back to the moon, which will be for a longer period of time, and even more importantly, when we envision a mission to Mars, which would be a 3-year mission, as you can see, there's still a lot of unknowns. So the unknowns can be characterized in different elements, some of which, as I mentioned, are related to the effect of radiation. Probably space radiation is one of the main limiting factors to long duration space flights. Isolation and confinement, there would be limited capabilities for you to return. As I mentioned, I mean, you would be very far from Earth, and communication would be obviously an issue. Close habitat, so if there's an issue with the habitat, then you're big trouble. And then there's transition from microgravity environment to partial gravity, and so certainly this is something that we don't have a lot of information on. And so when we look at human spaceflight, certainly there's a lot of factors which are associated with human in general. And then as I just mentioned, those 5 different risks which are currently being tracked by the program. And so by combining those two, then there's a lot of work that needs to be done, and there's a need for talented scientists like you and using those high throughput methods to be able to try to solve some of the risk and mitigate those risks so that we have a sustained space exploration program. So I will argue that this generation is a lucky generation because the 21st century is the century of biology. And as you are well aware, I mean, the central dogma of molecular biology states that DNA contains the instructions for making a protein, which are copied into ribosomal and the RNA. And so here you have all those different methods which enables you to try to get some information at the different stages. So obviously, by doing sequencing, you can get some information about the DNA, by transcriptomics analysis of the RNA, proteomes, and so forth. And so all those new type of methods and high throughput methods will enable us to actually be able to discriminate from a healthy individual to someone that might have a disease, either short-term or like long-term. And so by combining all those different methods, I will argue that this is the key for us to sustain a safe human spaceflight and long-duration spaceflight in particular. However, when we look at space research, I will argue there is a lag phase. So a few years ago, I published a review article about specifically that. And so it is a busy chart, but it's pretty straightforward. So on the y-axis, you have different type of methods. So going from imaging to genomics. On the y-axis, on the x-axis, you have here time duration. So let's talk about imaging. So for example, there was a microscope that came out into the market in '96. It took actually 15 years, 15 years to be deployed, a similar version of the device to be deployed on board the International Space Station. So very, very long period of time between the moment the device came out in the market from the moment where it was integrated on board the International Space Station. Another example, for example, if you want to look at transcriptomics, so RT-PCR came out in the market as a new technology in '96. There was a new system that came out, which is called the Cycle cycle in 2000. And it took us roughly 16 years to deploy that specifically device on board the International Space Station. And as you can see, I mean, this is way too long. We did better for sequencing and the mini-ion in particular. For example, mini-ion came out in market in 2011 and actually was on the station, tested on the station and validated in 2016. So here's Kate Rubins using the mini-ion. And again, that was very, very exciting for us. And certainly because this is a small device, doesn't require a lot of resources, certainly this was very much appealing to the program. However, you still need to take samples, you still need to do some amplification steps and so forth to be able to do a reading and be able to use the mini-ion. And so if you want to transition to an environment where we could, for example, either use commercial off-the-shelf instruments, and so certainly there's advantages in doing so, because then that would be something which is new and very much advanced, and therefore that might require less development to be able to use that technology. However, from a space perspective, well, then, you know, there would be an issue for automation. Does the device adapt to the space environment? And then maybe we need to do some qualification. On the other hand, if you, for example, from the get-go, from the beginning, try to design a space-based platform, and therefore you will be optimizing the 3Ds, it will be automated and miniaturized, but then that will require a long time to develop and test the technology, and most likely that would be a less advanced type of technology. And then you have that hybrid platform system, which could be kind of plug and play, which is a kind of mix of commercial available platform and maybe combining it with space-based platform. And so I've been lucky enough to be involved in the development of such technologies, one of which is to design an automated nucleic acid extraction, and the other one was to specifically test and validate a prototype to do gene expression analysis. So again, I don't have much time to go into details, but here I'm giving you snapshots of an instrument which was developed and tested actually at Yellowstone National Park. And the device, as I just mentioned, is automated, so it enables in automatic fashion. You can have 6 different cartridges. So it's based on magnetic with bit beating. and that enables you to extract in automatic fashion in a very short period of time nucleic acids for downstream applications. So certainly it's very appealing because that takes away the input from the crew. And so we tested and validated the device on the field, and actually we showed that using the device as compared to standard protocols to extract nucleic acids, and then using Illumina technology or the nanopore technology, actually we showed there was no differences between using the device, which again is automated, or standard laboratory practices. Similarly, as I just mentioned, I was involved also in the biological validation of a prototype, which was specifically designed for a nanosatellite technology. So here you have a 10 by 10 a device which enables you to grow cells, then to extract. So to kind of lyse those cells and then extract the nucleic acid and RNA in particular, and then hybridize it into a chamber, which enables you to measure gene expression. And so the idea there is to have that system on board a nanosatellite and better understand the effect of space environment on microbes, for example. And so we did show that extraction from the system, again, which is miniaturized and automated, was similar to ground control. And then we ran in comparisons looking at gene expression analysis from RNA extracted from the prototype versus laboratory practice. And we showed that there was a very, very strong correlation between the two. So again, I will argue that if we want to use space-based technology on board space habitat or spacecraft, I think that for going from now to the next 3 years, I think all the PCR-based or microarray-based and mini ions can be used. I think within the next few years, we can implement certainly the next generation of sequencing and then have microarray-based proteomics, but it might take a little bit more time to be able to have a proteomics and metabolics type of platform that would be very, very useful for human space exploration. So again, I will argue that within the next few years, we can deploy those omics-based capabilities, and I think that we all have a role to play in helping developing those new platforms. And I think those will be tremendous and critical to enable to minimize risk associated with long duration spaceflight. So before concluding, obviously, I just want to also use a minute or two to kind of as an outreach platform, and maybe some of you might be interested. And again, I will argue we are living during interesting times, and I think there is many opportunities for many of you to be involved, either in academic institution or within space agencies, or maybe you have your own idea, and if so, then maybe you can start your own startup. Certainly interact with the community, either at conferences, and it's important to network. I think Graham will be talking a little bit about Blue Marble because I am also affiliated with the institute, so I will not talk about that. But certainly, I also participated in the International Space University program. So there's different programs that enables you to, in some fashion, to explore your interest in space research. And again, there's many different programs which enables you to be involved. And NASA actually have a postdoctoral fellowship, which is actually open to anyone, not just a US based student, but also foreign nationals. And then there are the UN Office of Outer Space that have many different programs for countries that don't have a space program, and so actually they are sponsoring you to do some flight experiments or to develop some CubeSats. And then if you're interested in space biology and human space flights in particular, there have been some special issue, one of which I was managing, and that kind of give you a snapshots of what are the state of the art in relation to space biology and human space flights. And then GeneLab. GeneLab is an interesting database that NASA has developed throughout the years, and actually that enables all space-related investigation to deposit all the omics data into the platform, and then that enables talented scientists that are interested to go and mine the information. And so certainly there are also multiple type of tutorials inside, and actually there's also working groups that you can participate if you're interested in that. And then, I mean, currently I'm involved with the Jet Propulsion Laboratory in characterizing some of those samples, and certainly there's always a need for us to have people which have an interest in and actually some talents in bioinformatics to help us better characterize all those different samples. So I will stop here. Here's my info. I mean, certainly you're more than welcome to reach out if you have any questions, and thanks again.
Great. Thank you so much, Dr. Correa. That was a great presentation. So we have a few questions for you. Number one, what qualifications do you think are most important to have if you want to get involved in astrobiology?
I mean, I don't think there is one fits all. I think that at the end of the day, you need to do something that you're passionate about. And I think this is key. And so certainly if you want to be involved in astrobiology or space biology, well, I mean, having some background in biology would be critical. But there are many different venues for you to be involved, as just laid out in my, I guess, last two slides. And so again, at the end of the day, it's just, you need just to try to do something you're passionate about, and there's no limits. And so certainly there's many different venues for you to be involved. Obviously, I'm coming from a US-based, I guess, institutions, and so I am familiar with many of the NASA programs, but these days, I mean, everybody at the international level is involved. And I think by networking, attending conferences, that kind of enables you to kind of have a better understanding of what type of science you would like to focus on. But certainly we need this next generation to be involved in a human spaceflight, because certainly there's many different opportunities for all of us.
Great, thank you. The next question is, what is the most critical challenge in astrobiology one might be able to solve using bioinformatics?
I mean, I think when we move forwards, they have all those risks that have to be addressed. And I think omics-based studies, and therefore people that are talented with bioinformatics. I think actually bioinformatics currently and computational biology and artificial intelligence would be probably the key type of disciplines that would be needed for us to be able to address all the risks that limit us to be able to explore our solar systems and so forth. So certainly, as I mentioned, get involved with the Gene Lab. There's all those different working groups that you can participate, because they have we have a lot of data which are currently available, and that just need to have talented scientists to be involved with them. So.
Great. And then I think we have one more question along similar lines. How can we create safe space exploration using omics methods?
Well, I think that kind of goes back to what I just mentioned. I think it's kind of twofold. One is related to the need of having omics platforms as an integrative part of the mission. As you're probably aware, I mean, currently we have limited capabilities on board the International Space Station. In theory, the International Space Station is a laboratory, but there's not much instrumentation for analysis. And so currently the way things works is that you expose your samples, or you take samples from the crew, and then you have to wait until those samples come back to the ground to be able to analyze them. And so that's not going to work well when we envision missions where we don't have those capabilities, right? And so if we envision the mission to Mars, which would be a mission of 3 years, most likely, and so therefore you need to have all those put all those omics-based platforms inside the habitats or inside the spacecraft and continue monitoring the health of the crew or the health of the environment. Obviously, the crew is important, but also you need to samples the environment just to make sure there is no issues. Certainly, life support system will be also a key component, and therefore you need to study that. And having all those omics platforms will be critical to minimize the risk and be able to monitor the health of the crew. And so, certainly, there are a lot of venues for us to be involved, either from a science perspective or from a technology development.
Great, thank you. Actually, one more quick question. What is the most exciting thing for you about in situ proteomics in space?
I mean, I have an interest as a scientist, so certainly having those platforms help us to stay away from the long cycle of how we do science these days, and space science in particular. I mean, it roughly takes 5 years from designing a study to implement it and to analyze the sample. So that's way too long. And so having those platforms and proteomics in particular, I would argue that these days it's all about genomics. But yes, I mean, having information about the DNA is important, but having an information on what type of proteins and metabolites have been secreted by cells or the human system is even more important. And by combining all those different methodologies and technology, then we will be able to address all those risks and make sure that the health of the crew are kept at bay, and making sure that we are returning them healthy back to Earth. And so definitely a combination from a science perspective as well as from a technology development.
Great. Well, thank you again so much, Dr. Correa, for speaking with us. It was a very informative talk.
Thank you.
All right. So it is my pleasure to introduce our next speaker, Dr. Graham Lau. Dr. Lau is an astrobiologist and a communicator of science. With an academic background spanning biology, chemistry, astrophysics, and geology, Dr. Lau is an expert on how living things affect the environment around them and how we search for alien life beyond Earth. He serves as the Director of Communications and Marketing for Blue Marble Space, as a research scientist with the Blue Marble Space Institute of Scientists, and as the Director of Logistics for the University Rover Challenge, and also as the host of the NASA-funded show, Ask an Astrobiologist. Dr. Lau also serves as a meditation instructor and a public speaking coach. So please join me in welcoming Dr. Graham Lau.
Thank you very much, Laura, and thanks everyone. I'm going to do the share screen thing here very quickly. Hopefully everyone can see this. All right, it's a pleasure to join you all today to talk a little bit about careers in astrobiology. And we're going to step back a little bit from targeting just really research focused things and talk a bit more grand scale about what astrobiology is. And then I'll give some of my personal suggestions on how to be involved in what I think is the grandest challenge, the grandest quest of human history, is to know where we've come from, what we are, and whether or not we're alone in the universe. Astrobiology truly is the quest to understand the nature of life, to understand our position here in the cosmos on this blue marble. This, of course, is the famous blue marble image from December 7th of 1972, when the last Apollo astronauts were on their way to the moon. They snapped this beautiful shot of our world. And I love sharing this image, and I think often of this image and other images of our world from space. It's a view I'd love to have personally one day looking down, but it reminds me, that we are together on this planet. We share a biosphere. All of you here, from young undergraduate students, maybe graduate students, and others who are interested in omics research and other forms of research involved in understanding life, are part of the grand quest of astrobiology. In careers in astrobiology, and I'm going to talk just briefly about some of the different things astrobiologists do, and then I'll give some of my personal take-home messages for what I think you can do to be even more involved. We have people just like Tony Gia, who talked this morning, a researcher at ELSI and BMSIS, who's involved in understanding the origins, the early transitions into life on our biosphere and maybe on other worlds. We have researchers trying to figure out, did the material for life come to us from space? Did it all start here on our world? Could life have started elsewhere and then come here? These are all really big questions. We also have people studying geology and planetary history, the formation of worlds and the processes that happen to them through time. Researchers studying meteorites to better understand the history of our solar system. Researchers using large telescopes to look at worlds out there to better understand not just how worlds form, but also what kinds of worlds may have life out there for us to target even more and to look for things out there. And many of you here who might be involved in omics research are probably in some way involved in, again, this quest to understand how life functions here on Earth. Not just the genes that various populations have here on our planet, but the expression of genes that have led to such beautiful biodiversity across our world. We are very lucky to live in this extremely diverse biosphere with so many different forms of life. Often I just kind of nerd out and get super excited just thinking about all the weird, almost alien things that live right here beside us. In the next talk, you're going to hear from Gary Trubel about viruses and soil ecosystems. We don't really know yet how to even classify viruses relevant to life. Are viruses alive or not is a big question I've heard throughout my life. I personally lean towards them being part of life and being alive. We also have people studying, extremophiles living in very hot systems like Grand Prismatic Spring here in Yellowstone National Park, and in very cold or very saline, high pressure environments, ways for us to better understand how life pushes the limits of chemistry and physics to inhabit so many environments across our world. And of course, there are many people involved in going out into our solar system with robotic missions and even hopefully in the near future with human missions to explore other worlds. We have some candidates for potential life in our own solar system that are worth looking into and exploring further. Of course, we've sent lots of things to Mars, but there are also other potential candidates like Europa and Enceladus and Titan, icy worlds in our solar system. But we also have potential for life in the clouds of Venus even, and it's worth exploring that potential to better know if life only happened once in our solar system or if maybe life has happened several times in our own solar system. But you might have recently heard the news that there are now more than 5,000 known worlds around other stars out there. When I was a young child dreaming of becoming involved in astrobiology and space science, we didn't know of any exoplanets at all. And now we've passed 5,000. And that makes me wonder, when do we pass 10,000? When do we pass 100,000? When will humans look out to well over 1 million worlds that we know for sure exist out there? Based solely on the numbers we've found so far, there should be something at least around the realm of 100 billion to maybe even 1 trillion planets in our galaxy alone. And that to me makes it seem like if there is life out there beyond our solar system, it makes it even more likely that we might find some of that life. And so if you want to be involved in astrobiology, you can be involved in the biological side, in studying the populations we see here, but also trying to figure out how would we find signs of life on some of these exoplanets out there. Right now, we're really just limited to looking at their atmospheres. So the James Webb Space Telescope has been launched. It's successfully aligned now, and we're going to soon start getting some really great data back. Some of the first exoplanets that it will be looking at are ones that we already know exist within the Goldilocks zone for liquid water around their stars. Worlds like the TRAPPIST worlds, the TRAPPIST-1 exoplanets, and others that might be good candidates at looking at their atmospheres for possible signs of life. And so we need young people who want to be involved in this exploration in helping us to bring together all of our knowledge to figure out what could be out there. And if you're a nerd like me, maybe you also want to know if there's intelligent alien life out there in the cosmos. The Drake equation was originally developed in 1960 by Frank Drake, primarily to help him organize a meeting of other scientists to answer a question of what do we need to know to find out if there could be communicating aliens out there in the universe. But but it's also a very intriguing expression, primarily because of the final factor in the equation, the L factor, which is the average time span for a civilization to exist. It was poignant and beautiful for Drake to have added that at that period of time in 1960. We were becoming aware, very aware of our potential to destroy ourselves, primarily through nuclear war and a nuclear holocaust. But there are many other potential existential threats that we face and that other civilizations most likely have faced or do face as well if they're out there. And so that factor, the L factor, makes a lot of us wonder, what is the future for our species? You know, in Fatih Korea's previous talk, he was talking about the factors that are involved in space exploration and how we can study the expression of genes and things like that in space exploration, but we've only really just begun human space exploration. We don't really know what happens to our species with another century or a millennium of space exploration, where our species goes. A big question is, will we see this idea of the singularity of all of us kind of merging into our machines when our technology advances so fast that we basically become the machine, or the machine takes over and leaves us behind? We don't know what that looks like. It might be likely that a lot of life out there in the universe is post-biological life, that most life is artificial intelligence, and basically written in silicon and other materials of metal, rather than being built in organic chemistry as we are. But all of that said, there's a lot of need for a lot of people to be involved in astrobiology, a lot of people involved in answering these questions. And again, you know, we also face this potential for our own destruction, and we should question what our future is. One of my favorite science fiction stories is Star Maker by Olaf Stapledon. It's a story written in 1935 originally, where a human consciousness leaves its body and travels out beyond the Earth and looks back. It travels beyond our solar system. It travels out into the galaxy. The consciousness itself experiences becoming a type 1, a type 2, a type 3 civilization on the Kardashev scale. In this story, this idea of galactic and then universal exploration, it's a fun way to kind of explore what it would be for a human to leave our bodies and to explore the universe. But one thing I really love about the book is that Stapledon faced some issues with others questioning why he would write such a fable about space exploration at a time when Europe was on the brink of war. And in the 1937 intro to the story, he wrote this, that perhaps the attempt to see our turbulent world against a background of stars may, after all, increase, not lessen, the significance of our present human crisis. It may also strengthen our charity toward one another. And I often think that way about astrobiology, thinking about our place in the universe and the origins and the evolution, the distribution of life out there. It makes us think a bit more about our own fragility, our own precariousness, and our own preciousness as a species in this biosphere. And I think it makes us be a little bit kinder to each other and want to do better for the future. And so I would love to see a lot more people involved in learning about astrobiology, if not becoming astrobiologists themselves. And the interest has certainly grown over the years. This is Charles Coquel's course for Coursera. It's had well over 70,000 students now have taken this free online course. My friend Sid Pandey, who leads the Amity Center of Excellence in Astrobiology at Amity University in Mumbai in India, a couple of years ago had an Introduction to Astrobiology course that had 795 students from 53 countries take part. And I love this graphic created by a young student who previously worked for me, Catherine Herrera, where she created a graphic bringing together 36 different languages of how you say astrobiology. So all of that long-winded kind of idea of this grand idea of what astrobiology is brings me to this. I have a few things to share with you if you're interested in astrobiology, but honestly, these are kind of my career advice suggestions for any field, really. The first one is to be a generalist as well as a specialist. What do I mean by that? To become a research scientist, so I'm a research scientist. My background is primarily in geochemistry and mineralogy. I have a paper coming out here very soon about some geological structures from the Arctic and how they relate to our Mars exploration, for instance. And so I'm a specialist in the realm of geochemistry, applying it to field sites on Earth, to other worlds. But I'm also a generalist, as I understand a lot of different things in biology and astrophysics and philosophy, history, and other realms. and that helps me to frame my thinking. One of my favorite arguments ever for a liberal arts education comes from Star Trek: The Next Generation. There's an episode where Captain Picard is looking out at the cosmos and talking to a young man named Wesley Crusher, and the young man laments that that he can't really read this book the captain gave him because history is not going to be on his exam for how to pilot a starship. And the captain responds that the most important things never will be on your exams, but if you study things like theater and poetry and history and philosophy, it'll make all of this make sense. And he looks out across the stars. And so becoming a generalist will help you in your career in every way you go. Here's a great page from NASA Astrobiology, their career path suggestions for getting into our field. And astrobiology, it really is a very diverse realm of study that brings together many disciplines. But you can learn about astrobiology through courses, through people like me online, and shows that we have, various social media. But a good place to be is to earn a degree in biology or chemistry, geology, and then start to specialize in the questions that you really drive you. Specialize in the realm that you want to work in yourself, but again, be a generalist. Try to speak the language of all these different sciences. And again, we need everyone, and anyone can be an astrobiologist and join us in this adventure of coming to better know ourselves and the universe. Secondly, I think you need to own your own learning. When you pay money to go to a university and earn a degree, that's great. And it's great to have a formal education. But you should never let a formal education get in the way of your own learning. The classes you take, the courses that you pursue, certificates that you add to your CV, those things are okay. But you really need to be in charge of how you approach your learning, how you approach your reading and your writing, and the time that you spend in research. So there are some suggestions we have for our Young Scientist Program participants. I'll mention that some more later, even though Tony and Fathi both have mentioned it already. But we also recommend, amongst learning about science, is to learn about how to be a good researcher, how to learn more. And so I recommend books like Limitless from Jim Quick and Deep Work from Cal Newport, different ways that you can study and learn a bit more about how to be a good learner and how to own your own learning. And of course, there are lots of books out there that you can read to learn more about our explorations of the universe, from the writings of Carl Sagan in Cosmos and Pale Blue Dot to David Grinspoon's works, Chris Impey's works. There are many great books. I recommend my Amazon shopping list. I put together a small list of books related to learning astrobiology that I found helpful for a lot of young people. Thirdly, I think you need to build a network, build a community around your research and what you want to do. If you're working in bioinformatics, find networking, find communities of people who are working in the same field, who can help you answer questions, but also who can help give you ideas of where to look for job postings to further your career. And astrobiology, there are a lot of great organizations who are doing things relevant to astrobiology, organizations like SegaNet, of which I've been a part, the Planetary Society, the Mars Society. Tony mentioned the Astrobiology Graduate Conference. For graduate students, if you're interested, the AbGradCon and AbGradE, the European Astrobiology Graduate Conference, are both great ways to start meeting other graduate students. And for undergraduates, sometimes they accept undergraduate students who are really good at what they're doing. And then Tony mentioned, and Fathi mentioned as well, this Young Scientist program that we have at Blue Marble Space. Not only am I a mentor for the program, I'm also the program's director. And so if you're interested, this is a summer research internship from June through August, and we'll have it this summer. Here's just a small list of the projects that we have available with our researchers for this summer. Things like graph theory, viruses versus the human immune system, ancient microbes, a lot of things that involve coding and programming in various ways. I have a project through ELSI as well with Tony called The Landscape of Community Engagement in the Origins of Life Research. Lots of great projects. If you're interested in learning more, you can check out our website, bmsis.org/YSP/projects. The applications are due fairly soon by April 15th, but if you have any questions, feel free to ask me about that. I'm always happy to talk further about our program. Through the years, we've had now over a few hundred young students be involved in this process, learning about not just research in science, but also learning about moral philosophy applied to science and about how we communicate and share our science. Also, in Laura's intro for me, she mentioned that I'm the host of the show called Ask an Astrobiologist. This show is primarily targeted towards the younger kind of undergraduate level, and even for those who aren't actually involved in astrobiology to learn more about our field. But during this show, which we air live on YouTube once each month, I interview other astrobiologists to talk about their career paths, their research. I even just the other day was speaking with a young student. He watched my episode 37 with Melissa Traynor, the deputy PI for the Dragonfly mission going to Titan. After watching my episode with her, he reached out to her and just asked, like, do you have any research available that I could work on? I would love to learn more about Dragonfly. And it turns out she did. And he ended up joining her for a research project. They now have a manuscript in publication. And so he'll be involved at least somewhat for some of the research going on with Dragonfly going to Titan. Finally, if you can, and you want to be involved in engineering competitions and science competitions, there are lots of ways to network and join various groups. I help to lead the University Rover Challenge, where undergraduate student groups from various universities around the world, they build Mars rovers and they bring them out into the desert of Utah after what is usually a year-long or even multi-year process of development and testing and showing us that they're ready to come out to the desert. And then we give them a very hard challenge at the Mars Desert Research Station, which you can see in the background here in this image, where I've also been an analog astronaut. We have the students running their rovers around in different kinds of tests, including science. There's a science site where the students have to drill or sample in some way to learn about the biology in this desert environment, to learn about the geology of this desert environment. And so there's lots of really cool ways to get involved and build your community and build your network if you want to be involved in astrobiology. And so finally, I'm going to end just with this real quickly, that it's really important in your life and your career, no matter which field you pursue, is that you find the right mentors. And I've seen this a lot, I think, because of how we structured academia. A lot of young people, a lot of early career researchers, they think a mentor is just a teacher or an advisor, like an academic advisor, and that's really not the case. A good mentor is far more than that. A good mentor is someone who knows when to be your friend and when to be your boss. It's someone who knows how to teach you, but how to help you to learn how to teach yourself. A good mentor is someone in your life who is really going to be there for you to help you answer questions, to steer you in the right direction when you feel like you don't know which way to go, but also to allow you to grow in your own ways. And so a mentor is someone whose hindsight can become your foresight. For those of us out here in the world who've been very fortunate and blessed and honored to be mentors for young students, and I'm so fortunate that I've had the chance now to impact so many other lives and to help others out there, I can tell you it's an honor and a privilege to be a mentor, but it's also something that many of us take very seriously. And so if you have a teacher or an advisor and they're not really taking that job seriously, they are not your mentor. Your mentor doesn't have to be in the same program, the same research group, or the same university. Your mentors in your life can help drive you forward and help you to find what ends up becoming your career. And like I said earlier, I really hope it is in somewhere in this grand scheme of understanding the nature of life out there. So I'm going to leave it just with this slide of how you can reach out and find me out there if you're interested. I will say I'm giving a talk in Denver on the 4th of April at the Museum of Nature and Science about Mars exploration and Sarah Johnson's book, The Science of Mars. It's part of the Science Friday Book Club program. I'll be at the University of North Florida on the 22nd of April. If anyone's down in Florida and wants to come hear a talk about chemistry and astrobiology. I'll be at the Analog Astronaut Conference the 6th through 8th of May. So if any of you have been involved in analog astronautics or want to be involved, I think there are still some tickets left to come down to Biosphere 2 and to hang out with a bunch of us, including astronaut Cyan Proctor. And then finally, here in May, I'll be at AbSciCon from the 15th through 19th. If any one of you are going to AbSciCon or the Astrobiology Science Conference and want to meet up, feel free to send me an email, drop me a line, find me on social media or my website. I'm always happy to chat. So with that, I'll take any questions that are available.
Thank you so much, Dr. Lau. That was an awesome presentation. So we have a few questions for you. The first one is, what is your own belief for how life began on Earth?
Oh, I love it. You know, I don't know, and I really don't think we're ever going to know. So the way that, you know, the history of our planet works, you know, the oldest rocks we have on Earth are around like 3.5, 3.8 billion years old. The oldest minerals we have on Earth are 4 to like 4.2 billion years old. But those minerals, usually zircon grains, get trapped in younger rocks. But the very early history of our Earth is erased in the geological record by the fact that we live on a dynamic planet with plate tectonics. And so I think the only way to really ever know for sure is if we had some really nice alien neighbors who've been out there watching us this whole time. And one day we can meet them and join the Galactic Federation, and they can say, hey, this is how it happened on your planet, just so you know. I will say that I am a big fan of the concept of panspermia, that life might have started first on Venus, for instance, and then gone to Earth. I think starting on Mars and coming to Earth is less likely, but still also a possibility. But I don't want to rule that out. It is very possible. But we don't have any evidence yet to support that. And so it does seem like life at some point very early in our Earth's history started here on our planet.
Great, thank you. Our next question is, does biology or bioinformatics support the theory of astrobiology, and how does it go about doing that?
Well, I would first state that there is no theory of astrobiology. And one thing I really loved in the original Cosmos by Carl Sagan, is he spoke about in chemistry and physics, we have, defined laws. We have this idea of the physical nature of our universe, but we're still trying to figure out what life is. We have some attempts at a definition of life, but all of them are somewhat questionable. The current best one is the NASA definition of life, that life is a self-contained chemical system capable of Darwinian evolution. I was even just chatting with Richard Dawkins here this past week about whether or not all alien life, all life out there must be Darwinian, which he argues it must be, but I don't know. I don't know if there's some other possibilities for life out there. And so for any of you out there who are studying biology, who are doing work in bioinformatics, you are helping us to better understand what life is. You're helping us to understand the populations of organisms out there, how their genetic information is expressed. And sure, alien life might not have the same information systems. It might not have the same kinds of structures of encoding information and getting it out there. But by doing this work in bioinformatics, you're helping us to start kind of preparing for what we might find if we find life out there.
Great. Thank you again. Well, I don't know if there are any other questions for you, so I think we will move on. But again, thank you so much for presenting. It was an honor to have you here.
Thank you all.
Okay, so moving on, it is my pleasure to introduce Dr. Gary Trubel. Dr. Trubel earned his BS in environmental science with an emphasis on microbiology at the University of Arizona and worked for the USDA for 2 years researching how to use fungi as a biocontrol on crops instead of pesticides. He then did an MS in environmental science and health at the University of Nevada, Reno and the Desert Research Institute, where he researched active bacteria inhabiting the sub-zero temperature Lake Vita brine in Antarctica. Gary finished his studies by earning a PhD in microbiology at The Ohio State University, where he characterized viruses in soils underlain with permafrost and their roles in ecosystem carbon processing. Gary did his postdoc at Lawrence Livermore National Laboratory in 2018, where he used stable isotope probing combined with metagenomics and viromics to track microbial and viral activity and substrate utilization in various soils. Gary is currently a staff scientist at the Lawrence Livermore National Laboratory, where he continues to use metaomics combined with other techniques, for example, isotope probing, to characterize viruses, microbes, and virus microbe dynamics. So please join me in welcoming Dr. Gary Trubel.
Hi, everyone. Can you hear me?
Yes, we can hear you.
Great. So this is such a cool time to go. Graham, that was such a good talk, and I knew I was going to be going after you. Such a great speaker. I think that was perfect to go right before my talk, because it really has us take a step back and think about what are we doing for astrobiology? What are our goals? What do we need to think about? I think a lot of us get bogged down in what an astrobiologist has to be. And, as you heard in my background, the way I got into astrobiology is different than other ways that people get into astrobiology. And I would arguably say an astrobiologist is somebody that questions life. Like, what are we doing? Why are we here? What is life all about? I think one of my favorite quotes is from a NASA Astrobiology Institute poster, where for astrobiology it says, "What is life? Where is life? And how do we find it?" And that's really what it's all about. There's no direct path that you have to take to become an astrobiologist. So here I'm going to give you kind of some vignettes of work that I've been doing, ways that I'm pushing the boundary to really detect and look at life. I arguably think viruses are alive at some points. We have our working definition of life, but there's no set definition. But for instance, when a virus infects a host, it redirects its metabolism, and the host only does what the virus wants it to do. So is it the host that's alive, or is it the virus that's alive? So I chose soils to go into because they're the most diverse ecosystem. And to really look at detecting our limits for life, we need to be looking in soil ecosystems. So I'll have some shout outs at the end for some astrobiology and astrovirology endeavors that I've done. But I want you to take some time to really learn about viruses and soils. And please ask questions if you have any at the end. So the first thing I want to point out is that metagenomics and meta-omic processes, these are metagenomics, metatranscriptomics, metaproteomics, metametabolomics, there's so many out there, have really revolutionized how we look at viruses and just microbiology in general. Here's a figure I took from a paper by Simon Root in 2019, where in blue and green, you can kind of see our isolates of viruses that we just accumulated over time. But in just such a short span of time, we've shot up the number of viral population genomes that we've gotten in databases so quickly. And to kind of help you understand that, what a metagenome is, the way we're looking at viruses without needing to cultivate them, is that we're taking a sample like soil and we're extracting all the nucleic acid. So for a metagenome, this would be all the DNA from the soil, and we're sequencing it. Then we're reconstructing all these genomes. We're also reconstructing the variants that go along with these genomes. So we like to call them population genomes. So this is what I've been pushing, is really looking at viruses from soils using metagenomics primarily. So until recently, if you looked at viral studies, they haven't really been in soils. We had some of our first studies in the oceans, and then we had everyone caring about human viruses taking off. And there's nothing wrong with that. I like human viruses, but I specifically focus on viruses that infect microbes, especially bacteria, and we call these bacteriophage. So these viruses are host specific. They can only infect bacteria, as we know. But there's so much that they can do in the environment, and this is what I'm exploring, and they have the largest diversity of any viruses out there. So what I've done with my time now at LNL, Lawrence Livermore National Lab, is really push soil viromics, a study of just focusing at the virus comportment of viruses in soil. So we've been pushing those studies up there. So the problem, why am I shooting for soils, and how does soils help with our detection of life? When we take a metagenome, when we sequence all the DNA from soil, less than 2% of our data, we like to call this assembled reads, actually go to viral sequences. Less than 2%. So we're not really seeing viruses in our main way in which we're detecting viruses. And the reason for this is if you look at soil, I have this small area that I zoomed in for you. There's so much going on in soils. You have your plants, your roots, the rhizosphere, it's so rich. You have your macrofauna, you have your mesofauna, the microfauna. Then if you zoom in, then you get at your microbes. You have your bacteria, archaea, and if you zoom in even further, now you can see the viruses. So when we're taking this DNA, we're taking all of this DNA. And so the viruses, yes, they're highly abundant. They're the most abundant biological entity out there. But they have these small genomes that get swamped by these large genomes. So I've worked for my PhD and since on really being able to get as much information we can from the tools that we have, and then bringing tools together to increase the detection. So the first thing I'm going to talk to you about is when I use, from a postdoc and now, is stable isotope probing to really look at microbes and viruses in ecosystems, but specifically soils. So if we look at microbes, we have bacteria here, they can exist in many different metabolic states when they're in soil or any environment. I'm just going to keep saying soil. but the majority of them are actually dormant or deceased. And when we take all this DNA, we're looking at this whole community. So we're not actually seeing who's contributing. We're seeing all the members. It's kind of, if we looked at the human population, instead of seeing everyone alive now, we saw everyone who was ever on the earth. And for different research questions, we kind of care about what's going on now or who's contributing right now. In order to bypass this, I'm going to talk to you about two different versions of isotopes that I use with compounds to look at viruses. So the first one we call heavy water. So you have traditional water, which is H2, around 16O, that's the most abundant isotope of oxygen. But we can change that oxygen isotope to 18O. This is enriching it. This is less abundant, so when we add this, any organism that uses water, which is everything that's active, because water is a universal substrate for life, it's going to incorporate this heavier oxygen into its DNA or RNA. So we can actually pull that out and we know who's using the water, so we know who is active. So with heavy water, we're going to label all active organisms and the viruses that infect them. Another experiment that I'm going to talk to you about is where we can use specific substrates to target certain parts of the community. And so instead of all organisms, in this study, I'm going to use enriched plant biomass. This is where we grow plants with 13 CO2. So the plants will take up the CO2 for photosynthesis. They'll be enriched with 13C carbon. So the whole plant is now 13C labeled. So any organism that's munching away and breaking down that carbon is going to become labeled. So I'm going to specifically look at the organisms that are responsible for degrading the plant biomass. But once we have that organism labeled, we then can extract its DNA, and then we can do ultracentrifugation to separate the DNA, physically separating the DNA based on its density. So in these tubes here, we have our dormant, deceased, or non-degrading 13C plant biomass DNA up towards the top. And then the one that's enriched with the 13C or the 18O is more dense, so we can physically separate it. We can pull them out separately, and we can sequence them separately. So this is helpful because now we can sequence who is active and who's contributing, but also for comparative reasons, we also know who else is there. And the reason is when we sample an environment, we're taking a snapshot in time. So just because something's active right now doesn't mean it's necessarily active later. And we're still trying to figure out this time scale and this temporal sampling to really understand how the environment and the dynamics are changing. It could be I sample something now, and then a minute later, those are no longer active. This is what we're really trying to figure out. So we take both and then we can compare. Also, by looking at both, we get this nice genetic catalog of everything that's possible in the environment. And this can help other fields such as biotechnology or looking at the health of the soil possibly. Okay, so going into my first paper that I want to talk about. So this is something that we published last year and it was exciting work. It kind of brought me back to my PhD work where I was working on viruses in permafrost, which is frozen ground. So here we looked at permafrost, which is ground that's frozen for 2 or more consecutive years in Alaska. And what happens is every year, the permafrost has an active layer that seasonally thaws and the microbes become active. And then during the winter, it freezes back up. Now, it's currently thought that all the carbon that's leaving the soil, leading to climate change and global warming, is from microbes coming from the summer. That's when they're active. That's when they're respiring, releasing CO2. But there's been mounting evidence to suggest that CO2 is leaving during the winter as well. So we really wanted to look at this. So we went and we sampled this peat, which is this highly organic, nutritious soil that just hangs around because it's so cold and the conditions aren't right for microbes to break it down readily. So we took this peat and we simulated winter conditions. So we brought it down to below freezing. We made it anoxic, so there's no oxygen, and we incubated it for a full year, and we sampled at a half year and then at the end of the full year. From this, we physically extracted the DNA and separated it, and then we sequenced 23 metagenomes. During this time, we also measured carbon dioxide being produced in these bottles. All right, so the key takeaways of what happened here. We realized that there were active viruses and bacteria in the sub-freezing anoxic permafrost soils. And that when we compared who was active to who was not active, the communities were very different. So in this figure, we have these larger pills, which are bacteria, and then we have these hexagons, which represent viruses. If they have numbers and they're larger, they were active. If they're smaller, they were inactive. Now, I don't show you their abundance, but if you look at the pink ones down the bottom, typically when we look at these permafrost soils, these are actinobacteria. These are the most abundant organisms. They just dominate. And if you look, most of them actually were not active. And the ones that were active were our green ones and our yellow ones. So these are our firmicutes and our Bacteroidetes. And what's crazy about this is if we had just taken a traditional metagenome, we would have never looked at these organisms. Their abundance is so much lower, we would have ignored them. But they're actually who is there and who's active. So we would have missed the important part of our research. So by adding the stable isotope probing with our metagenomics, we got to see this. We also got to see that while only a small portion of the bacterial community was active, a large portion of our viral activity was active, over 70%. Looking into the metabolism of these bacteria, we saw that they had the capabilities for fermentation and for breaking down carbon. Okay, so we also know that they respired and produced CO2. So we physically showed that respiration was occurring over winter. So this makes us rethink how we think of the activities of organisms in different environments, that in these sub-freeze environments, there is carbon dioxide coming from, they are active. We were able to link a third of these active viruses to hosts. And when we looked at their genes, the viruses actually carry these genes that when they infect the host, they can make the host use these genes. And it can help the host scavenge nutrients to help survive the winter, or possibly help them break down other plant biomass. Another thing as we saw is that as the bacterial community shifted over the year, the viral community shifted towards the bacterial community. So we know from previous research that viruses will lyse their host and kind of this kill the winner hypothesis. So they maintain diversity by you have one population that shoots up and the viruses kill it and control it and bring it down. This way it feeds all the other organisms and gives them an ability to actually compete. So now we have a more diverse community. And this could be really important for how our Earth is changing with climate change. Okay, now moving to a completely different environment. We're leaving the cold, the frigid, anoxic environment. Now we're moving to the lush Puerto Rican rainforest. And as you can imagine, this is where we're using our 13C. So we have these lush rainforests, which are known to have this high diversity and are critical for storing carbon on our planet. But one thing that a lot of people don't know is that the rainforests have these crazy redox conditions in the soil where you can get this downpour that just floods the soils. And these microbes are highly diverse here. So what is going on? How are they surviving this? They go through periods where there's oxygen and then no oxygen, and they have to be able to respond and survive that. We wanted to know, how are they surviving and who's active? Are they constantly dying and other ones are emerging? Are viruses active? And more importantly, how does this redox condition, the switching between oxic and anoxic environment, affect the fate of the organic carbon there? Is it helping store carbon, or is it causing a problem releasing this carbon? So we set up a treatment experiment where we had 4 different treatments. We had oxic the whole time, anoxic the whole time, a high frequency switch every 4 days, and then a slower frequency shift of 4 and 8 days to mimic rain patterns of the area. So this is where we used a 13C enriched plant biomass, and we incubated it with soil over 44 days. And remember, we're not looking for all active organisms. We're specifically looking at the organisms and viruses that are part of breaking down this plant biomass and the fate of this biomass being released into the atmosphere or stored in the soil. So we were able to maintain this kind of headspace of oxic or anoxic using a nitrogen gas. And then we incubated for 44 days. So here, we also wanted to compare, are we actually getting increased resolution on our viruses and bacteria by teasing apart the active versus the inactive? So we did a comparative study. We took 10 traditional metagenomes, which I call bulk soil metagenomes, and then we had 85 SIP fractionated metagenomes, and this is where we teased apart heavy fractions and light fractions and compared them. So the main takeaway from this is, if you think about it in comparison to our Arctic samples, the diversity was so much higher here. We had 46 active bacteria before. Here we had 326, and that was just looking at the carbon. There was other ones that didn't care about breaking down the plant biomass. And these bacteria span 20 different phyla. So this huge diversity. And we also had 640 viral populations, almost double compared to the last one. And this is just looking at the carbon only. When we compared our CYP fractionated to our traditional metagenomes, we see that we did increase the resolution on our viruses, because when we compare them, we got about 7% more viral population, so we increased our diversity that we captured. Now, I want you to focus on this figure here. Let me help you break it down, because I know these heat maps can get crazy. So let's focus on the one on the left. At the top, I have the black ones. These are kind of my bulk samples. And then in blue, I have my control samples. This is just regular plant biomass. And then in the red at the bottom, this is the 13C enriched plant biomass. And what I want your eyes to see is, if you see white, there's no abundance. That's a viral population that wasn't doing anything. You see some orange, there's a viral population there, it's doing something. You see green, not only is there a viral population there, but it's taking off. It's killing its host, it's redirecting its metabolism, it's influencing the environment. So that's what we care about. I also have zoomed in on the figure on the right, just the active ones, because these are the ones we really care about, right? These are the ones that are controlling the carbon right now. So you'll see that in the oxic, we had the most. Each line is a viral population. We had the highest diversity of viral populations, and this decreased along the redox gradient. So when it was anoxic and there's less oxygen, there's less viruses. And if you think about it, that makes sense. When you're a virus, when you're taking over your host, you either want to kill it and make more progeny viruses. You're like, oh, my babies. Or you want to lie low in it and you want it to become strong and efficient so it can survive this hard time before you make your babies later. So what you want to do is you want to be able to use a fish Earth, the kind of the fastest, the strongest metabolism, arguably, is oxygenated metabolism. But then if you look at the anoxic environment, you'll see we have these green ones. Remember I said that was important. These are viruses that are highly abundant. So even though we have less viruses in the anoxic, in the no oxygen soils, they're highly abundant. So we have these viruses that were able to fill this niche space that other viruses couldn't. And they're taking off. They're loving it. They're living their life to their fullest. They're happy. So this is actually a paper that we're still developing. And we're really trying to look, how are these viruses different? How are they dealing with this metabolism that doesn't require oxygen? And that's going to be critical to look at what genes are using, what they're changing. And our viruses to the bacterial host. We knew which ones were involved with plant biomass, but are they only affecting the plant degradation by killing hosts and preventing them from breaking down the plant biomass? Or are they doing other things? So we surveyed their genomes and we found that they actually encoded genes that when they infect the host, they're helping the host break down the plant biomass. And this is a reoccurring story that we're finding in soils. Typically in the oceans, you don't see these glycoside hydrolases that help break down biomass, but in soils, more and more of these genes that viruses are carrying to help the host. So they're having these roles in carbon cycling that we just don't know the whole story on. Okay, I'm moving on to my LDRD work. So at a national lab, you can get research funded and what's called an LDRD. This is lab-directed research and development. And this is something I love about working at a national lab. These are called high-risk, high-reward studies. This would be, you spitball an idea to a friend, and they're like, ooh, that sounds crazy. I feel like it would fail. That's what they want to fund here. They want a high-risk, high-reward. You dream something big, and it's either going to be amazing or it's going to fail, but you're going to learn something from that failure. And that's what I got funded, and that's what I've been working on, and that's what I'm going to tell you about right now. Okay. So kind of stepping back, we talked about metagenomes before, and I just want to remind you from the metagenomes, we're getting this low resolution on viruses. So I set up this experiment using a grassland soil because we found out recently that they could be super important for carbon storage by having these long roots. So we took a grassland soil and we had 4 different treatments. We either had our water or we had our heavy water to look at the active ones. And we added phosphate because we thought, viruses use a lot of phosphorus when they make their capsids. So we were wondering if it's critical for infection. So we had some treatments where we added that. We also wanted to know how these viruses changed how they affected the ecosystem and themselves, how they changed over time. So we sampled over the course of a month. before we're adding the water and after we're adding the water. And what's important here, if you're not from California like I originally was, and you don't understand this, that the whole summer there is no rainfall. It's great for tourism, but you would think it sucks for the organisms in the soil. How do they survive half the year with no water? So they have no water for half the year, and then come to the fall, they have the first rainfall. You get this birch effect. You get water being added, and all these organisms come to life. And so much of the carbon is lost after that first rain event because we're now giving substrate to these organisms to reactivate. So this is what I want to study. So we're adding the water here to these dry soils to really see what's going on here. But it wasn't just metagenomics I wanted to do. I spent my whole PhD optimizing methods for what we call viromes, or viral targeted metagenomes. And as you can see from the figure here, it's a multi-step process where we take soil, we wash it with different buffers, and then we physically separate by size fractionation, typically a 0.2 or 0.5 micron filter, to get things of virus size out of the soil, and then we sequence that. So I'm showing here visually comparing the circles. We get so much more information on the viruses from this virome. We also, funny enough, get information on ultra small bacteria and archaea as well. So it's a really cool technique. And so the con about this, though, is if you just take a virome, you're not necessarily getting all the hosts and less of the ultra small hosts. You kind of want to take both if you want to relate viruses back to the ecosystem level consequences from mitigating their hosts. On top of this, though, we wanted to take a metatranscriptome, and this is going to be looking at all the RNA from the soils. And this is important because there's all these viruses out there that are RNA. Viruses are the only biological entity that we know can have double-stranded and single-stranded DNA or RNA. Some can reverse back and forth between that as well. So we needed to get the RNA as well to look at these viruses. Finally, something that we kind of hit on here and invented some methods for is we call this eDNA metagenome. So in the past, it's been called ancient DNA, relic DNA, degraded DNA, blah, 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 or environmental DNA or exogenous DNA. So this is just DNA that is in the environment. And I wanted to see, okay, if we have DNA in the environment, what's its origin, what's its function? Is it being used in biofilms? Is it coming from organisms dying? Are other organisms eating it? Is it a buffet? What's happening? And by taking all these methods together, we can get a better understanding of the whole virus here from all these different components. Okay, so relating it back to eDNA, we've only gotten to a little bit of data, so it's preliminary, but I wanted to share some of it with you. So in this figure here, I have our time points. The T0 is pre-wet up, and then we have 1 week, 2 weeks, and 3 weeks after we added our water. And on the Y scale, we have the log scale, right? So huge changes between the amount of eDNA we're seeing. So at the T0, we have a lot of eDNA. So which means if you're taking a metagenome, you're also seeing DNA, not from inactive organisms, but also not even inactive organisms at all, just in the environment. So what's going on there? Okay, so it accumulates over the dry summer. This is my leading theory so far as I'm investigating. And then we have this downshift. So what's going on? Okay, well, we added water. Organisms now have this water to use to fuel their metabolism. And this DNA is juicy. They're hungry. These are cheeseburgers or salads if you're a vegetarian. They're loving it. They're like, no, 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 no. So the eDNA is dropping. The microbial populations are increasing. And we see that. We see the number of microbial populations and their abundance increasing. Then we kind of have a level off a bit. And we have viruses killing microbes. So we have the microbial populations dropping. We have a little bit more of eDNA going into the environment. So viruses are contributing to the eDNA in the environment, not by killing the host and their necromass going out there. But then we see maybe the community stabilizing, where we have this arms race where viruses are killing microbe, bringing the population down. That microbe becomes resistant to the virus, so then it can start increasing. So we kind of get this leveling of the eDNA in the environment. And when we actually compare, where is this eDNA coming from? So on this other figure I have here, I have our viral populations on the y-axis, and I have our microbial populations on the x-axis. And then the dots represent who does the eDNA belong to? And the majority of it, if you look at our line that I provided for you, actually belongs to the microbes. So they're coming from these microbes that are being killed, or the microbes could be exuding it to build biofilms and survive. Okay, so I want to make sure I have enough time to give everyone questions. But one last thing I wanted to talk about before going into my astrobiology shoutouts is I want to talk about other ways besides bioinformatics and omics that you can look at viruses and look at detection of life. So nanosims, which is nanoscale secondary ion mass spectroscopy. This is where we can use a beam to destroy a substrate, a particle, something, and we reconstruct it into a picture. So we've been pushing to be able to look at viruses this way. And what's cool is we can have nanosims on a rover to go somewhere else, and we can actually see things that are alive or that we consider alive. So I have here some TEM images where we've been purifying to look at viruses. So we have this Cephalviridae. It's a Categorizing, it's a double-stranded DNA bacteriophage that we were able to see with the nanosims, and then we are on TM, and then we put on the nanosims. On the top right, we've been able to see these other cool filamentous viruses, these really cool ones. And the specialty about seeing something with the nanosims is you can physically see it, so you know it's there. It's not just a dot or something that we have to infer. You're seeing it, that it's there. But also, we can add in isotopes to actually see if it's active. So here we use the 13C, so we know where carbon's incorporated in the virus, and we know that the virus is active as well. So you might have heard about the Viking missions in the past. We've come so far since then. So this is exciting stuff to think about for the future. Okay, so my quick shout out is that we really wanted to push viruses in astrobiology and really get astrobiology. You don't have to be someone who studies viruses to get into it. There's no correct or direct path to be an astrobiologist. If you study life in any form, whether it's physics, chemistry, biology, you can become an astrobiologist. You care about the questions you're asking. So we did a white paper to increase awareness and funding for viruses, and that's available now if you want to go to the link. And this is really just how to incorporate viruses into astrobiology. And we had questions that we asked. And what was really cool is I got to work with Penny Boston, which was the former head of the NASA Astrobiology Institute, Ken Stedman, who is at Portland State University, and my good friend Catherine Bywaters. We also, so that was already published, we also wanted to change our framing and get other people to be excited about astrobiology as well. So the first one is everything viruses. Viruses are cool. You should love viruses as well. Our second one, which I have in the bottom, which we're calling viruses as modulators of cellular metabolism. This is specifically how we can use viruses for long-term space travel or short-term space travel, and what are the effects on human beings of viruses, or even on keeping our ecosystem. Viruses infect plants. So how do viruses affect plants in space? Viruses infect algae that we're using to clean up our waste or make food. They also affect us, or if we come back. We need to understand this. Viruses are also really small, and they've been detected even in our clean rooms. So we also have to think about viruses for planetary protection as well. So that's one that we submitted last fall. And so hopefully it's gonna be published this year. So just keep an eye out for that. I also wanted to point out that in 2019, NAI funded our astrovirology workshop without walls. This is a free 2-day event that we did spaced out over 2 parts of the day to really engage the whole world. So I have a map here of everyone that was active during it, but I've had many people from other countries reach out to me that was helpful. So both of them are available on YouTube. You can watch the whole thing, the questions, everything is there. We've tried to organize it into 2 themes, but everything's about viruses on Earth and the potential for space and how viruses can have all this different nucleic acid. So I implore you to please check it out. Email me if you have questions. I'd love to get feedback on it. Hopefully we can do more workshops like that in the future. The final thing I want to say is we're going to AbSciCon this May. Graham was talking about how he's going to be there. I'm going to be there as well. Please come see us. Please join our session. We have 2 amazing speakers, Dr. Rachel Whitaker and Olivia Negro, and we're talking about viruses, everything viruses. And what's cool is at the end of the session, we're going to have kind of a town hall with all the speakers to really discuss where the field is going, what are questions you have, how you need the field, where do we need to push to really get people to start thinking about viruses. Whether you consider them alive or not is semantics. They are integral to life. As far as we know, they've been here since life, if not before life. So when we're looking for life, why not look for the most abundant thing that's related to life, the most abundant biological entity, which are viruses. With that, I just want to acknowledge the diverse teams I'm working with at Berkeley, at JGI, at Lawrence Berkeley National Lab, Lawrence Livermore National Lab. We have our science focus area grant that I'm working on. I also want to talk about funding from our LDRD, these high risk, high reward that have really pushed our science and have been foundational for really understanding viruses in all ecosystems. I also would just want to do a land acknowledgement for where our studies are taking place and the people who took care of those lands before we got there and are taking care of them now. So we appreciate that. And then I want to end with questions. So here I have a nice diagram that Penny Boston made about, viruses are not considered life sometimes. They're not incorporated into the tree of life because we use a marker gene that viruses don't have. But viruses infect all life. Viruses even infect viruses. So we need to be thinking small and thinking viral. And with that, I'll take questions.
Great. Thank you so much, Dr. Trubel. That was an amazing speech. Okay, so we have a few questions for you. Number one, with such a small proportion of reads mapped to viruses, how do you know of the actual sequences found with low coverage?
Well, that is a great question. So let's take a metagenome and let's say you have 100% of your data. This is the data you get. These are what we're going to call our reads. So less than 2% go to viruses. But if you actually look at a traditional soil metagenome, depending on where it's from and how they're doing it, not all those other reads are going to be used either, right? I said 2%, but less than 2% of the assembled reads. So we have all these, the majority of reads actually not being used because life is so diverse and so complex that we just can't even tease it apart. So then we're just looking at data that we can assemble, so the data that we are aware of. Okay, so looking at this usable data, how do we know if something's virus or not if it has low coverage? Well, we need to increase our coverage. So that was why I traditionally pushed for the virome approach and really looking at the viromes. And then you're saying for those few viruses that we get from metagenome, how do we know that those have coverage? Well, that's what's tricky is in a metagenome, you're also getting hosts, right? So a virus can be integrated into a host. When it's integrating a host, if that host is highly abundant, every time its genome is duplicated, the virus's genome is also duplicated. So if the host is highly abundant, the virus is also highly abundant. So when I said less than 2% go to viruses, the ones that do go to viruses go to those highly abundant viruses. So we get low diversity, but we get high coverage or relatively high coverage of them. Now, you're right, though, it's hard, especially with variants in either a technology or that just exists within populations. How do we know what is and what isn't? So a lot of times we get incomplete genomes where we're just recovering the parts of the genome that have a lot of coverage. Or we use great tools out there. Simon Rue made VirSorter, which is one of my favorites, and now there's VirSorter 2, that really look for hallmark genes or parts that relate to a virus, like strand virus or short genes, to really say, hey, this is a virus, and it gives you confidence and rules with that as well. But yeah, to really increase the resolution of viruses, you have to decrease the complexity of the sample. I had tried viromes before. I'm now using stable isotope probing. There's so many other methods out there that we can push to really increase our resolution on viruses. There's now single cell genomics. There's microfluidics being used. Arguably, you could use a TEM to try to look at part of a sample. I mean, there's so many different ways we can do, and I hope that answers your question.
Yes, thank you. The next question is, has there been any evidence to suggest that there are archaea that associate with viruses in soils?
Oh, absolutely. Yeah, sorry. It seems biased here that I'm like bacteria. I just want to step back and say, most of the viruses we see in soils are bacteriophage, which are viruses that infect bacteria, and most of them are double-stranded DNA viruses. And if you think about metagenomics and our sequencing, we were set up to look at what we thought of as life. So this is humans, plants, fungi, bacteria. They have double-stranded DNA genomes. So our sequencing technology is set up for that. So it's no chance that, this is why we do it. This is why we have double-stranded DNA viruses, right? This is why we can easily see them, or easily. So There are ways out there which you can start to get a single-stranded DNA viruses. I had published some papers that way. We have to really think about when we're prepping our samples for sequencing and think about that single-stranded DNA, or if you're thinking about relic DNA or ancient DNA, the highly related DNA is also usually single-stranded. So if you think about them as the same single-stranded DNA viruses in line with eDNA, you kind of are going to find methods to help you get that. Finding RNA environments is really difficult. So there hasn't been a lot of work with metatranscriptomes, and I've been trying to push that. Others have as well. This one day we're going to get an RNA virome to really look at the encapsulated RNA viruses if they are the majority out there. So yeah, I don't know. Can you rephrase the question or say it again so I can make sure I answered it?
Yeah, just asking about like archaea associated viruses.
I was going off on a tangent. So sorry, everything was set up towards bacteriophage. So for archaea, they also have viruses, but what we know about viruses is largely come from isolates, and we can more easily grow bacteria compared to archaea. So we also have more isolates on viruses than in fact bacteria, so we know more about them. So I talked about virus order before. Well, it looks for enrichment of genes for caudovirales, which are bacteriophage. So they also push towards bacterial viruses. We have found some great archaeal viruses. Ken Stedman has done it in hot springs, which are not soils, but you get some cool lemon-shaped ones. We know they exist in soils. There's some previous papers. I'm trying to think of the names right now. We know that they're there. Why we don't see them as much? People have said, oh, archaea have more CRISPRs or less CRISPRs. You've heard it both ways. They're less abundant, so viruses don't care about them. I really just think it's our sampling. They are there, but we know very little about them, especially in soils. As you probably know, that if we're going to find them, it's usually in hot springs or these other aquatic environments. Sorry, I hope I could answer your question, but there's not much more I can give you on that.
Yes, definitely. No, thank you. Okay, so I have one more question for you. Sure. Would you consider virus to be life's fail-safe against mass extinction, or are there more of an intermediate step that have found a new way to propagate?
So let me make sure I got this right. The fail-safe against mass extinction in terms of the virus controlling these populations to make sure everyone else has a fair share. So if I think of it in that way, yeah. I would say yes and no. I mean, I don't think anything's engineered that way. I think you have a biological entity that is selfish. It wants to replicate, so it goes after the most food. If you had a buffet out, that's not the best comparison, but because, yeah, you're more likely to go where there's resources. So I guess a better comparison would be if you knew you had to survive somewhere for 3 days and we had a beach that was barren or desert land, and then you had a condo here with all the amenities, you would choose a condo with the amenities because you want to make sure you're caring for your progeny, your offspring. You want to make sure they're still going. So viruses, I think, have just evolved to kill the most abundant. I don't know if it is a way for nature to control itself. I mean, there's the Gaia hypothesis is, it's self-regulating this giant mega ecosystem. And if you want to go down the road, I could agree with you that viruses are doing a good job at keeping diversity, at regulating that. Now, what's tricky, though, is they don't do it alone, right? The hosts have to adapt to survive. If it was just up to the viruses, they would kill and make a population go extinct, right? And in cases where we have like phage therapy, that's kind of the goal. We're trying to get a population to go extinct. So we're also relying on the host to be able to evolve to outcompete it. So is it the host that's the failsafe, that it learned its lesson and is coming back, if we're going to personify it? But I don't know. And then I think you're leading to, when it comes to humans, that gets more tricky because we metaconjugate. We can think about thinking, we're self-aware, and we can change our actions. So unlike this microbe that has to evolve genetically to survive, we can take stances where we can fend off the virus, right? Where we can stop the thing that's trying to control us. So there's so many different ways. I don't want to go too far down that rabbit hole, but I know what you're thinking, and I don't think life is malicious. I think everything is trying to do its best, and the virus itself wants to propagate and it wants to keep itself happy, so it's going where the most resources are.
Definitely. Okay. Thank you very much.
Of course.
Well, that was, again, a great talk by Dr. Trubel. And now I think we are going to have our prize distribution ceremony for the poster session.
Yes. Thank you so much, Laura. And thank you so much, Dr. Gary, for your wonderful talk. Now, let me share my screen. And the participants who can switch on their cameras, it would be great if they can, because it would be nice if we all could applaud all the winners together. Okay, so we have had a lot of abstract submissions. We then finalized some of them as the posters that we wanted to have it up in the competition. And we have 3 categories. The first category is for best technical and research project on a competitive topic. And the award goes to, it's for Ponraj Vridangana, and her topic was on microbiome of 4 different lung diseases, a comparative 16S RNA metagenomics to understand the diversity and functional profiles. So congratulations, Brenda. Can you please turn on your camera and say a few words? I think we need to give her the permissions. Yeah.
Hello,
yes.
Can you hear me?
Yes, we can hear you.
Yeah, thank you very much. And thank you, OmicsLogic and all the judges to recognize my work. It was a great pleasure to present in this international symposium, where I got opportunity to learn and explore recent advancement in bioinformatics from scientists across the globe. Thank you very much. And thank you, everyone.
Thank you so much, Brenda, and congratulations once again. And the second prize was a tie between 2 groups. And the first person who has got the second position for most promising research direction for future potential is Dr. Iman. And Dr. Iman's research topic was on racial disparities in thyroid cancer biomarker testing. Does one size fits all? Congratulations, Dr. Iman. Can we hear it from you?
Hi. Can you hear me?
Yes, we can hear you.
I'm so delighted to receive the award at this research symposium for 2022, and I'm really humbled and encouraged by this recognition. But really, I can't emphasize enough that I would achieve nothing without my best mentors and colleagues. Their hard work, dedication, and innovation are a pleasure to be part of. So it was a great opportunity now to share a bit for 1 minute my experience, if you can allow me for that. It's during my research fellowship in biotech, I was so grateful to learn new things every single day, stepping from diving into omics to actively engage into real life research. And that was really amazing for me, being able to complete the work, connecting the dots, inspired even by other ideas how to improve myself. So with the current problem is and challenge is the thousands of omics studies that's available online and published each year. It's becoming more difficult to wade through the sea of information to achieve insights and make new discoveries that will be the basics for your future grades and projects. And that's why scientists, even those who are non-specialists, need to have minimum knowledge, skills, and tools to interpret this immense amount of data on what has been done before and understand how this is related to your own data. That will be saving money and time. So I'm supporting those not only specialists in the genetics or bioinformaticians to be enrolled in this fellowship, but also all other specialties, even clinicians, even non-scientists, because minimum knowledge of these will upgrade your research activities. So while my primary research interest was oncology to understand disease onset, risk, and progression, I would like to pinpoint that there was other opportunities to learn other cross-omics domains like metagenomics, genomics, infection, as we saw in these 2 days. So it's like a multidisciplinary opportunity in a single path. I've witnessed continuous improvement in the content of the courses during the last 6 months. I get this is based on the continuous feedback of the research fellows. And I even learned from the experience of other fellows and the troubleshooting they faced through the weekly research fellowship meeting. That was very fruitful. So you have now availability, the courses, the projects, the programs that's available in the OmicsLogic and the Tumor pipeline had the potential to accelerate our research goals from just discovery, new target identification, validation, to drug repositioning, to cross-omics and saving lives. So at this end, I would like to thank everyone, even those behind the scene like Sonalika, for supporting me in the long journey. And I'm thankful to my mentors who I kept sending a lot of questions all the time, day and night, and they kept replying back. So thank you so much for supporting me in this learning journey. And I hope everyone else got enrolled soon and enjoy this experience like what I have busted through. Thank you so much.
Thank you so much, Dr. Iman. These are very kind words, and it feels good to hear about the research fellows' experience, that they could learn so much and gain a lot from this program. Congratulations once again. And the second prize was a tie, as I told you all. So the next group that is also selected for the most promising research direction for future potential goes to Mr. Ojasvi Datta and Mr. Shubham Kumar. And their project was on effects of microgravity on high PSCs, cardiac and muscle activity. So this was a group project between Ojasvi and Shubham. So can we have them both? Yes, Ojasvi, I can see you. Ojasvi, what would you like to say?
So I want to thank everyone here, and especially Dr. Raghavendran and Ilya. So without their guidance and support, we could not have completed this project, and they pushed us a lot to complete this. And thank you so much, Sonalika, for making me join Pine Biotech. And this was a great journey for me till now here, and I have learned so many things, from starting from transcriptomic analysis and ending to the astrobiology project. So I have done what I feel is this is the really amazing time for me today, and I am grateful to receive this award.
Good to hear that. Congratulations, Ojasvi. And what about you, Shubham? Are you here with us? Yes. I saw him. I think we need to, yes, he's a co-host. Yes. Hi, Shubham.
Hello.
Okay, he has a background showing that he has an astrobiology project. That's amazing. Yeah.
So I would like to thank Ilya and Dr. Raghavendra for their support, and obviously OmicsLogic and Pine Biotech to make me join as a research fellow, and to Sonalika as well. And thank you everyone to help me in this research fellowship, and thank you to all judges.
Amazing, very good. Keep it up, and congratulations once again, both of you. And now let's see who has got the third position for most innovative approach in an established domain. It's Andrew Lee, and his project was on a cell type specific machine learning and protein interaction approach to identify autism risk genes. And also to add here, Andrew Lee won the first prize in our last symposium, and he has been doing amazing jobs with respect to his projects all the time. So we are pleased to have you with us once again, and congratulations. Are you here with us, Andrew?
Is there like
Yes. Angie, we can't hear you. Okay, so meanwhile, Andrew unmutes himself. Let's do one thing. Can we have a group photograph with all the winners and also the participants? If they can turn on their cameras, it would be amazing. So we can capture some beautiful moments from this session. I think I need to. Okay, so who all do we have switching on their cameras? Andrew, are you here with us? Thank you. Thank you, everyone. So with this, we are good to go ahead with our next session. And now I'll pass on the stage to Laura to take the session ahead. Over to you, Laura.
Thank you, Sonalika, and congratulations to all those winners. Let me share my screen. Okay, so our next speaker is Mr. Elia Brodsky. Mr. Brodsky is one of the co-founders and CEO of Pine Biotech. He has facilitated and managed collaborations with researchers at Stanford, UCSF, Louisiana State University, Tulane, and other academic and pharma R&D teams. Mr. Brodsky oversees the research projects and manages the development of the prototype, preparation of relevant data projects, and implementation of the system for the needs of the students and teachers that the project is designed for. So please join me in welcoming Mr. Elia Brodsky.
Thank you very much again. Congratulations to everyone. And it's really exciting to see how all of this actually connects. And I'm really pleased to be a part of this symposium today. And what I want to share with you today is actually how we're going to continue and expand the OmicsLogic program. So what we saw in the past couple of days are the multiple directions that bioinformatics can play a role, from precision medicine to pharmaceutical companies, biotechnology companies, astrobiology, origins of life, right? It's not really a domain-specific discipline, but it empowers researchers in many different directions. And so what I want to talk to you about today is first, how did we establish this program in the first place, where it's going, and specifically focus on one aspect, which is the program that we're developing together with the National Association of Idea Principal Investigators. So the National Association of Idea Principal Investigators is a network of idea-funded PIs. IDEA program is an NIH-specific program designed to elevate biomedical research. And so what is common across all of these different states that you see on the map is that they don't receive as much funding as some of the hubs. But I think that actually speaks to a general problem that is also beyond the United States. Unfortunately, the money is not equally distributed across all institutions. And so I think in the US, it's exciting to see how special allocation of funding is making high-level research possible in these states. But if you go to, we're located in New Orleans and Louisiana right here. And if you kind of compare how things are done here, you might see a lot of differences between, let's say, what's happening in Boston, Massachusetts, San Diego, or San Francisco, and what's happening here. And similar to what others experience around the world, there is a challenge to attract talent, and there's a challenge to make this talent work together, focusing on local problems and accelerating research that addresses local issues. And that is what this program is specifically designed to do. The funding, the IDEA funding, is really designed to help establish sequencing centers, cores, really enabling technologies so that other researchers that are striving to pursue advanced topics in different areas of research, especially biomedical research, can be successful. And across all of these states, there's a need in bioinformatics. So over the course of the past just several months, we've started a pilot with the National Association of Idea Principal Investigators to design a resource and implement programs to support training in biology as a data science. So I'll briefly talk about OmicsLogic for anyone who's here and who's not heard about it previously. So OmicsLogic is a portal designed to kind of combine multiple things that we saw today. And it starts from a portal that you can find on this link, learnomiclogic.com. You can register for free, and you become a part of the community once you register. It's a free registration. You get access to not only free resources, but importantly, a network that we're building. And I think a network that you have seen today that combines experts, students, faculty, researchers, and others. And importantly, what this portal allows us to do is it allows us to combine the learning and the research around a personal profile. So in the personal profile, we've designed elements like sharing about your background, connecting your social media, tracking your progress of learning, badges for different programs that you complete. And it allows the faculty to see how the progress in your learning matches with the objectives of whatever the program pursues. So importantly, we've designed a process of certification that actually allows us to make sure that people, when they become interested in a particular area of research, are actually ready and prepared and informed to apply bioinformatics in a meaningful way. And so after you receive this basic certification, then the next step is to make sure that your profile is complete and you're able to pursue your research topic, your idea. So some of the projects that we saw yesterday in the poster presentation, today we had Dr. Iman Touria and Ojasvi. So they were a part of the research fellowship, and actually many, many more students participate in this process every semester, attempting to complete novel, independent, or group projects using publicly available data. And so we've built this format because we think that, you know, beyond certification and kind of basic skills, really the validation that you know something is when you can apply it to something of your own. And because bioinformatics is such a diverse and dynamic field, because natural interests are so different, we cannot design programs around everything. So we're opening it up for anyone who would like to pursue a project to really be able to do that in a reproducible way. So once that is done, we are actually going to do biannual events like this one to bring the community together, select the most promising projects. And one of the things that we would like to do is actually turn those projects into opportunities for others to learn. So it's not just a way to learn and develop your own research, it's also a way to contribute and share what you have done with the broader network and the community. Beyond this, with this expanding library, and I want to pause here, by the way, and say that one of our objectives in the next few months is to open this platform to other contributors. And we've started working with several programs around the NEPI Southeastern region to start working on new materials to add to the library that we've developed so far. And this is going to be around several different basic topics like genomics, transcriptomics, metagenomics, or epigenomics, data science, and a general free course that we're going to make available for anyone to kind of take and use as they need, called Introduction to Bioinformatics, or Bytes and Molecules. And so that is really one aspect that I wanted to highlight today, is that One of our missions as a company is to make bioinformatics more accessible, make it easier to participate in, make it easier to apply it practically, and make it easier for someone who does not have any technical background to really benefit from it as soon as possible. So in a faster way to kind of appreciate the role and the meaning of bioinformatics. And so for that, we are creating this free resource. Anyone can use it. Anyone can, as a faculty, use it as a resource for teaching. And in fact, we're going to start a partner program that it will allow for the more advanced topics to follow after this brief and maybe optional introduction. And one of the things that we're going to include in this package is not just the basic modules like Introduction to Bioinformatics or Bytes and Molecules, or getting started with bioinformatics in R and Python, but importantly, provide a view of these many different topics like we heard in the past couple of days. How is bioinformatics used in precision medicine? How is bioinformatics used in astrobiology? How is bioinformatics used in pharmaceutical companies? Drug discovery, all of these topics, they are the things that drive bioinformatics forward, right? So bioinformatics, I think, is not driven by just computational and algorithmic innovation, but primarily it is driven by people that understand the data challenges that these domains bring. And so another part of what we are really passionate about as a company is not just the diversity of bioinformatics applications, but also how these bioinformatics methods and tools are used by experts. So obviously, it is great when we can have speakers like her today and yesterday come and present a topic that they have been studying for decades. But I think it's equally important to appreciate that even these presentations are based on a hypothesis that has been addressed in some way with evidence, but also reviewed by peers, by the broader community. And so that's why the key, we believe, is to focus on publications. So these peer-reviewed publications that we can review and extract kind of the methodology used by experts, including data that was submitted by them and made publicly available, is the foundation for these courses and projects that we develop on this portal. So the portal really provides 6 basic courses that anyone can take and kind of we standardize and make sure that we improve the content of these courses that include Introduction to Bioinformatics, Genomics, Transcriptomics, Metagenomics, Epigenomics, and an Introduction to Data Science. And on top of the basic level, we actually introduce specialization tracks. And this is how bioinformatics is used in oncology, in infectious diseases, in neuroscience, and in agriculture. And I'll talk a little bit more about some of these upcoming programs in the next few slides. What's important, though, is that as a part of this coursework, we have a package of enabling technologies. And these enabling technologies are designed to, again, lower the barrier for entry, speed up the process of going from base to more and more complex topics, and be able to identify the best track that fits your current stage and your objective in the future. What do I mean by that? A lot of students might be pursuing bioinformatics as a degree, but I think an even greater proportion of students are not directly interested in bioinformatics. And so for many, as we've seen yesterday especially, this interest in bioinformatics is something that shapes over the years. And so it's not appropriate to dump all the complexities and kind of the requirements for coding languages and statistics right up front in that process. And so we try to present a variety of paths that you can pursue to be able to analyze data and make discoveries, which is the exciting part. And if you choose to do so, enough depth for you to also learn how to develop your coding skills, how to get certified in different levels of biostatistics or machine learning, and even how to pursue a more technical resource for cloud computing, for example, as we will hear today as well. So one important aspect of this is that we have code and no code bioinformatics paths. And that means that you can either perform analysis using the big data analysis T-Bioinfo platform. And we've heard today about new sections on this platform. It's a dynamic place where experts contribute new and more efficient workflows, as well as learn languages like Python and R to learn how to program your own or use existing packages. And so in a variety of domains, I think that you will see how bioinformaticians range from biologists that are experts in using these packages with understanding of the different settings, with understanding of when to apply what kind of methodology to answer a specific question, as well as those that go the technical route, and they are able to engineer novel applications and make these applications user-friendly and more efficient for discovery. So with that, I think that, I'll briefly now speak about several different programs that are coming up that you can participate in. And we have our team here with us. So if you are interested in any of these topics as a faculty or as a student, I encourage you to reach out. And at the end, we will share some of the contact information for you to be able to connect with the right person. So I think there are exciting many new ideas and challenges arising in the field of gene expression and transcriptomics. And so traditionally, we've run a program called transcriptomic data analysis, or RNA-seq, where we explored bulk RNA sequencing, and the volume of bulk RNA sequencing has really been growing over the years. So at the foundational level, it was always important to understand what is RNA sequencing, how do these next generation sequencing technologies work, what are some important methods to explore and understand this kind of data, design statistical hypothesis testing techniques. And as of recent, it's been really important to also understand the differences between bulk RNA sequencing and single cell RNA sequencing, which is a technology that is becoming more and more popular and applicable in many different domains. So we've designed a program and standardized it to include some specific aspects that can be taught either in a short setting where face-to-face students meet, or supplement that with asynchronous training exercises that one can complete both on the OmicsLogic portal and utilizing the T-Bioinfo platform. So for that, we again have a section. The section is color-coded and explained, and it really covers how you can analyze data from understanding the inputs and outputs to the logic behind the analysis that is color-coded and explained in detail. Why do we have to perform each one of the individual steps? And I think what we've seen as a result of doing this program multiple times is how this approach helps students appreciate how any kind of analysis can be structured in a very logical way. After they understand the logic, we then introduce a variety of different types of projects where they can see the application of RNA sequencing techniques in oncology, in precision medicine, in infectious diseases, and epidemiology. With the popularity of single cell, we've seen a kind of like an expectation that this is just exactly the same as bulk RNA sequencing, and it really is not. There are a lot of new methods, there are a lot of specific approaches, and a lot of emphasis is put on downstream interpretation. And so as we have heard today, now we have a full section that kind of automates and simplifies the downstream analysis of single cell RNA-seq data using such packages like Seurat and Monocle. And importantly, we provide a step-by-step breakdown of each one of these logical steps using exercises that you can perform independently. And I'm excited to say today that this course is also available, and anyone can find that course on the OmicsLogic portal. Obviously, supplementing the basic training with project examples is a big part of what we are trying to do. And so we take these publications that have an impact in this domain, and we break down the methodology portion and really describe how one can analyze such data and replicate an analysis found in a publication. With so many different types of omics data, now it's important to not only understand how to process, analyze, and visualize a single type of data, but importantly, integrate multi-omics data as well. And that's a big part of what we do on this portal is introduce multi-omics integration methods that could be contrasted and compared and applied in specific domains with a benchmark. So an important part of what we do in the projects are curated data sets that one can use to establish an understanding and really test how well you apply the methods to get the same results. As we've seen over the past 2 years, infectious diseases from being kind of a domain that suffered from a lack of funding became a domain with exciting new research and applications that touch the lives of all of us today. And so back in 2020, we launched our Bioinformatics for Infectious Diseases program. And we quickly adapted that program to address the challenges around COVID-19 so that not only we're able to understand pathogens and kind of study those pathogens using project examples, but also think about applications. And I'll briefly kind of mention a couple of these applications that we've seen resulting from this program. So as a result of understanding the methodology and seeing how next generation sequencing can be used to study these viruses and different types of infectious diseases, we have developed a number of collaborations around wastewater sequencing of COVID-19, monitoring variants of concern and other types of strange phenotypes that have been reported in literature. And I think that this kind of interest in bioinformatics as a result of the pandemic, of course, the pandemic in itself was an unfortunate event that we're still not out of, but I think it really gave a lot of popularity to terminology, methods, and even types of data that will, in the future, I think this generation that went through this pandemic will be so aware and so familiar with some of the methodology that they will be able to address future challenges of similar character in a much more effective way. And so we've designed several project examples, a variety of different viral epidemics, and an in-depth training resource for SARS-CoV-2, where both consensus level and sub-consensus level analysis could be performed. Not only at the sequence level, but importantly also translating the sequence level to the immunology and to the protein-protein interaction level. So looking at 3D structures of proteins and understanding how they have an effect on vaccine design and protein-protein interaction. And so really at the core of this, I again want to emphasize how significant it is that this is available not only for engineers. The T-Bioinfo platform really hides the complexity of analytical pipelines and complex algorithms that have been developed by experts in the field to make it possible for someone who's a student or a biologist or a biochemist to apply these complex methods and understand how they are used with practical examples. So methods like processing FASTA and FASTQ files for multiple sequence alignment and comparative analysis of different sequences, reporting, generating a full report on variants of concern found in different types of viral populations, understanding how this data can be visualized effectively and presented. And also importantly, as I mentioned, mapping the mutations that we identify and understanding how they impact different aspects of immunology. During these exercises, we actually also look at protein-protein interaction, really trying to understand the available resources like protein crystallography, but also how to connect the different types of data and really understand the way that multiple sources of data could be used together. Now, yesterday we spoke with Dr. Sona Vasudevan from Georgetown University Medical School, where she leads a program on systems medicine. And together with her team and her students, we've developed this program specifically to introduce a non-engineer to the basics of data science and machine learning using R and Python. I think that it's a very critical skill for anyone to learn how to code. And I think that it's non-trivial for someone who doesn't come from an engineering background to do so. But we've seen how these resources can make it much easier for someone to learn how to code quickly and develop a practical sense of the application of languages like R and Python to address bioinformatics challenges. And so the exercises that we have available on the portal include mapping mutations for metabolism of different drugs, really learning how to code from a syntax perspective, from the perspective of data visualization, and the application to many different types of data, such as genomic data analysis, metagenomic data analysis, as well as different types of big data repositories that could be explored and visualized using such tools. To summarize, I think that what we have seen is an explosion of interest in different domains, starting from very basic usage of genomics in the lab to such interesting concepts like space omics. And when I mentioned that we're opening this platform to other contributors, this is really what I am implying. In different universities and different labs and in different research fields, experts have dedicated years, if not decades of their life to identifying important topics and learning how to address them in a reproducible and high quality way. And we would like to kind of establish a way for those experts to be able to take their expertise and allow us to design high quality training materials that could help effectively many of the people that are locked out of these domains to be able to participate by first learning about these topics and then knowing how to apply their skills and their insights and innovation and maybe creative thinking to address the topics that are pressing and urgent in each one of these domains. So we have these programs on cancer biology, precision medicine, space omics, and many others. And we're excited to share with you that these types of programs have already impacted hundreds of students in Louisiana, thousands of students around the world, and we're hoping that many more across the National Association of Idea Principal Investigator Network as we are completing our first pilot, which lasted from January until the end of March, so until yesterday. So with that, I am going to be available to answer any questions. If anyone had any questions, I'm going to share my email address. If you would like to reach out to me, I'd love to talk to you about these programs. I've shared my email in the chat. We will have a few more speakers from NEPI speaking about different efforts that we are going to kind of combine into this program as well. And I thoroughly enjoyed seeing some of the results that we have seen. And I think that is really the demonstration of what we are trying to do, some of the research projects that we've seen from the students. So thank you again. Yes, Laura, back over to you.
Great. So we are still waiting on one of our speakers. So Dr. Crabtree, are you available to speak right now?
I am. We can go ahead and go if you'd like.
Great. Okay, let me introduce Dr. Judy Crabtree. Okay, so please welcome Dr. Judy Crabtree. Dr. Crabtree received her PhD training in biochemistry and genetics from the University of Oklahoma in one of the original labs that was funded to sequence the human genome. She did her postdoctoral work at the National Human Genome Research Institute at the National Institute of Health in Bethesda, Maryland, under the direction of Dr. Francis Collins, studying the biology of an endocrine tumor disease called multiple endocrine neoplasia type 1. From there, she entered the world of pharmaceutical drug development at Wyeth Pharmaceuticals. In 2009, she left the pharmaceutical industry and joined LSU HSC as an assistant professor of genetics. She is currently an associate professor of genetics and the scientific and education director of the precision medicine program at LSU HSC. More recently in the pandemic, she implemented the COVID testing program for the LSU HSC campus and has launched the viral sequencing efforts to track COVID variants of concern in Southern Louisiana. So please join me in welcoming Dr. Judy Crabtree.
Thank you, Laura. So I'm going to share my screen now. Get me set up. Oh, come on. Okay. So thank you for this opportunity. I mean, this has just been a great meeting the past 2 days. We've seen so many great talks and posters. I'm very excited. And so what's interesting, though, is as I was preparing this talk, I realized that today is 25 years to the day since I started my postdoc. And so I had just finished my PhD work under the direction of Dr. Bruce Rowe at the University of Oklahoma, where we were sequencing regions of human chromosome 22 as part of the Human Genome Project. And on April 1st, 1997, I began my postdoctoral study at the NIH under the direction of Dr. Francis Collins. Now, I can tell you, if you've never started a new job on April Fool's Day, it is quite the experience. And so as the new green postdoc, I was immediately the April Fool and the easy target of many practical jokes in my new lab. But what I didn't realize at that point was that this was the beginning of a translational career built on genomics and its application to human health. And so I was fortunate to not only contribute sequencing data to the Human Genome Project, but to also be in the epicenter of human genomic research in the US upon completion of the Human Genome Reference Sequence in 2003. So at that time, I was infused with optimism about what we could do to understand human health and disease, how we could further understand what makes each of us alike, but also what makes each of us different. So how are we different in our responses to environmental stimuli, to pharmaceutical drugs, to chemotherapy? And since this day in 1997, I've studied cancer and performed large-scale transcriptomic analyses of several tumor and cancer syndromes. I've worked in the drug development and pharmaceutical industry. And then now here we are 25 years later, I'm translating what I've learned in each of these varied research environments to supporting patient management in our CAP-accredited clinical precision medicine laboratory. Now, in order to understand the current state of affairs with respect to precision medicine, we kind of need to start at the beginning. So I hope this isn't repetitive to some people, but it's always fun to look at history. So in the late 1980s, a group of scientific thought leaders that included Francis Collins of the NIH, Eric Petrinos from the US Department of Energy, Eric Lander from the Broad Institute at MIT, and several others, they sat down over a pint of lager and outlined on the back of cocktail napkins a plan for mapping and sequencing the complete human genome. So this project launched in 1990 and ultimately took 13 years to complete at a cost of about $2.7 billion. And so here you can see the publications, the cover of the public effort in Nature and the competing private effort in Science. Now, the key thing that I want you to remember is that the Human Genome Reference Sequence is not the genome of one individual. It's a compilation from the 20 anonymous individuals, mostly anonymous. Further, the 20 individuals were selected from a pool of over 1,000 donors whose information was ultimately de-identified so that even those who contributed do not know if their DNA was used or not. And so this was done intentionally by the architects of the Human Genome Project to ensure that no single person's genome was exclusively represented, and also so that no individual could be identified by their contribution to the reference genome. Now, once the reference genome was complete, this opened up a huge number of questions about the genetic diversity of humans. And so one of the astonishing facts from the Human Genome Project was that single nucleotide variants occur approximately 1 every 300 nucleotides. So if you do the math, that means there are roughly 10 million single nucleotide variants in the human genome. And these variations are one contributing factor to the genetic diversity in humans. And so this opened up a whole new set of questions. So what is the intersection between disease and genetic variants? What's the relationship between drug metabolism and genetic variation? Is there a cancer genome, and does that cancer genome change over time and in between the primary tumor and a metastasis or a recurrence? How does the genome impact mental health, behavioral health? It had already been observed that some diseases were more prevalent in certain populations. And so then questions about what's the role of genetic variation in ethnicity. And so over the years that followed the completion of the Human Genome Reference Sequence, it became more and more clear that we knew too little. The genomic community as a whole began a sort of haphazard way collecting information we needed to answer some of the questions about genetic variability. But as time went on, the cost of whole and as time went on, the cost of whole genome sequencing decreased. So now it's approaching $1,000 with a turnaround time of 7 to 10 days. And so now the big picture idea was that if we can perform genomic testing at an acceptable cost, get private insurance and Medicaid, Medicare buy-in to perform genomic testing in the clinical setting, we can use this as evidence-based medicine to identify the best therapeutic and preventative interventions to best benefit our patients. And so to set this up in a more organized fashion, we realized that there was a need for a more concerted effort to understand genomic variation. So the NIH announced its support of precision medicine in 2015 when then President Obama in his State of the Union address described precision medicine as an emerging approach to disease prevention and treatment that takes into account people's individual variations in genes, environment, and lifestyle. So this was accompanied by a publication in the New England Journal of Medicine written by then director of the NIH, Francis Collins, and past director of the National Cancer Institute, Harold Varmus. This perspective further supported the convictions of President Obama that science offers great potential for improving human health. And this officially launched the NIH Precision Medicine Initiative in 2015. Now, a key part of the Precision Medicine Initiative is the All of Us research program. This was launched officially about 4 years ago, and this is a cornerstone of the Precision Medicine Initiative that seeks to enroll 1 million or more participants that represent the broad diversity of the United States so that we can understand variants much better than we do. So patients will complete surveys, provide biological specimens for whole genome sequencing, and then grant access, if they choose, to their medical records for de-identified population-based studies that correlate genetics and genetic variants with disease and environmental factors such as where you live. Now, the All of Us Network is composed of national partners and regional medical centers across the U.S. to enroll participants from all walks of life and all states of health within the United States. So just this month, the All of Us research program hit a key milestone and released the first set of nearly 100,000 whole genome sequences. More than half of this new genomic information comes from people who self-identify with racial or ethnic minority groups. And that's extremely important because until now, more than 90% of participants in the large genomic studies were all of European descent. And so this lack of diversity has had huge impacts. It deepens the health disparities and hinders scientific discovery from fully benefiting everyone. So I do want to point out that LSU HSC is a flagship institution in the southern region of the All of Us program. We affectionately call this program the All of Us Network, for those of you who know about the southern dialect. And this is in collaboration with Tulane University, the University of Mississippi Medical Center, University of Alabama, Birmingham, and other major institutions across the Gulf South. So this is the website where you can learn more information about the All of Us program, and there's still time to join either through the website down here or the app, which looks like this blue banner up in the upper left corner. Once you've registered, you can then make an appointment to donate your sample for sequencing at one of our participating clinics. So all of this is going to contribute to our understanding of the human genome and how genetic variants can impact our health. And so what I'd like to do now is share a study with you that was one of the first studies to come out in PLOS Genetics in 2011 to show you the predictive ability of precision medicine. And so this study used next generation sequencing to evaluate human health in a predictive way. And in this study, the authors performed whole genome sequencing of 4 members of a family: mom, dad, daughter, and son. And as an aside, the family used in this study is the West family. And if you look at the author list carefully, you can see John West and Ann West are both authors on this study. So John West was the CEO of Solexa at the time. So this is the company that developed the current next generation sequencing technology that was then sold to Illumina and is now the foundation for their current almost market cornering technology for next generation sequencing. And so this is the pedigree for the West family. If you aren't used to reading pedigrees, the squares are males, the circles are females. Horizontal lines indicate either a marriage or siblings, and diagonal lines across the symbol indicate that the individual is deceased. Now the arrows indicate the 4 immediate family members of the West family. So we have John, his wife, the daughter Anne, and her brother. And note the multi-generational history of cardiovascular and blood clotting problems within this family. So there are several incidents of myocardial infarction, congestive heart failure, deep vein thrombosis, and hypertension. John was hospitalized twice with venous thromboembolism in 2 separate incidents during the period of time when he and his doctor were working out the appropriate anticoagulant for him to be taking for his condition. So the whole genome sequencing of this family identified, among other things, multiple variants in genes related to blood clotting. So this process utilized 2 carefully curated databases, VeraMed, which correlates genetic variants associated with disease, and PharmGKB, which catalogs genetic variants associated with drug responses. Now, there are many such databases available now. These were just the first 2 that were established at the time of the study back in 2011. Now, in a blinded bioinformatics analysis of these data, the investigators were able to correctly predict the type and the dose of the anticoagulant that John was already taking. So he and his doctor had settled on clopidogrel after an extensive period of trial and error to get the dose and drug correct for one that worked for him. Now, imagine if John had been screened for known variants before he even took any medication. Well, that's essentially what happened for his daughter. So the study predicted what medication she may need in the future based on her genetic profile. And interestingly, the medication is different from that her dad is taking, mostly because, remember, everybody inherits 2 alleles. Anne inherited 1 risk allele from her father and a different risk allele from her mother. So Anne was 17 at the time of the study, and now she's 29 years old and has no health problems as of yet because she was able to implement some simple lifestyle changes and increase her surveillance to help mitigate her risk for cardiovascular complications. So this is just one example of how genomics can impact human health and disease risk assessment. The second example is within oncology. So the field of oncology is leading the way in precision medicine and targeted therapies because the identification of variants as biomarkers or targets of disease has led to new targeted therapies for patients with cancer. Now, past treatments have all been focused on tissue-specific mechanisms or systemic therapies. So, for example, the traditional chemotherapies such as microtubule inhibitors, platinum therapies, and cyclophosphamide. These are all cytotoxic drugs that kill all rapidly dividing cells. And so there are also other cancers such as hormone-responsive breast cancer or prostate cancers that have obvious hormone dependence. So the first line of therapy in these tumors is a systemic hormone deprivation. And the problem with all of these systemic-based chemotherapies is that they often have some really miserable side effects for the patient. And so more recent focus has been on variants and the targeted therapies. And as time has gone by, we've learned more and more about the molecular basis of cancers. And so shown here is non-small cell lung cancer and melanoma. And we know that each of these common cancers is actually a collection of more rare cancers. So for example, many variants have been detected in non-small cell lung cancer, including driver mutations in the genes for things like EGFR, BRAF, MEK1, AKT, and PTEN. And then within each of these driver mutations, there's individual variants that add further complexity. So for example, within the EGFR mutations in non-small cell lung cancer, there are at least 7 known individual variants. And in melanoma, it's similar. The common driver mutations are in the BRAF gene, and these include 6 individual variants. So imagine if we had drugs that were specific for each of these individual variants, and how cool would that be, right? Well, and that's where we are, albeit in the very, very early stages. Right now, the majority of the drugs that are seeking and receiving FDA approval in oncology are targeted therapies that are designed to hit specific molecular targets that are defined by a genomic variant. So these new drugs interrupt cancer cell proliferation, but in theory, they leave other non-cancer cells more intact with the goal of less intense side effects for the patient. There are also new immunotherapies that capitalize on the patient's own immune system to fight types of cancer with specific genetic signatures. And so now the challenge is, can we identify all the variants that impact disease and develop therapies that work best for each of those different disease-causing variants? So the All of Us research program will help with that in the US, and similar programs are in place in other countries. And we also have to start to convince big pharma to change their strategy and begin to develop targeted therapies for newly identified variants instead of seeking that next billion-dollar blockbuster drug. Now, in the clinical setting, we identify DNA variants in a variety of ways. We can use single gene sequencing analysis to look for germline mutations in particular genes, such as BRCA1 or 2. We can do screenings for variants in the genes that are metabolic enzymes, such as the cytochrome P450 enzymes that are responsible for drug metabolism in the liver. And we can perform gene panels, such as Oncotype DX or MammaPrint, that contain smaller number of genes to identify variants and predict tumor recurrence in some particular tumor types. Or we can do what we're doing at the LSU HSC Clinical Precision Medicine Laboratory, which is focus on the next generation sequencing-based approaches. So this includes TSO500, whole exome sequencing, and whole genome sequencing. So our initial approach for TSO500 will support solid tumors, and then we'll expand into hematological malignancies. And then we'll implement hereditary cancer screening, risk identification, and some non-invasive prenatal testing, or NIPT. All of which runs on the same next generation sequencing platform. And so this testing is one of the pillars of support for our future bid for NCI Cancer Center designation in the region. And so this is the instrument we have from Illumina called the NextSeq 550DX. This instrument is currently in use for genomic sequencing of SARS-CoV-2 for COVID-19, but we will be pivoting soon to run TSO 500, which is a panel that contains 523 oncology-related genes. And you can see some of those listed here, along with the different types of cancer with which they are affiliated. This panel also provides an assessment of microsatellite instability and defects in mismatch repair. These support the use of checkpoint inhibitor therapies, as well as overall tumor mutational burden, which is an indicator of immunotherapy treatment success. So the way it works in our laboratory is we have tests that are ordered by a physician. Our pathology partners review the sample and submit the tumor and normal samples for sequencing. In the lab, we process those samples, perform library generation, and put them on the sequencer. And then the large amounts of raw data that result from the NextSeq are run through quality assurance, quality control, and base calling algorithms to convert those raw image data files into a text file format. You guys have already heard about FASTQ. And there are also the associated Fred quality scores that are associated with each base. And then following this base calling, the sequencing reads are aligned with the reference genome and resulting in a BAM file. Map sequences are then used to identify variants and result in a variant call format, or a VCF. So we partner with Purion DX for this service, and their algorithms identify and prioritize single nucleotide variants based on multiple criteria. These include variant allele frequency, strand bias, sequencing depth in the gene region, quality scores, et cetera, as well as it draws from their extensive database of patient outcomes from other NCI designated cancer centers nationwide to recommend treatment strategies. So the identified variants are annotated as to the gene and location, that's the AVCF file. And then they're often manually curated to ensure accuracy, quality, and adherence to the current nomenclature and naming conventions. So all of this data is distilled into a report that's integrated into the EMR, the electronic medical record, and it's made available for tumor board review and for clinical decision making. So those sample reports look like this. So it has patient information at the top, and then it has a listing of the variants that are identified in the analyzed sample. So there's also information about the responsiveness of this tumor with, or a tumor with this profile to certain drugs. And you can see that here in the middle, as well as the opportunities for clinical trials that are associated with this type of variant. And so we can set the distance parameters to limit available clinical trials to within so many miles of the patient, mostly for convenience sake. And then down here at the bottom, you can see the results for tumor mutational burden, which is really the emerging marker associated with greater sensitivity of a tumor to immunotherapies, and also microsatellite instability status, which when high, this example is low, but when it's high, it predicts sensitivity to immune checkpoint inhibitors. So all of this leads us to the precision medicine ideal of delivering the right drug to the right patient at the right time. And so right now we're sort of in this top level here where we have patients receiving a drug, and then there's a small proportion of those patients who actually respond. What we'd like to do is get to this stage at the bottom where we have a different drug for different genetic variants that are available within the oncology and other space. This doesn't necessarily have to be only oncology driven, even though oncology is leading the way. But this is where we want to go, where we get the right drug and the right patient at the right time. And so with that, I'd like to thank you for your attention and this opportunity, and I'd be happy to answer any questions.
Thank you so much, Dr. Crabtree. That was an amazing presentation.
Thank you.
Okay, we have a few questions for you. The first one is, how much data is being generated by your lab at LSU HSC? So is it just genomic data or is an SMP data, or do they also generate other types of data?
So right now, the only data being generated in our laboratory is viral sequences for SARS-CoV-2. We are not currently running the TSO 500. We are not currently doing whole genome or whole exome sequencing. That is our the next endeavor, mostly just because we kind of got derailed to do sequencing for SARS-CoV-2, and we have yet to pivot.
That makes sense. Okay, our next question is, how many people are you planning to get sequence data from in the state of Louisiana and also just in the South in general?
Sequence data for whole genome, whole exome? Or is this a question of how many samples are we going to run?
How many samples are you going to run?
Okay, so once TSO is up and running, so the instrument that we have is a moderate throughput instrument. There's one bigger that we do not yet have, but we can always buy if we end up having more samples to process. Our laboratory is not going to only support LSU. It's going to support the entire region. And so at the moment, we have the capacity in our current instrument to run 8 tumors normal pairs per week. But then, you know, as need and demand increases, we can always scale that up.
Got it. And then one final question. What originally inspired you to become involved in genomics?
So this is kind of a cool study. Or a cool story. So I had a science teacher named Jim Martin, and he would die if he knew I was telling the story. Back when I was in high school, we were in basic biology, and he gave each of us a laminated piece of paper that had either a circle on the end or a triangle on the end. And some of us had the points and some of us had the correlating V, right? And so essentially what he handed out to the whole class was nucleotides. And he lined us up in the hallway, and some of us were the parental strand, and then others of us came in and formed the replicating strand. And so that was really my first introduction to, to DNA and anything having to do with DNA sequencing. And so he taught us DNA replication in the hallway of the high school. And so that was really, that was many, many years ago. But that was really my first introduction to DNA. And it's kind of stuck ever since then. Gene therapy, I've got a huge interest in not only DNA sequencing and genomics, but into gene therapy and that translation of what we can learn from genomics to patient care.
Great, that is a fun story. Thank you for sharing.
He would die.
So we have so we have one more question. You already have a list of known somatic variants for certain diseases. Have you come across a patient where there's a new unknown variant associated with their disease?
I personally have not, mostly because we have not had TSO up and running yet, but we do have the ability to detect those new variants. We call them de novo variants. And as those begin to show up in our population, we can track those in the familial ways. And then also seeing if there's a particular variant that shows up within, certain geographic regions or, that kind of thing. And I'm sure the All of Us program, the more they analyze those 100,000 sequences that they just made available through their interface, as they analyze those sequences, that's going to most likely identify new de novo variants that we hadn't already defined.
Great, thank you. Well, thank you again for the great talk. It was a pleasure to have you here with us.
Thank you, everyone.
All right. So up next, we are going to hear from Mr. Mark Allen. So it is my pleasure to welcome Mr. Mark Allen. He is a Google Cloud account manager for the National Institute of Health. He's focused on extramural researcher enablement. Prior to joining Google, Mark worked at Microsoft, aiding his higher education clients to adopt the cloud. Mark has a BS in Business Administration from the California University of Pennsylvania and an MSIT from Carnegie Mellon University. So please join me in welcoming Mr. Mark Allen. Oh, can we mark a co-host, please?
Hey, can you guys hear me okay?
Yes, we can.
Excellent. Thank you very much. Thanks for the intro, Laura. I really appreciate it. I also invited Ashoki, who is a business development rep here at Google Cloud, and she's going to share off the slides. I'm working from my phone and my iPad today, unfortunately. So, Ashoki, if you wouldn't mind sharing out the slides, please.
Sorry, who's Ashoki? Is it Akshay?
O-S-I-O-K-E. And then her last name is Diofo. There you go. Perfect. Excellent. All right. I think we're good. Thank you very much, Ashoki. So as mentioned, I'm Mark Allen. I worked at Microsoft for 9 years before joining Google. And again, very much helping my higher education customers adopt the cloud. I'm based out of San Francisco, California, but I did work for Microsoft 2 years on the East Coast outside of New York City. Prior to joining Microsoft, I was in the customer space. I was actually in education. I was a director of IT and worked on a lot of infrastructure stuff. I won't get into all the details, of course. So I kind of still maintain that customer perspective, really. Next slide, please. So here's our quick agenda. We're going to talk a little bit briefly about why Google Cloud. I'm going to try my best not to mention any product names. I think I'm going to mention one. And then we're going to talk about NIH and the STRIDES program, of course, why you're here. How to join the program, and then what happens after you enroll, right? And then we'll switch to a Q&A, and we'll go from there. So first off, why Google Cloud? So next slide, please, Ashoki. So when you think of Google, you probably think of the search engine, right? The very generic white background, beautifully colored letters, and you type in your search, and within milliseconds, you get your results, right? Next slide. But what you don't think of is the infrastructure behind it, right? There are thousands of servers that are running the google.com search engine, as well as all of our applications. Matter of fact, Google has been building, consumer-based applications since '97, right? So if you go to the next slide, you'll see kind of a great timeline and a history of the major products that Google has released. We've been in the consumer space since before '97, and each of these applications on here has 1 billion users for each application. 2 of them actually have 2 billion users. And you'll note that all of them are 100% powered by machine learning, right? So you're gonna have access to a slide deck, and if you look at the little blue notes beneath each application, there is at least 2 of the machine learning powered features of those applications, right? I guarantee if you have children, they're using YouTube. I know my nieces religiously. And so, we've definitely helped with machine learning to do some better thumbnails, better recommendations, right? Always looking at the algorithm and making sure that our consumers are kind of getting the needs that they have. Next slide, please. A few more little tidbits about Google, right? So Google is the world's largest purchaser of renewable energy. In fact, all of our global operations have been powered by 100% renewable energy since 2017. Here you got a couple pictures of the big windmills, of course, the wind farm, and then the solar panels as well, right? So we're very committed to sustainability and going green. And so if you partner with Google, you are definitely making a green choice as well. Next slide. One of the reasons I joined Google is their dedication to hybrid and multi-cloud solutions. Other cloud providers certainly have this. I'm not here to knock on them, but essentially, their approach is more of a hardware lock-in or a vendor lock-in, right? Google's approach, even outside of hybrid multi-cloud, is a software-based approach, right? So we have developed, Google Kubernetes, and now that is certainly a de facto standard out in the world, right? The only product I'll mention is Anthos, right? Anthos is built on that Kubernetes engine, and this is kind of a big game changer. It's a software-based solution that allows you to essentially run Google Cloud services on premises or in other clouds even, right? So we have a lot of great flagship products, and maybe you say, hey, my data is on premise and I need to keep it on premise. You can bring the cloud down to you with Anthos, right? So we're very committed to hybrid and multi-cloud solutions and the approach. If, ideally one day everybody's in the cloud, but along the way, we're going to have, certainly a hybrid approach as well as many cloud providers involved. Next slide. A couple slides real quick on security. Our approach to security in 2 words is trust nothing. And if you go to the next slide, essentially what I'm talking about here is we don't consider traffic, any internet traffic, whether it's internal or external, to be different, right? There's no difference as far as we're concerned. So we build in layered defense in depth, right? We essentially look for mitigations and multiple defenses within each layer and in between each layer, right? And so with this approach, we've essentially been able to create a very secure cloud computing area. Next slide. If you have any compliance needs, we certainly meet all or exceed all of the government requirements for security and privacy protection. We're happy to give you some security attestation reports. You can just go to our Cloud Security Command Center and take a look at all the reports yourself as well. But we have all the big ones, of course, FedRAMP, we have the High Trust, we have HIPAA. There's a bunch of ISO ones as well. So whatever your needs are, chances are we meet those needs. And if we don't, we're certainly willing to work with you on getting those compliance certifications. All right, next slide, and then slide that. So what is STRIDE? So take a brief second to talk about the National Institutes of Health. So NIH is comprised of 27 institutes, centers, and offices. We just call them ICs. So apologies if I use acronyms, but when I say IC, I just mean one of the ICs regarding the NIH. I got a couple examples up there. The presentation before me, I think, mentioned the NCI as well, so I think you're familiar with that one. I put a link for the Reporter up here. Reporter is an NIH tool that they have built that allows anybody to kind of see all of the grants that are out there per institution or even per PI, principal investigator, right? So you're welcome to use that Reporter and kind of see, you can see how much funding your institution's, received, what grants your PIs are working on, all the good stuff there, essentially. What I didn't actually put on this slide is regarding how NIH's funding is set up. And it's very, very complex, but at a very high level, if you read their strategic report, 80% of their funding goes to extramural universities, extramural institutions. Out of that 80%, 70% is roughly all higher education, right? So that's around 50% of their total budget. Their total budget for 2022 was around $49 billion. So NIH is committed to extramural research and allowing others to kind of, do the research that makes sense from a biomedical standpoint. Next slide. Okay, so we're keep it on this slide actually, Shoki. So STRIDES, right? Let's talk about STRIDES. STRIDES is an acronym, of course. It stands for the Science and Technology Research Infrastructure for Discovery, Experimentation, and Sustainability. Essentially, it's a program they built because they see the value in cloud. And they're really just trying to accelerate biomedical discovery, right? Google was first to enter this partnership in July of 2018. AWS signed on recently after that as well. And then I think Microsoft Azure just signed up in July of 2021, right? So all the three major cloud providers are in there, but we're a little unique in that not only did they partner with us first, but our AI and ML capabilities, as I kind of mentioned before, are differentiated enough that we have a very big say in kind of how that AI is done, right? I won't talk about AlphaFold here, but that's certainly one of the hot buzzwords when it comes to machine learning, right? And that's kind of true to just Google's testament of AI and machine learning. Next slide, please. From a benefit standpoint, when you sign on to the STRIDES program, the first benefit most people realize right away is those deep discounts on Google Cloud. Now, normally I put the pricing table in here because we're being recorded. We like not to do that, but I can verbally say at least that there is 25% discount on compute and storage. The 25% discount on compute applies to all compute resources, including GPUs. So if you have high compute needs, the discounts are there and they're provided to you, via Google Cloud from NIH, again, to get more research for your dollar, right? And hopefully to accelerate that biomedical discovery. Other big benefits we always talk about are the access to training. There's a discount on training programs as well. We also have some free and no cost training options. I'll talk about this briefly at the end. You also get a direct relationship with NIH, right? We are happy to kind of make connections to program officers, people within ICs, whoever it makes sense to kind of have a conversation to talk more about the research, the programs, and then if you're enrolling in STRIDES and you actually have some, architecture discussion questions, we can certainly make those connections as well. And of course, that last big benefit is that faster time to insight. Again, we're always trying to get the results quicker so you can spend more time on the science versus on the infrastructure itself. Next slide. And then, so one big thing I want to point out is there is no upfront commitment to joining STRIDES. There's no money you pay to get into the program. It essentially is provided at no cost because you're going to use NIH dollars for your research in the cloud. And then next slide. So another great benefit is increased advocacy, right? So everybody on this call has an account team at Google Cloud, right? Every university, every college, you have an account team at Google Cloud. That account team usually consists of an account manager like myself. You probably have a customer engineer, who's kind of the more technical person on that call or engage with you. And there's probably a few others that are on your account team. On the NIH account team as well, right? I'm one of the account managers. We have support, we have consulting, we have customer engineers and architects, data scientists. It's a really large account team. And if you click on the slide real quick, Ashoki, so when you join the program, you essentially get, the advocacy from both your account team as well as us, right? So we're here to help any NIH funded researcher get what they need to out of Google Cloud, essentially, right? Obviously within reason. But my role as account manager is to be an advocate for NIH as an account, but also anybody I'm working with, right? And I'm focused on extramurals. So it's around 3,000 institutions I'm working with. Next slide. Oh, and then there's a quick little red blurb. Yeah. More advocacy within Google and within NIH as well, right? Next slide. Public data sets. So one of the big, another big feature of the STRIDES program itself is trying to get access to, open data, right? Trying to make sure that the data that biomedical researchers need is available in the cloud. I think on the presentation before this, I heard all of us. And so all of us is actually working with the team there, and we're getting all of us on Google Cloud as well, right? So there's a lot of great public data sets we have available. We have some that are publicly available, but not in the program itself, but you can still access them. There's things like imaging data commons, there's SRA toolkits, and we're always adding more data sets as well, right? So I'm currently working with a PI out of UCSF who manages a 400 terabyte database, and we're working with the program manager for the public data sets program to get it in the cloud, right? with the way we see it, the more data that's in the cloud, the quicker your research can get done. You don't have to spend time moving your data around, massaging it. It's already there, it's already ready to go, and you can just kind of start your research. Next slide. All right, flexible options to join. Let's dive into this, right? So Shoki, I'm going to have you click when I say click, if you don't mind. This slide builds out, right? So if you don't mind, click once. And so this is you. You are a researcher out there, and you know, maybe you're getting NIH funded dollars. Maybe you're getting funds from other organizations. You know, whatever it may be. Click again. And so you are probably reporting up to a department. If you click again, that department has other researchers, and click again. And then again, that department's up into a school or an institute. And if you click again one more time, you know, that reports up to a university. That might have more schools. It might have an academic med center, right? But essentially there are NIH funded researchers all throughout this organization. And so what our goal is to sign the STRIDES agreement at the highest level, right? We're happy to sign individual researchers, departments, schools, institutes, whatever it takes, right? Because sometimes that university procurement process can take, you know, 5, 8, 9 months, right? So when you have NIH funded dollars and you want to kind of experience the benefits, especially the discounts right away, we're happy to work with you to get you signed in as quickly as possible, right? If you click one more time, the one thing that we really like to have too is kind of an IT support as well, right? So we understand not every organization has IT support or maybe research computing. And so it doesn't need to be a full-fledged IT. We just need somebody that is kind of going to be that point person for billing IDs and really understanding the process, right? So we work with that person or team of people to make sure they understand the process, what information is needed to get an ID, billing ID, and then kind of, you know, giving out from there, right? So this is kind of like the pyramid that we always do, the pyramid of what we're trying to get to, right? The higher we sign, the more benefits all the NIH researchers. Next slide. So I'm going to talk about these options to sign on very quickly because they can get in the weeds very quickly as well. So we have 3 major options. First option is direct to Carahsoft. Carahsoft is the federal distributor of the Google Cloud service for NIH, right? And for many of our actual public sector customers. If you click once, this is a very simple process. You would essentially engage with Carahsoft. We would set up those conversations and you can essentially sign on very easily. If you go to the next slide, this is where we have a little more flexibility. And this is where most of our education customers kind of want to fall, right? And so you'll notice I have the same image kind of twice. And so we'll talk about it twice essentially. So we have a few resellers we work with. The 2 that most of our customers sign through is Onyx and Burwood Group. Both of these resellers are a value add reseller, and they first, there's no further margin, right? It's not like they're cutting into the discounts at all. You sign on with them and it's still no cost, but both of them actually have a very unique billing tool. And if you are that kind of IT person that is going to be doing all the billing IDs, those billing tools that they provide are just hugely helpful as far as being able to kind of differentiate where the money is going per grant and per project, right? So if you are interested in joining STRIDES, we will definitely loop in Onyx and Burwood. You can have a conversation with them. If you elect not to go with a reseller and just stick direct to Carahsoft through option 1, the billing is going to be kind of up to you, right? Making sure that you are able to report on the billing is going to fall on your hands. And the reason for that, for all of this essentially, is all of these funds are federal funds, right? Which means they are subject to congressional reporting. And NIH is very big on this, of course. And we all should be too. It's our tax dollars. So, but essentially, if it's not reported well, then there's definitely a lot of questions, right? So, engaging with Onyx and Burwood, your best way to go. They have a very great tool. On the right, they'll notice the only difference is Internet 2, right? You are welcome to sign on with Internet 2. You can be a member or a non-member, but if you sign the STRIDES agreement through Internet 2, you also get to realize not only the benefits of the STRIDES program, but the benefits of Internet 2 as well. And so I'll cover those in about 2 more slides. If you click once, there's another little red blurb, no increase in price with the use of a reseller, right? Next slide, please. Now, the last one's very quick and dirty. Essentially, if you already have an existing agreement with Google Cloud or with Carahsoft, we can leverage that agreement as well, right? There's not many customers that fall into this category. It's typically, let's use a reseller. Let's use, make sure we also get Internet 2. This is something you're also welcome to explore if you want to. Next slide, please. This is just a quick summary. I'm not going to cover this in the interest of time, but you are welcome to read it and ask any questions later if you need to. Next slide, please. So Internet 2. So there's an Internet 2 Net Plus GCP agreement, right? This was pre-negotiated with 4 universities to kind of provide benefits to members and non-members, quite frankly, for Internet 2 and for Google Cloud, essentially. So essentially, there's the biggest thing we always talk about is if you join through Internet 2, not only will your NIH workloads get the 25% discount, but any non-NIH workload will receive a 5% discount on compute and storage as well, right? There's also a lot of other benefits, of course. We invite you to reach out to that email address, netplus@internet2. You work with a guy named Bob Flynn. Bob is from Indiana University, one of the initial 4 universities that helped negotiate this contract. And Bob's amazing. So I can't talk about him enough. He's very knowledgeable. He's very customer focused, and he'll set you straight. So if invite you to reach out to that email address and talk to Bob, and he'll tell you all the cool stuff. Next slide, please. All right, here's just a quick list of Internet 2 Net Plus Google Cloud subscribers, right? Your college or university may be signed on to Net Plus, but maybe through Azure, maybe through AWS. Maybe you're a non-member of Internet 2 as well, right? So this is a quick list, and I put the link in the notes. So you're welcome to kind of peruse that as well. Next slide. All right. So after enrolling in Strides, we engaged with Internet 2 and we got some feedback, right? That we asked a lot of questions, or Internet 2 asked a lot of questions, I should say, on kind of, hey, you have access to the Strides program. You're an NIH researcher. Why is there a disconnect? Why are you not using the Strides program, right? And the biggest result we got back was actually just communication. They just weren't aware they had access to it. And so we have kind of created a success plan to make sure that going forward, when a university or college or med center or even just department signs on, this is kind of the keys to success to make sure that all the researchers there know they have access to it and how to get access to it. So to start again, we have an IT support process, and it's more about the billing account ID process. It doesn't need to be IT. It can be research computing. It can be a PI or a PI's assistant. We have a few of those examples. Just somebody that's kind of there to say, yep, I know I need to get this type of information from a researcher. And then I can send it to NIH and I will get my project ID, my billing ID, right? So very, very simple process. We just kind of need that one person at least. The other major things we found is having a website. I think on this page it says a public page. It doesn't need to be public. We do have 2 examples from NYU and University of Wisconsin-Madison. Welcome to read those. But it could be on your intranet as well, but some type of web page that for researchers that kind of says, here's the NIH Strides program, here's why it's important to you, and here's how to get access to it, right? After that website's completely finalized, an email needs to be kind of sent, some type of communication, whatever that may be, sent out to the research community at that university saying, hey, you have access to this, check out the webpage. If you have any questions, here's some email addresses or people to reach out to, whatever it might be. And then finally, a launch event. This used to just say webinar. I've added seminar because things are opening up now, and hopefully we can get on-site to customers more. But essentially having a large audience of people that are interested in NIH-funded research and interested in Google Cloud, join the webinar or seminar, kind of saying, hey, here's all the questions, here's all the answers to your questions, here's how the program works. Google's happy to join those. NIH is happy to join those. The resellers, Internet 2, they're all happy to join. We're here for you guys. So after you guys sign up and we want to get to that point of like, hey, let's make sure we evangelize the Strides program. We're here to help make that happen. Next slide, please. So another piece of feedback from that Internet 2 survey was around training. Now, I'm going to talk briefly at the end about some free and no-cost training options, but I also wanted to highlight that we work with NIH very, very closely, and last year we helped develop custom training, right? The title is Introduction to Biomedical Data Science in Google Cloud. We delivered it 7 times in 2021 to the intramural researchers. I think the first 3 times there's, kind of reiterations of let's make sure we change this or modify this or speak about this more, this thing less. But after those 3 initial trainings, the last 4 went off without a hitch. We had a lot of success, and we do always seek anonymous feedback. And so there's a quote there from a researcher who joined one of those sessions. There's 16 attendees per class. There's a sample agenda on the right. If you're interested in training, whether it's just custom training or other training options, feel free to reach out and we'll put you in touch with your account manager to talk through those. Next slide. Okay, I am almost finished here. So this is an eyesore, I apologize. And this is screenshots from a doc I have. And so you're going to get access to the PDF with all these links at a very, very high level. I'm going to talk about just a few of them, right? The top left is the Google Cloud free trial. So everybody is welcome to get $300 in credits of Google Cloud services. It does require a credit card, but I will point out that it will not charge your credit card until you say so, right? And until that $300 expired. So what happens is you put your credit card in, you get $300 in credits. There's also a free tier available where we have certain cloud services that are always free. You're welcome to use those credits until they're exhausted. After they're exhausted, then you're prompted. Do you want to use your credit card or not? You can say no, and that's it. You won't get charged. It'll shut down your services, and we can all move on, right? So I know there's always hesitation using credit card, but Google is very open on this and very aware of the fears. So please don't be hesitant to sign into that program. I'm going to jump down to Google Cloud research credits. If you have a research project that you are working on, and maybe you applied for an NIH grant and you didn't get it, but it's a cool research topic, you're welcome to click on that link and apply for research credits from Google. I think we do up to $5,000, and so we'll help fund your project in the cloud. The next two links are about learning Google Cloud, right? There's a lot of free online labs, quests, courses. There's a lot of great stuff in there about free training, essentially. Invite you to check those out as well. The link below that is Google Cloud for researchers. So if you are a researcher, you're also welcome to get some credits and some free training with that. Below that is Google Cloud for students. Again, if you are a student, you're trying to learn some more about Google Cloud, you're welcome to apply to that program as well. The top right, though, is if you are a faculty and you are saying, hey, I'm actually going to teach a class on biomedical research, and we're going to use Google Cloud, that's the program you would use where you would get the credits then dish out to your students, right? So we're very big on free education programs and providing credits to students and faculty as well as researchers. There's a few more links in there. I think one of the ones I would definitely want to highlight is the public sector cloud community. It's the third from the last. We call it PubSec Connect. It's run by a woman named Kate Johnson, and it's there's no sales discussions allowed, right? It's individual to individual. And so essentially other customers talking to other customers about their experience in the cloud, their challenges, their successes. It's a great community. There's a call, I think, every week, and a lot of great presenters on there as well. Google Cloud Communities below that is basically you can kind of engage with product managers here at Google on your questions. And then I put a certification link there below as well. If you click once for me, Ashoki, a nice big red box should appear. So as much as I try to highlight some of these discussion points, there's a session by Alice Kamen to help put most of these programs together. That was delivered yesterday, so I invite you to check that out on the YouTube. She goes in-depth into most of these programs. And again, if you have questions, feel free to reach out. Next slide. So next slide is, I think, just a quick call to action. The call to action is very simple. It is basically, if you have any questions, if you're interested in learning about strides, Google Cloud, Google Workspace, if you have any questions at all, feel free to reach out. And if you go to the next slide, we have some email addresses up here for you, right? So the top is me. I am Mark C. Allen. Had to use my middle initial there. Ashoki's on the call as well. Like I mentioned, she's a business development rep. If you have kind of light or high level questions, she would probably be best to email. She could probably get back to you much quicker than I can, but you're welcome to email me as well, right? Please don't think I don't want your emails. I do. The last email there is the Google Cloud Strides team. And so this is the entire account team for NIH. It includes people from support, consulting. We have architects and data scientists on this email. So if you kind of have a broader question, feel free to email that alias, and we'll be happy to answer your questions for you. If you have any doubts, feel free to just email me and I will direct you to the right people, and we'll get you squared away. And the next slide is just Q&A. So I will open the floor for any Q&A.
Thank you, Mark, for that very informative talk. So in the interest of time, we are actually going to have any questions that you have for Mark to please forward to us, and we can pass those along to you later. Is that all right, Mark?
Perfect. Thank you very much. Sorry if I went over, I apologize. And but this is a lot of fun.
Worries.
So much, yeah.
Yes, thank you again.
Thank you. Bye.
All right. All right. Now I would like to introduce Dr. Gus Kasoulis. So Dr. Kasoulis is a professor of virology and biotechnology in the Department of Pathobiological Sciences and the director of the Division of Biotechnology and Molecular Medicine. He is the director of the NIH-funded Center in Experimental Infectious Disease Research and the director of the NIH-funded Louisiana Biomedical Research Network Molecular and Cell Biology Core. So please join me in welcoming Dr. Gus Kasoulis. Dr. Kasoulis, are you able to unmute your microphone?
Yeah, now it's unmuted. I couldn't unmute it earlier, it was blocked. Can you hear me now?
Yes, we can hear you now.
Okay, great. Okay, thank you, Laura, for the introduction. I wanted to sort of update a little bit what you said. I'm also department head in pathobiological sciences, and we are pleased here at the LSU School of Veterinary Medicine that we have 2 additional centers, the Lung Biology Center that's led by Dr. Samitabi Jayashilan, as well as a preclinical cancer COBRI that's led by Dr. Joseph Francis. The molecular cores of both of those centers, as well as the previous center on infectious diseases, it's actually administered by my division of biotechnology and molecular medicine, with essential elements, the core facility, Gene Lab, that provides next-gen sequencing and bioinformatic analysis internally, as well as in collaboration with Pine Biotech. But I'm here to talk to you today about the nonprofit organization which I represent as the president. And Laura, can I share my slides?
Yes, you should be able to share those.
Okay. So you should be able to see, I hope, my slides now. So the National Association of Idea Principal Investigators is a not-for-profit organization representing all the centers that are funded by the National Institutes of Health, the National Institutes of General Medical Sciences. That includes more than 170 centers for biomedical research excellence that are funded by the COBRI mechanism, 24 of the IMBRE mechanisms, as well as multiple clinical translational centers and other programs that are run through the manpower division, capacity division of NGMS. So the purpose of NAPI is really to promote the IDEA programs. Again, we're a not-for-profit organization that is principally to foster interactions and to promote research sharing. And the website is as shown here, at nepi.org. There's a number of very important information, including resources and training resources that we have made available freely, either through participation of different programs and also through collaboration with Pine Biotech. And I really urge you to explore this website and look at the opportunities that we offer. In addition, NEPI is partnering with LSU to manage the National IDEA Conference. It happens every 2 years. The next conference is called NSBRI. It will be the first week of October, and it will be virtual, and it would allow a glimpse of all these 23 states and Puerto Rico programs, the COBRI, the IMBRIs, the CTRs, the co-funded programs, as well as some of the specialized programs. We expect over 1,000 people to participate on the virtual meeting. The contract has been awarded by NIH to LSU, and NEPI is collaborating with LSU to manage this national meeting. The next meeting after that will be 2024 and 2026 in person in Washington, D.C. The first meeting on 2022 will be virtual meeting, and it will have a number of other resources, training, as well as participation of major companies. We hope Google, Amazon, IBM, and others. So the next slide, this is the leadership of NEPI. I am the current president of NEPI, and these are the officers for the executive committee. We also do have a steering committee having 4 members representing the different programs from each of the 4 geographically different portions of the country, south, east, west, north. And we meet periodically to ascertain how to promote better the interactions with NIH and also with all our constituents. Of course, as I reiterated, to reiterate now, the mission of NEPI is to provide leadership and communication for the development, promotion, and improvement of biomedical research through the INBRI, COBRI entrepreneurship hubs. This is a new type of program that has been developed to also facilitate entrepreneurship within the idea states, and of course the CTR idea programs. The objective is to foster interactions, to promote resource sharing, to enhance the visibility of the idea program, to develop a consensus of priorities, and to identify, disseminate best practices, and to identify opportunities and develop strategies to achieve the common goals of the idea program. One of the major emphases of NIH, as well as of the idea program, especially through the INBRI mechanisms, is really the training of a pipeline of investigators, starting from undergraduate to graduate students to postdocs to faculty in data science and bioinformatics. So therefore, one of the major goals of NEPI is to share resources and provide avenues for training in data science and bioinformatics through NEPI, as well as through the individual components that represent the IDEA program. So in terms of the history of the IDEA program, it started in 1993. The purpose was to broaden the geographical distribution of NIH funding for biomedical research and behavioral research. It focuses on health-related research, and the reasoning is to enhance the competitiveness of investigators at all institutions located in states, which the aggregate success rate for the application NIH has been historically been low. If you look really at the trajectory of this program, it's actually quite amazing that the IDEA program has achieved a number of different important thresholds, including the graduation of young investigators with R1 nationally competitive applications at a younger age and with increased success rate, even comparing all NIH programs across the board. So we are really to promote science and create a pipeline of investigators, starting from undergraduates all the way to faculty and hopefully through tenure and the promotion of program grant funding. So this is the type of funding that has been received so far. In 2021, there were $396 million allocated to the IDEA as part of the overall budget. The goal of NEPI is to facilitate, in collaboration with the EPSCoR Foundation, EPSCoR Coalition, up to 1% of the NIH budget to be targeted to IDEA programs. We're a little bit below that at this point. The projection for 2022 is at $410 million, but still under the projected, or at least the what we wish, which is 1% of the total NIH budget. Mind you, for 23 states in Puerto Rico, practically half of the states in the country. This is the type of contributions, different programs, highlighted only a few programs, but you can see in North Dakota, more than half of the funding for all NIH funding for that state comes from the IDEA program. In other states, like Louisiana, a little bit more successful. It's a smaller portion. But critically, I think this particular component here facilitates a number of people that are actually now nationally competitive are able to compete and get traditional categorical funding through competitive awards beyond at the national level. So it's a very critical component of what NIH is doing. These are the principal components of the IDEA funding, the COBRI programs. The purpose of this COBRI is to graduate people with independent funding, hopefully R1 funding over a 15-year period. The IMBRI program, it's only one per state, and that really focuses on the pipeline from undergraduates all the way to graduate and faculty. It has a number of primarily undergraduate institutions, and it has a critical component in teaching, mentoring, and training in data science and bioinformatics. In fact, one of the principal cores, which is mandatory for all these programs, is a data science bioinformatics core right now that we also have in our own program. We also have the clinical translational research centers. They're really sort of smaller clinical translational centers, the CTSAs as they're named, and these are really targeting the IDEA states for clinical and translational research. One important component of the IDEA program is co-funding. People that go for our 1s or 15s or S10 applications may get a boost and get funded probably within 5 percentile points through co-funding between IGMS IDEA and the Categorical Institute. And of course, the new addition is the regional technology transfer hubs, STTRs. There are 4 they're active right now. There may be additional ones coming up in the next round of applications. And the idea is to really increase the number of SBIRs, STTR grants that are funded in the IDEA states, since they're not really participating to a large extent in this particular pool of funding that comes from federal sources. Part of the IDEA networks is also the use of cloud computing that you just heard, the STRIDE program for Google and Amazon. And there are special allocations and supplemental funding for that. There are promotion of statewide regional networks that are promoted through in a variety of different ways beyond just the CTRs. And this involves a specific co-funding coordinator that has been appointed for each NIC Categorical Institute. The regional hubs also have pilot programs, and they also teach fundamentals for technology core directors to promote entrepreneurship. There are also supplemental fundings that came to increase connectivity between IMBRIs and COBRIs, CTRs, and so on. We've been the recipients of multiple of those supplemental funding. And part of the idea is to connect all these IDEA programs together through regional centers and promote collaboration among all the programs. And then, of course, there were specialized programs because of the pandemic, the COVID-19 patient registry, cloud computing, as already mentioned. And then there is NIH-wide research initiatives that are also linked to programs within NIGMS and the IDEA program. This is the map of the IDEA states. As you see, they're all over the country. And the focus is to also bring those states up to become competitive for NIH funding as well as other federal funding. So NEPI, through its website, has a number of very important activities. It funds IDEA undergraduate traveling to Washington, D.C. for the meeting, as well as for registration into our virtual NSBRI meeting in 2022. We have awards for undergraduate and graduate students that we actually already have allocated to our regional IDEA meetings that happen in between the national NSBRI meeting. And we were happy to award up to $3,000 to individual undergraduate and graduate students and assistant professors in each of those regional meetings that already have occurred. We can have access to IDEA-wide databases and research expertise through the NEPI website, which is actually increasingly populated with new information. And ultimately, we do provide discounts to virtual courses in that data science and bioinformatics through a variety of programs, but also through collaboration with Pine Biotech and through a grant that we received from the Tober Foundation that allows certain of these scholarships, as well as support for certain seminars that will be promoting in the national NSBRI meeting that will happen the first week of October. Part of the sponsored symposia was the previous Omics Research Symposium that happened, the Omics one in August 21 and 22. We also have advertised and support this Omics Symposium that has been put together in collaboration with Biotech that provides, I think, important information. And all these recorded sessions will be available through the NEPI website that we have. So this is what I had to tell you about NEPI. I would certainly encourage you to look at the NEPI website. We are going into a major campaign to organize this virtual meeting. Originally, it was supposed to be in person in June. However, we only just received the notice of award for NIH for the next 3 meetings. So the next meeting would be the first week of October. I believe October 4th, 5, and 6. It will be a virtual meeting, and we hope that the registration for this particular meeting will open hopefully within the next 2 to 3 weeks. And I would urge all of you to participate in this meeting, because you're going to see the breadth of all the research that is being supported by the IDEA program, not only our IMBRI programs, but also the COBRIs, the CTRs, some of the specific networks that are being funded by NGMS for child health and cancer and beyond that. So I would point you to that website, the NEPI website, for more information as it becomes available. So thank you for participating. It's a very important activity that Biotech in collaboration with others has put together. And I look forward to attend some of the next sessions and answer any questions that you may have. Laura?
Okay, thank you very much, Dr. Kasoulis. That was a very informative talk. We have one question for you. How can NAPI members participate in the educational resource development NAPI is doing?
Yeah, so basically through the website, there is a form for any content that you have that you could actually share. If you're willing to share it, NEPI will promote it. We have over 1,000 people registered on our automated emailing system that would alert them of that resource. We are actually in the process of mailing out to major providers, which includes Google and others, to link some of their content through our NEPI website and potentially provide some discounts or support as it has been done with NIH. So I would urge you to contact the NEPI webmaster and let us know. You can let me personally know what you may be thinking, and I will be happy to share it with you or point you to the right material.
Great, thank you. Another question. So can faculties at universities that aren't involved in NAPI get involved?
Yes, actually the NEPI website is really free, accessible to all. So all the content is available, and we welcome any participation from non-IDEA states that could share with us content or information or resources. Obviously, we're trying to make this information for the most part free or heavily discounted for our NEPI members. But absolutely, yes, we welcome anybody through our NEPI website to communicate with us and offer advice, material, whatever that may be.
Great, thank you very much. Well, I don't think there are any more questions for you, so thank you very much again for presenting.
Okay, thanks.
All right. Well, now I would like to present our next speaker. So Dr. Luis Vasquez Quinones is an assistant professor of biology and molecular cell biology at Ana G. Mendez University. He is also the co-director of the Bioinformatics Resource Core program from Rbd and leads the orientation portion of Puerto Rico's Community of Practice in Bioinformatics. Dr. Vasquez Quinones was first exposed to bioinformatics when he was an undergraduate student through an introductory computer science course. During the course, he learned some programming skills, but it wasn't until he was re-exposed to bioinformatics through a graduate course that he became interested in data analysis. Later in his career, Dr. Vasquez Quinones was able to integrate aspects of bioinformatics into academic courses and research so that his students could be exposed to bioinformatics similarly to how he was. Dr. Vasquez Quinones is also involved in teaching outreach programs and encourages students to reach out to experts to help them learn. Apart from his teaching and research responsibilities, Dr. Vasquez Quinones is also the co-director of the Rbd Bioinformatics Research Cohort and an active member of the Puerto Rico Community of Practice in Bioinformatics. The goal of this community practice in bioinformatics is to reconnect the Puerto Rico institutions together to create a community of active and open research with a focus on data science and bioinformatics. So with that, please join me in welcoming Dr. Luis Vasquez Quinones.
Hello, everyone.
Hello, welcome.
Can you see my screen?
Yes, we can.
Okay, hello, everyone. Today, I am going to talk to you about the bioinformatics training opportunities that Puerto Rico INBRE is offering during this grant cycle 2021-2025. The title for my presentation is Puerto Rico INBRE Bioinformatics Training Opportunities to the Community of Practice Model. The general outline for my presentation is: where is Puerto Rico INBRE located on the world map? What is a community of practice? The stages for a community of practice, what we have been doing for the implementation of the community of practice with an update, bioinformatics community of practice training opportunities, and other training opportunities to the Puerto Rico INBRE network, benefits of the bioinformatics community of practice, acknowledgements, and answer questions. For those of you that do not know, Puerto Rico is located in the Caribbean, and Puerto Rico INBRE belongs to the Southeast region of the INBRE program. A community of practice is a knowledge-based social structure that enables groups of people who share a passion, in our case bioinformatics, for a domain to interact regularly and deepen their knowledge and expertise in that domain. A community of practice is comprised of three key elements: the domain, that is a common interest in a field or area of knowledge, consists of key problems experienced by members. In our case, the domain is bioinformatics, with the goal of increasing bioinformatics and computational skills among our members. The second element is the community. That should be our voluntary members that share the interest in the domain and actively participate in designing activities and dialogues for the fundamental goal of increasing knowledge and solving problems through interaction. So in our case, the community will be the scientists around Puerto Rico, researchers, teaching faculty, students, undergrads, and graduate students as well. The third element is the practice, that are a collection of methods, techniques, tools, information, stories, resources, etc., that members use together to advance the goals of the community of practice. In our case, our practice will be for research and education. In this slide, you can see that a community of practice has 5 stages. potential, coalescing, maturing, stewardship, and transformation. During the potential state, faculty from predominantly undergraduate institutions, also known as PUIs in our case, have a high academic workload among other responsibilities that represents a barrier to keeping up with changes in the field of bioinformatics. In addition, these institutions also lack computational capabilities. In the coalescing stage, this model, the community of practice model, will bring together a community of scientists and trainees from around the island to facilitate collaboration at the research and educational level that will increase the number of scientists using bioinformatics and to prepare the next generation of scientists. The third stage is maturing, in which activities directed to increase bioinformatics and computational skills in non-bioinformatics scientists will be designed and developed in collaboration with Is Rbd Bioinformatics Resource Corps, also known as BIRC, to offer community workshops, intervention courses, or laboratories at request of the institutions. The fourth stage, stewardship, this community of practice level is reached when a satisfactory bioinformatics proficiency has been reached among the members. The fifth stage, that is transformation, this is the ultimate stage of the community of practice when the scientific community is integrated and willing to collaborate beyond the walls of each institution at the research and educational level. Currently, we are at stage number 2 and 3, coalescing, because we are bringing together every scientist around the island with interest in this initiative. And maturing, because we already started developing, we already developed the initial workshops is in Linux, and we are designing more workshops to be offered. Is the purpose of Puerto Rico Rbd to increase the interaction between researchers, teaching faculty, and students around the island. The community will play an essential role to foster these interactions to increase the bioinformatics skills among our members. So starting with the training opportunities, the Training and Education Support Subcorps, led by me, designed the Linux workshop in which the members of the bioinformatics community of practice will have access to the computational resources offered by the Boca Raton cluster of the High Performance Computing Facility at the University of Puerto Rico. This, the High Performance Computing Facility is led by Dr. Humberto Ortiz Sosaga of the University of Puerto Rico, and he is supporting us and his team by providing the accounts for our members. The operating system of the Boca Raton is Linux, and the participants will be learning the most common commands to be ready for future bioinformatics workshop. In this figure, you can see that we have already identified a total of 30 institutions around the island. In the lower left corner of the slide, you can see the legend. And the colors of the stars reflects the progress that we already achieved with each institution. The number next to some of the stars indicates how many of the 3 Linux workshops have already been received. A total of 20 out of the 30 institutions have already accepted the invitation, have already identified a leader for the community of practice, the local community of practice, have already coordinated or are in the process of coordinating the initial meeting to talk about the initiative, or already received the orientation, started coordinating the workshop, or already received them. Currently, we are still paying efforts contacting the third of the institution that haven't responded yet. In this slide, you can see pictures of the trainings that have been going on around the island in a virtual or in-person fashion. I have been visiting the communities around the island, and this one is in the north part of the island, in the north part center. This other one is in the northeast part of the island. This one is in the north, and this one is in the south. We have been providing 3 Linux workshops with enough time of interaction with the participants to answer their specific questions, but as well to explore the community interests. So once when they have completed the 3 part of the workshops, follow-up meetings are scheduled to explore new workshops based on their interest, or coordinating additional Linux workshops for additional faculty and students. In this slide, you can see, in this picture, you can see a typical slide to explain a command in an easy step-by-step way, in which this increases the execution of the participants. And all participants receive these detailed presentations by a OneDrive link. So those presentations serve as reference for future and the near future additional workshops. Continuing with the training opportunities, the next one will be for next generation sequencing workshop. The Oxford Nanopore MinION will be used as a portable sequencing device to train our members in sample sequencing, data processing, and assembly. The objective is to include new laboratory experiences in biology curricula to impact our students around the island, but also to include these approaches for research. The Linux knowledge is important for this kind of workshop. The next training opportunity that we have been identified is relational databases. Database constitutes an ideal framework for representing and analyzing large data sets in biology. A structured query language is one of the most common tools used in the field of data science that you have heard a lot during this symposium. Relational databases allow users to formulate and answer questions about their data. Our participants will learn an efficient way to manipulate the large data sets. Currently, nowadays, having our skills are very important for biologists. So in that sense, we are going to be offering our workshop in which our members will have the opportunity to learn R, to analyze their data and generate plots. R is used widely in several disciplines. It's free, which allows scientists to analyze their data without expending in commercial programs. In addition, R has a lot of bioinformatics free tools that are available to analyze data for current biological and emerging biological assays. Another opportunity comes from the metagenomics subcore led by Dr. Filipa Godoy Vitorino to the Puerto Rico INRE network. This subcore will train researchers of pilot projects that include a metagenomic component, such as microbiome studies. The subcore will assist in the implementation of projects, data production, data analysis, and manuscript publication. In addition, the subcore will be offering a metagenomics workshop to the members of the community of practice later on the semester or during the summer. Another good opportunity for our members will be comes from the professional certificate in data science led by Professor Carlos Ortiz, Dr. Abiel Roche Lima, Dr. Istoni Daluz Santana. This data science certificate is comprised of 4 courses: Introduction to Data Science, Main Applications in Data Science Application with Machine Learning, and Data Science Final Project. The role of the community of practice will be to promote this certificate among our members so they can benefit from this opportunity. In this slide, you can see a picture of the most recent participants that have already completed the certification. And currently, more than 168 participants have completed the certification from people around the island of Puerto Rico. I think that two or three persons from El Salvador and 2 from the main USA. Another opportunity that is available for our members comes from the computational biology subcourse led by Professor Anatoly Chorny. He collaborates with Puerto Rico INBRE researchers, students, and doctoral associates on research projects relevant to bioinformatics research. Currently, he is developing a Java desktop application called Assistant of Metabolite Selection for gas chromatography mass spectrometry research. We will be inviting Professor Anatoly Chorny so he can provide training opportunities for the members of our community. So he will become an instructor, as well as Dr. Perichi Guerra that will be invited to become an instructor. He is leading the support in statistics, and he provides statistical services to predominantly undergraduate institution researchers during the production of scholarly work and to those preparing grant proposal. This subcourse works in collaboration with the computational biology subcourse to support our researchers. And in this slide, I am summarizing the benefits of belonging to the bioinformatics community of practice. Access to the high-performance computing facility located at the high-performance computing facility through the Boca Raton cluster. Our members will be able to request accounts, and those accounts can be used for research purposes, education, like in courses or laboratories, and for training purposes. Educational materials for courses and labs will be shared with faculty members that will incorporate these experiences as part of their courses or laboratories. Bioinformatics and computational training, scientists around the island and institutions around the island will benefit from a faculty-developed program in bioinformatics for their faculty. Bioinformatics seminars that are coordinated through Puerto Rico INBRE will be available to our members so they can attend to those as well. The directors of Puerto Rico INBRE course will give orientation talks to our members so they can know the services that they provide here at the island, so they can consider those services when they are writing research proposals, educational proposals, or both. The last one, research and educational networking. The scientists around the island will have a system in place in which they can collaborate ideas, efforts in order to increase bioinformatics, and with people they can collaborate for solving issues that other people are facing nowadays. I would like to acknowledge that this project is supported by award number P20 GM 103475 from the NIGMS of the NIH. Thanks to all my colleagues at the PR INBRE here in Puerto Rico, and thanks to the organizers of this symposium for inviting me to give this presentation.
Thank you very much, Dr. Vasquez Quinones. That was a wonderful presentation. We have a few questions for you. The first one is, how did you get involved with bioinformatics and PR INBRE?
Okay, my path in bioinformatics started as an undergrad when I was exposed to the programming course. Then I got involved as a graduate student in the first bioinformatics course taught at my institution. And then I reconnect with the people that taught me bioinformatics at the Pittsburgh Supercomputing Center and my past mentor in bioinformatics. And then one day I was at the Medical Sciences campus talking to Dr. Jose Rodriguez Medina, that is the PI. And we were in the hall and he was asking so many questions. And it was funny because I told him my vision to improve bioinformatics skills around the island. And I talked about the device, the Minayan device. And few weeks later, I was receiving the device. And then he invited me to form part of the team that was writing the renewal for Puerto Rico IMBRE, and that was that. I started my path dealing with bioinformatics, and the NIH brought me the idea of the community of practice so we can benefit here in Puerto Rico around the island, increasing the bioinformatics skills.
Great, thank you. Another question, how many students do you aim to train in the next year?
Well, currently I have been teaching the bioinformatics course since I arrived here at Universidad Ana Geméndez. So I have been impacting more than 100 students. So I think that we will be impacting a lot of students because I creating a critical mass by attending first the professors that are teaching the courses around the island. So in the near future, I think that everyone will be receiving training in bioinformatics because my vision is that we no longer have any students, mainly from biology, that exit a program without these skills. So I have to say a lot of them.
That's great to hear. Okay, I don't think we have any more questions for you. Thank you again for presenting. That was a very wonderful talk. All right. So now I would like to introduce our next speaker. It is my pleasure to welcome Dr. Nicole Garrison. Dr. Garrison is currently an assistant professor of biology at West Liberty University in West Virginia and a member of the W.V. Embry Bioinformatics core faculty. She earned a PhD at Auburn University working on the phylogenomics, transcriptomics, and systematics of spiders. Her research utilizes a variety of next-generation sequencing technologies and involves the development of custom bioinformatic pipelines for a wide range of biological applications from conservation biology to evolutionary biology and biomedical research. The primary foci of her current position are aiding researchers in the pre-processing, analysis, and interpretation of their sequencing data, as well as educating undergraduate students in bioinformatics methods and programming. So please join me in welcoming Dr. Nicole Garrison.
Thank you. Get my slide up here. Hey, so hi, thank you for having me. I am, as she said, a junior faculty and assistant professor of biology at West Liberty University. I have kind of a hybrid position, so I work with the genomics core, but I also teach courses, so general biology courses and specifically bioinformatics courses. So I'm really there to kind of develop this bioinformatic expertise at this primarily undergraduate institution. So this talk is just kind of about the approach that I'm taking, that we are taking in West Virginia to start training our students and begin that pipeline of bioinformatic expertise at the undergraduate level. I think for a lot of us, our exposure to bioinformatics happens kind of late in the game. And so we're trying to shift that timeline back a little bit earlier and get some of these younger students involved in learning bioinformatics. So West Liberty University, if you're not familiar with it, which I wouldn't really expect you to be, is a very small school in kind of a rural setting in West Virginia. So we have the distinction of being the first institute of higher education in the state. We actually predate the formation of West Virginia. Little fun fact. We have only about 2,500 undergraduates. So it's a very small campus, not a whole lot of students there. Many of the students that come to our university are local to the area or the tri-state area. A lot of them are non-traditional students. They're returning to education after being away. And many of them are dependent on scholarship support. So it is kind of an economically depressed area in some senses. A lot of our students may be first-time or first-generation college students as well. Despite all of this, we have developed a really strong biology department at West Liberty. We have a ton of undergraduate research currently happening. And so we have students in the lab generating data. And with the advances in sequencing, we're generating more and more big data sets. So there's this need, this increasing demand for bioinformatic expertise at our university. We also have a developing postgraduate program. So we have MA, MS programs, and we also have a professional physician's assistant program. So there's a lot of biomedically oriented students that are attending the university at different levels. We are part of the West Virginia Embry Network. So we're a partner institution. We work really closely with particularly Marshall University, especially the Genomics Core and Dr. Jim Denver, and West Virginia University. We're the site of a major research project. Among many other research activities that are happening there, Dr. Horzempa has a very well-developed lab that is looking at erythrocyte invasion by Francisella tularensis. Lots of microbiology, fun sequencing happening at West Liberty. We do a lot of our sequencing, most of our sequencing at the Genomics Core facility, which is located at Marshall. And so this is kind of the other half of my duties, is to be the connection between the Genomics Core and the West Liberty faculty. So when they generate data, when their students generate data, I'm there to kind of act as the go-between and help them analyze and interpret what they have. So there are multiple sequencing projects ongoing, many of them having to do with Francisella, but we are developing more and more independent kind of research programs that are utilizing this Genomics Core facility. So like I said, I am in this hybrid bioinformatic support position. So following along with these Embry goals, which I'm sure you've already heard multiple times, we have similar goals in West Virginia. We need to build expertise and capacity for biomedical research. We want to develop those biomedical research programs and increase competitiveness for external funding. We want to generate student research opportunities. We already do a really good job of that, like I said, but particularly research opportunities in bioinformatics is kind of where we're heading in the future. And so we're trying to train this future workforce that is equipped with the tools to do bioinformatic work, or at the very least, communicate with bioinformaticians that they may collaborate with in the future. So I feel like bioinformatics is the missing link right now at West Liberty and probably at a lot of institutions across the country. Researchers have very interesting questions. They generate a lot of data because it's much cheaper to do that in a lot of senses these days. But they face these bottlenecks in data analysis that are really common. They also have difficulty interpreting results that they get. If they have someone help them analyze it, they may not be able to fully interpret what they are presented with. So there's just a lack of bioinformatic expertise at the faculty level, graduate level, and that all comes from this lack of training at the undergraduate level. So that's what we're trying to correct here. We have some really common challenges that I'm sure you're all aware of, but there is just this lack of awareness amongst the students, the undergraduates, of bioinformatics. If you ask any given undergraduate at our institution, how would you define bioinformatics? they would probably give you a blank stare. They don't have a great understanding of what it is or why it might be important or useful to them in their education. So we're really struggling just initially with them understanding that, you know, this is a thing and it could be a useful thing for them in their future research. There is a lot of training and resources available free online, but I think that's part of the problem that they face is that they don't know where to begin. So really guided training with one-on-one interaction is really important at this level, I believe. And it's very hard for them to come by. They may find the training materials that they find on their own and access on their own a little unapproachable. So they're not really sure how to get started. They don't really have the exposure to bioinformatic careers as an option. So a lot of them come in, they think that they're going to be pre-med, they're going to be pre-vet, pre-professional programs, or they're going to, work in a zoo. We have a huge zoo science program, but they don't really have this concept of bioinformatics as a career that they could attain. And they also are deterred in a lot of cases because they're biology students. It seems like they have an aversion to anything really heavy in computer science or statistics, or they perceive bioinformatics to be really heavy in both of those fields. What they don't necessarily realize is that there are many different ways to come at bioinformatics. You can be a biologist who picks up some computer skills. So you don't have to be a computer genius to begin this journey. So some potential solutions that I've been exploring at West Liberty, just first of all, demystifying bioinformatics. So present those career options that are out there in this field and do it as early as possible and as often as possible. So kind of working this into conversations, working this into lesson plans in more general bio courses, I think will be very helpful for us. People who are bioinformatic ambassadors, I consider myself to be one of these, are very important. So having someone on campus who has the expertise and some experience and can talk to you about how they found their way to bioinformatics and what that actually means on a day-to-day basis in terms of the work that they do. And again, just emphasizing that there are many different ways into this field. We also need to emphasize the role that bioinformatics is playing in research findings. So, again, this emerging increase in big data in all fields of research is really important to emphasize that bioinformatics is going to play a key role into the future for analyzing that data and understanding biological questions. And then I'm going to try to empower and engage these students. So that means providing training opportunities, those guided training opportunities at every level, really. In this talk, I'm really focusing on the undergraduates, but I think this probably should start even at lower levels. I'll talk briefly about some K through 12 outreach that we're doing. And so to develop this, this is a really nice, simple figure that I pulled from a recent paper. So you need to identify your training needs, which is what we're working on doing. Curate materials. Again, we're kind of in this beginning stages of developing our content for these bioinformatic training opportunities. And all of these things go together into what we teach the undergraduates. We collect feedback from them. And ideally, we would publish these findings. So we're not quite to the publish openly phase of this process at West Liberty, but we're hoping to get there someday soon. We also, because we have this foundation in undergraduate research, are able to incorporate more bioinformatic analysis into the students' research. So they have generated their data. Now they can sit down and analyze it rather than just handing it off to someone and then getting back the finished figures. So we're trying to work on getting them to figure out how to actually analyze their data a little bit with us. And just in general, emphasize that this is problem solving. You don't have to have all this code memorized. That's not really how bioinformatics happens, but you need to know where to find the tools and how to use them and be able to think your way through these problems that come up. So like I said, I've done some K through 12 recruitment. Of course, with COVID, it's been kind of limited. So this is very recent development. Actually, last weekend, I did a small recruitment event where I used these Ozobots, which these are little programmable robots that follow color codes. So you can teach young kids how to program in a really intuitive sort of way. So I had grades 3 through 5 young girls working on programming their Ozobots to solve a problem or find its way through a maze. And then for the older kids, I have actually gone into the high school classrooms and I've talked about next generation sequencing, introduced the concept of bioinformatics, and then we've done some really basic mapping and variant calling on a very small scale. So just to kind of get their feet wet, get them thinking about when they go to college, this could be an option for them. Kind of adding on to that, coming from that, we want them to have an option once they get to college. So we're developing a bioinformatics major at our institution. I am developing it. So right now it's just me. And I'm teaching a few different courses that are going to ultimately be part of this major. So the first one is biological programming, which sounds a little scary. Got to work on that name, I think, a little bit to make it more palatable to biology students. But this is just really basic computing skills. So you get introduced to Linux, the command line, a lot of text-based bioinformatic file processing. So lots of FASTQ, FASTA file processing, just learning how to automate tasks, learning the basics of programming that they probably haven't been exposed to at all before. So it's a class for complete beginners, and it's kind of amazing how much progress they can make in just a single semester. I will also ultimately be teaching what we're going to call Bioinformatics 1 and 2. Right now, I'm teaching Bioinformatics 1, and we are doing that through the Galaxy interface. So this is a completely online, server-based, interactive bioinformatic analysis workflows. And I find that this is really approachable for biology students who want to use bioinformatic pipelines for their research, but they are not so sure that they want to go down the route of Linux and command line and supercomputing. They just really have these small projects that they're working on. They want to know, how do I run an RNA-seq pipeline really quickly without installing anything on my computer or finding access to some kind of remote server. So it's pretty accessible, and we've been doing some different tutorials with them to kind of go through some really basic bioinformatic methods. And then we have a ton of affiliated courses, or what I'm terming affiliated courses. So things like phylogenetic theory, which I teach in the fall, applied microbiology, where the students actually get to sequence microbiomes, and then they will analyze it with my help. Genetics courses, we're working more bioinformatics, more computing into the basic core genetics that we teach at West Liberty. And then we have the opportunity for these independent student research projects as well. We're also elsewhere on campus, they are developing a data science program that's through the math department. So I expect there will be a lot of overlap between those two majors. So we're going to enhance research experiences by allowing them to actually generate their own next generation sequencing data. We have an Oxford nanopore MinION, and we have just secured an iSeq. So we will have Illumina, short read and long read data for them to analyze. So it's a really exciting time for the bio department. They're going to have so much data, and they're definitely going to need to know how to analyze this for their class projects. So that includes everything from amplicon sequencing, some people interested in doing eDNA. We've had a tick microbiome project done. I've used it for conservation genomics, so looking at relatedness of organisms of conservation concern. And then, of course, we can do RNA-seq as well. We also offer summer training opportunities. We have the SURE program, Summer Undergraduate Research Experience. That's an ongoing program at West Liberty, and I'm hoping to join in more with that in the future and actually training one-on-one students over the summer to do bioinformatic research. And then really specific to bioinformatics, we offer the bioinformatics bootcamp every summer. I say every summer. Last year was our first year offering it, and this year will be our second. But this is a 3-week event. It requires no previous experience. We go from having no computational experience to doing basic bioinformatic pipelines by the end of this project. So we start with an introduction to Linux and the command line. Then we go in the next couple of weeks into specific pipelines. So we give them a script that has the pipeline, and then we break this pipeline down into digestible chunks for them so that by the end of the week, they have run the entire thing and they can reproduce those results if they wanted to. So we do this for COVID-19 variant analysis and a mouse RNA-seq analysis. And the data that we use is actually from the genomics core. It's been generated by INBRI researchers in West Virginia. So here's kind of an example of how the lecture might go. We do a little PowerPoint introduction typically. And at the end of it, we may have some step-by-step directions for them to follow and explore and learn things about the input files and the analyses that they're going to run. So it's very one-on-one, very guided research. They work in pairs typically. That just kind of happened naturally in this first boot camp, people sitting next to each other, kind of helping each other out. And then Jim and I would kind of circle around and come in when we needed to help them troubleshoot and get through whatever problems they were having. So I think this is the most important aspect rather than the content of what we're teaching. That's pretty commonplace and I think pretty standardized in a lot of ways. But I think the interaction that we're having, the one-on-one, the guided help that they're getting is really important to these students. So here's an example of a path that we might follow, a pipeline that we might provide them. So this is our variant analysis on the SARS-CoV-19 data. So we would show them this at the beginning and they kind of lose their minds because it looks like gibberish. And then we walk through step by step, what is each of these commands doing? What does this mean? What happens if you modify it in this way or that way? And then they get to see from start to finish how this pipeline will get executed. So we go from the raw data all the way through to calling variants and then looking at them, comparing them across different samples assigned to different students, and they can compare their variants to one another. So this is currently just open to West Virginia INBRI undergraduates. So that's anybody enrolled currently in a West Virginia network institution. So it is kind of limited in terms of who can participate at this point. And we are capping it at about 10 students for now. This is still kind of in the pilot phase, I would say. We're still developing the content and the kind of core material for this bootcamp, but we plan on continuing doing this for several years to come. The students at the end of this get to do a poster presentation on an in silico manipulation of one of the pipelines. They get to kind of test out, manipulate some of the steps, use a different program, see what the output looks like, change the parameters of a particular program, and see how that affects their downstream results. And so that's what they present together at this poster presentation at the West Virginia Summer Symposium. So that will happen this year as well. It'll be at West Virginia University this year. So we have a lot of potential, I think, to expand participation in this event. Again, once we kind of have our core material down, our method figured out, we will hopefully be able to share and publish some of these curricular modules. So we are planning on doing this, but again, this is just kind of the initial phases. A really exciting time to be a part of the development of this bioinformatic expertise. So if anyone is on here that is a member of West Virginia INBRI, tell all of your students, have them apply. They can find us online. You can email me about it directly if you'd like. it's a pretty good deal. They get a stipend, they get housing and food paid for, and they get to learn all of these bioinformatic topics. And with that, I think I will take any questions that anyone has.
Great. Thank you very much, Nicole. That was an amazing presentation. We have questions for you.
Okay, great.
The first one is, what is special about people who return back to education later in life in terms of their ability to learn bioinformatics, and especially what career options they end up choosing?
So I'm not sure about the career options part of that, but I will say that the non-traditional students that I've worked with to date, and again, it's a small sample size, it's a small school, but they tend to be much more focused. They have a much greater appreciation of what bioinformatics is good for, what it could be used for, kind of more perspective, I guess, is what they come back with. So they are more open to kind of exploring these methods, and they seem to be more interested than a lot of the really young students that come, straight from high school in the career opportunities, especially. So, when I say that you can work, for a hospital, you could work for a private company, you could work for the Federal Fish and Wildlife Service, there are all kinds of different positions opening up for people with these types of skill sets. So I think they see the value in that.
Yes, definitely. So along similar lines to that, what is the minimal set of skills a biology-oriented student must have to kind of begin their journey into bioinformatics?
So I would say that I kind of hesitate to frame it as the minimal number of skills, just because I think that people with no experience are more than capable of picking up these skills very quickly. And I've seen it happen. I think what's more important is not having the fear that's associated with kind of the stigma of, oh, I'm not good at computers, or I'm not good at math. And they think that they need to be, like I said, like rock star geniuses when it comes to all of these different fields, when really what's most important is they love biology and they're willing to learn and try to learn the computer part of it. And so students who really don't like to fail seem to have a lot of trouble with bioinformatics because it's mostly failure. You try something, doesn't work, you troubleshoot and you figure out the problem. So it's definitely a skill and a muscle that not all of them have developed, but I think it's definitely worthwhile even for students who don't want to pursue a career in bioinformatics, just for the problem solving skills alone. I think it's very valuable.
Yes, definitely. Another question, does such a small undergraduate population have benefits when it comes to bioinformatics research?
I think it does, and it's kind of the same set of benefits that really are in our favor when it comes to research. We get that one-on-one approach. When the students have a question, they just walk right into my office and talk to me about it. So they have a lot of access to expertise that I think you might not experience at a larger school where you're kind of just one of many students in a course. And when I teach a bioinformatics course, I have less than 10 people typically, especially at these early stages of developing the coursework. So I get to work with them directly and really adapt my teaching style and the content of the course to them and their interests.
Definitely. So another question, which of the programs that you've mentioned seem to have the greatest learning impact on young students?
Of the different courses?
Yes, I think that's what was meant.
Okay. I think the biological programming has the potential to have a big impact because it teaches these basic programming skills that I think it's like a foot in the door into computer science that they may not have experienced before. So they know pretty quickly, I think, whether or not they like it. And then that's kind of like their first taste that brings them further along this career path toward becoming a bioinformatician.
Great, thank you. And then one final question. Have you had any of your bootcamp projects result in a publication?
Not yet, but that would be fantastic if we could get there one day. It's such a kind of short program in the grand scheme of things. We do have, like, the SURE program is like a 9-week program, and we have had student publications result from that. So I think it might need a little bit more time to develop than just the 3 weeks. But if we had a student that was interested, they could always pick up with student research at their institution or collaborate with West Liberty into the future. So there's the potential there, but it hasn't happened yet.
Definitely. Thank you. Okay. I think that is all. Thank you again very much for coming and speaking with us today, Dr. Garrison.
Thank you.
All right. Now I think we are going to move on to our panel discussion. So we have several speakers today that are principal investigators from Idea States. They are going to discuss today a bit about the research projects at NAPI, some expectations and outcomes, how researchers are using data to embrace collaboration and answer questions faster, and how initiatives like STRIDES are helping to democratize research funding. So I think I will give it over to you, Elia, if you are ready.
Yes, thank you, Laura. So again, we have here with us Dr. Gus Kasulis. He's the president of NEPI, Mr. Jesus Gomez, who's the strategic business executive for Google Cloud, Dr. Nicole Garrison, she's the assistant professor at West Virginia Embry, Dr. Luis Quinones, who's the co-director of Puerto Rico from Embry, and I don't see Dr. Judy Crabtree, who is going to be here as well. But the main question is, as I understand, NEPI as a network is trying to solve a problem that is common across the network, Embry's, and the major challenge is, as we just heard from Nicole and from Luis, how do you actually make this accessible to a lot of students? How do you combine bioinformatics and data science training in non-bioinformatics and data science training curriculum? How do you help students use bioinformatics in research projects? And then how do you actually move from education to careers? What do they do after this? So maybe, Gus, you can start by kind of sharing, what is your vision? How do you address this common need across the network?
Yeah, sure, Elias. So first of all, I'm happy to participate in this panel. I'm also the principal investigator of the Louisiana Biomedical Research Network, LBRN. So in that capacity, I could speak also to that in addition to NEPI. But I think, one of the big challenges that we all face is that, as correctly, I think Nicole mentioned, this fear of computer science, bioinformatics, how people get into it. We have a lot of people from biology that would love to use tools, and we have computer scientists that don't understand the biology. So, bridging that gap between those two disciplines becomes very important. And frankly, the only way I see that, obviously, our 23 states in Puerto Rico are doing their own efforts within each state. There's a lot of resources, a lot of manpower, a lot of talent that's devoted in each state. We are through NEPI, we haven't seen maybe as much networking and commonality, allow resources to be shared by others, understand best practices, what works, what doesn't work. There may be some specificity to each program, but maybe there are some common things that we could focus on. And what we hope through NEPI is to make available everything that everybody has that could be shared freely, could be made available, including their personal stories, right? Interviews, what Nicole just talked about, or our Puerto Rican colleague mentioned. What works, what's the personal story, how certain students have been approached, and I think through examples I think everybody else can learn to adopt some of this, because some of them may be specific to our own states and institutions. Some of them may be common, but I think what NEP is trying to achieve is become a medium of communicating across the board of all these stories and successes and see what could be common. My own, how do you transform things, right? Within LSU, at least, we have sort of decided that the only way to do this is to really promote new degrees. Try to get an undergraduate degree in data science and bioinformatics, something that commits people into taking specific courses and graduating specific credentials. We've tried the certificate issue, the training issue, but it's always within primarily at the graduate institutions, and my colleagues could comment on this. It's always typically very hard to get a student to participate in something that's extracurricular or certificate, because the primary interest is to graduate and get a degree in something. So unless you have a degree program that really focuses, on data science and overall, and perhaps bioinformatics specifically, or how data science plays into bioinformatics or the other way around, you wouldn't have probably the impact. So one of the ideas here at Louisiana is to try to promote within PUIs the creation of bachelors of science, either as a major or a minor, depending on what the university is. And then at that level, through our Louisiana IMBRI LBRN, try to help them teach that, maybe through free content, through things that we could provide, other through freely available tools, through collaboration with biotech or whatever we can. It's a difficult issue because, as you know, probably most of you, changing curriculum, promoting a new course, it's a very slow process, at least here in Louisiana. But we at LSU are trying now to create an undergraduate degree and hopefully a master's degree. There's a master's degree already offered in New Orleans. It hasn't really been launched yet. We hope that to make it available across the institutions in Louisiana. But unless we really somehow put this into the undergraduate curriculum, at least as a minor, I don't think we're going to have the impact. And I'll stop there and let my colleagues chime in.
Yeah, Luis, I know that you've thought about this yourself as well. What is your thought about this?
I agree with Dr. Casulas that we have to share our common efforts to the IMBRE web pages and to the NIPE web pages, so everyone can have access around the nation. So if I can provide something that can be useful to Louisiana or to Virginia, it will be a great pleasure. So I prepare the stuff not just for me. I want to share it with everyone around the island, and that is what I'm doing right now, and starting toward that path to share everything that I got with everyone. But in that sense, I have to give the training. So I am creating right now a critical mass with people with interest in bioinformatics, so they can start teaching this stuff in their courses. So people that wants to belong to the community of practice is a voluntary step. But people that don't want to participate of this initiative will receive the impact and the training, because teaching faculty are going to include this stuff in their courses. So across biology, they will teach this stuff, and no students will escape at least receiving some kind of knowledge and training. So they will be prepared to face the requirements after they exit our programs.
Nicole, did you want to add to that?
Yeah, I mean, just to kind of reemphasize, that's true. We should be sharing. I think that part of the problem is not that there aren't resources, but that they're not organized in a way that students can really access. And so it is important to get the faculty to incorporate these lessons into their curriculum. So one thing I've been trying to do, and people are very willing to let me do this, is do guest lectures for a particular topic that is more bioinformatically oriented. Even if it's very basic, just introducing the students to me as a professor and to the topic seems to be very helpful. And then they start asking questions about, like, where can I learn more of this? If I take this class, will I get more of this kind of stuff? Because they just really don't even realize what bioinformatics is. So I think that really does help to get other professors in non-bioinformatic courses teaching some of this type of material.
So with us, we have Jesus, who hasn't had the chance to introduce himself yet. So I hope that he does this now. But also, I hope, Jesus, you can put aside kind of your professional role for a second as a person who has maybe a product, a solution. And think of this as, this program, this IDEA program, targets a very specific audience that has unique needs and special kinds of objectives. So just as a company, as someone that has that industry perspective, how do you view this perspective? Maybe starting from an introduction of who you are and what you're here to represent, and then into that problem that we're discussing.
Yeah, sure. Can you hear me? Yes. Okay, perfect. My name is Jesus Gomez. I am part of the Strategy and Innovation Group within Google, and I am responsible for the solutions in education and academic research. I've been in Google over 5 years, almost 6, and have worked with many research groups on bioinformatics across universities, biobanks, the Broad Institute, etc. I have been also engaged on the Strides program. We were the foundational member, first participated member of the Strides program. So one of the things that we keep hearing from the bioinformatics that is very aligned to your comments is when you ask researchers, for example, this is just an example, how much time do they spend in trying to reproduce results? Sometimes they say that 30% of their time is just trying to reproduce results. So imagine that you have a grant where you have to apply personal medicine pipeline and you work with another institution. That institution has to provide results. You have to validate that result. You get the pipeline from them. You spend 30% of your time, maybe even 30% of your grant in trying to reproduce that result. And that's a challenge, right? So one of the things that we are doing from the technology perspective is we're doing two things working with NIH and the rest of partners here. One is technology development. It's how much you can abstract researchers from the complexity of technology so that they can focus on science as much as possible. Okay, so if you run a pipeline in a university and you run in another university tomorrow, you should get the same result easily. That's one of the things that we are doing. The other thing that we are doing is we are working with Strides to create personalized training. Okay, where we say like, okay, this is for bioinformatics, this is for medical imaging, this is for interoperability of healthcare data. And that's, to your point, that could be this type of training that is created between the industry and academic can be great to be incorporated in the curriculum in the new programs that you are launching in university. Type of transversal skills. There are some basic transversal skills that will apply to everyone. Those are training that are funded by NIH that you can leverage. NIH is being allocated that budget. They send people to your university to train on that, to use this tool, to know how to use this tool. And then you on the top, you add your own curriculum content, your science knowledge, right? And I think if we can do that, we can help the community to actually get much more out of the grants much faster, right? I think we are at that point where we are at the intersection of technology and science. And sometimes we have the problem that we are making too difficult for great scientists to use technologies. You know, they don't have to become an expert in technology. They have to use the tools. And our job is how easily you can make them to use this tool so that they can focus on science.
Thank you. But, you know, I think for all of us, that brings us to the next question. The question is funding, right? So a lot of the efforts that are being described here are funded through NIH grants, you know, the Senate grants, etc. So Gus, you know, as somebody who's managed and received millions of dollars in grant funding, how do you see a more effective use of that funding to incorporate such training, you know, introduction to different tools? You know, these are tools that perhaps are not very cheap, like Google Cloud. You know, how do you see that just from a funding perspective?
Well, obviously, the funding that's provided by NIH or other resources is fairly limited. And typically is driven by large programs that are funded on a competitive basis that unfortunately for us sort of excludes or doesn't focus on the idea states. And one of the basic principles of NIGMS at this point is that in order to have the country be competitive, all states have to participate and be elevated. So I think it's a responsibility not only of the federal government, but also companies like Google, Amazon, IBM, to really focus and provide resources to those 24 states, because that's basically where perhaps even the biggest impact could be achieved in terms of, you know, manpower development. So I would definitely try to attract a institutional, private, public partnerships to create an environment where it's conducive to be transformative. And, you know, I would turn the table to our Google representative, Jesus, and ask him the question, if Google was to think of an undergraduate curriculum that focuses on data science and bioinformatics, you know, what would it be? Because, you know, the perspective is different, right? You know, in the university setting, we're teaching biology, we're teaching, you know, a lot of molecular biology, and sort of by the way, you first ask the question and then you go about finding how to answer the question. And one of the approaches could be to use a bioinformatic tool. A lot of the other approaches are more wet lab based that a lot of people concentrate. But we're at the point that the reverse may be happening, where basically through systems biology, a lot of the questions could be posed that are totally different questions that you would ask from the biology perspective. So, and I think sitting in companies like Google or Amazon and so on, I suspect their perspective is very different. So I don't know, Jesus, whether Google has been thinking, what would it be the ideal curriculum for an undergraduate degree in data science / bioinformatics?
That's a great question. So this is something that we keep refining based on things that we heard from you, right? Because you are the ones that actually knows all the challenges and how we can overcome. There are two things that we are trying to do as an example. In our side, the first thing is for students, for example, we give all the consumption is free. Any student globally can access our tool for a teaching and learning practice at the school at no cost. Okay, that's the first thing that we do. Like everyone that is curious to use this tool should be able to use it, even if they cannot afford it. And that's why we have our credit program. At the same time, we have created free training that is available in internet that you can incorporate in your program. For example, Johns Hopkins Medicine School is using our free training as part of the curriculum. you could include that as well. With basic skills that we think are very important for the undergraduate students so that they can start asking the right question about the data. Okay, and I will give you an example. All the NIH reference data sets are on cloud through Strides. Any student can access GCGA, DBA, Gen3 data, the public data set, and can run analysis with these tools at no cost. And one of the things that you can check, for example, is getting people that has a variant specific genomic and compare with the reference data set on the average people, and you can start seeing the difference. Then you can pick on 13 genomes, start seeing the difference. So what we are trying to do is like how easy we can do to encourage that curiosity of the student, right? Then there is one gap that is the most difficult one, is that, yeah, we can create that content, can give access to the technology, but behind that content is the science knowledge. And this is something that we will never be able to replace. This is where you need your programs, right? You need to make sure that you can connect a student with these tools and you help them to reason and to find this answer by themselves, right? So I think in our case, what we are trying to do is just helping your students to be able to answer bigger questions much faster, okay? But then that is the challenge of why you need a program, right? I think is not a challenge, it's a reality. You need a science program to be able to ask the right questions.
Yeah, I will let my colleagues also chime in, but you know, and you don't have to answer this right now, but I'll pose this question. You know, with Google and under your leadership, be willing to partner with NEPI and the IDEA program in creating the IDEA curriculum?
Actually, yes. Actually, yes. This is something that we can do. We have people in our team that sit with your professor, and your professor say, I'm teaching this and this, and say, okay, so in this section, this tutorial is relevant. And it helps you to map the menu to what you are teaching. And that's something that we can do for you. Okay, so there is a challenge. We all need this type of skills. And to your point, we all need to make sure that this gets out of all the institutions. You know, I'm telling you, I got my degree in Spain. In Spain, nobody has access to large data centers. Nobody has access to tools. I had a challenge. Right now, we have the possibility to get rid of that challenge to everyone. So everyone has the same access to the same tools. And this is something that we take it really seriously. And we have resources for that. So if you are looking into incorporating this kind of tool in your program, that is exactly why we have created these tools. And we are happy to collaborate with you, because for us to be better as a society, we need to develop skills in all type of researchers everywhere.
Yeah, thank you. We're going to draft you for the NISBRI meeting October.
So maybe just to give also an opportunity to Luis and Nicole add to this. I mean, what do you think is the role of NEPI? I know that NEPI exists in part to kind of connect all of these different efforts, not only just to connect them, but also to seek funding, right? So, I mean, Nicole, Luis, who wants to add to this discussion about, you know, funding challenges?
So I need to correct you, if I may. We're not-for-profit institution, so we don't have lobbying authority, but you know, we are promoting networking to make people competitive.
Right, exactly. Yeah, in that sense.
Do you want to go first, Dr. Garrison, or do I go first?
Go for it.
Okay. I think that I have seen NEPI in action during the last year's Southeast regional conference. And they brought a lot of information, shared a lot of information with us, and spread the word among the participants. So I think that NEPI performs a vital role in the IMBRE programs, and it's a way to unite all of us to increase the diversity and receive more information of the opportunities that are out there for us. So I think that the collaboration with NAPI and Google will be a hit.
That would be fantastic. I mean, we would love to do it. We even have, just to give you an idea, we even have worked with NSF and NIH to create a separate budget just for minority institutions. Okay. And just to give you an idea of how we help in two ways: to unfunded researchers, to students, I already say, right? We help you to incorporate in the curriculum. They have free access to the tools. Even for postgraduates and principal investigators, we have programs that give them credit so that they can use this tool to come up with very competitive grant proposals for free at no cost. Okay. And once they receive the grant, we have just launched the last year the possibility that is a flat fee, so that you can use this tool whether you run one time the simulation or you're running 1 million times. You will never exceed your budget. This is something that we are doing because we understand that you have to be able to attract good researchers, and these good researchers need to understand that if they join your university, they can win grants. And for that, we have to bring this type of tools and programs. So we continually listen. So we would love to partner with you, apply the existing program that we have, and if we have to tweak it for some reason, let's tweak it, because if it's good for the community, that's our mission, right?
That's great. Yeah.
Nicole, did you want to add anything to that? Or?
I'm going to be honest, I don't know that I have much to add. I'm actually learning a lot about the connections between all of these different programs. Coming from being outside of the INBRE system, I'm still learning how everybody is connected and how we're working together. So I think it seems like the connections are very valuable, and the knowledge of how those all work together, all these different programs and how Google might contribute, is important for someone in my position who is the connection between the students and these bigger programs, because INBRE and NEPI and these don't mean anything to our students. They are concerned with, how am I going to learn this skill and how am I going to pass this class? But if we can show them, hey, here are the resources, they don't really care where it comes from. As long as they know how to access it and what it's used for, I think that would be beneficial to them. So I'm going to take what I learn here and try and make more connections myself.
Yeah, the community is interesting.
Yeah, go ahead.
We have been monitoring communities even the last 2 years. This, for example, healthcare hackathons has been the engagement level of all the users has been impressive, and we have been monitoring on the background. And we are a member of the hackathon event, and we see that companies like Pfizer, GSK are being part of those hackathons as well. And you will be surprised on how, once you create with a small effort that community self-organized many times. You know, where you were mentioning before, I think that the challenge is, as a group, you had to decide what is the right organization so that people can find things easily. But once you create that, people start sharing pipeline, best practices. You know, there are, for example, companies that say like, hey, I have this problem. This is the topic of the hackathon. And there are groups from students from many programs that are heavily engaging, because it's also the possibility to get visibility and get a good job. And I would say don't underestimate the power of the community, because it's really important. And you know, there are a lot of people that volunteer and do a good job managing that.
And you know, that's actually a great point. And we have been thinking in NEPI to actually be able to do what I call active participation as opposed to passive. So we need to think about hackathons or health related things that would be across the board, you know, because, you know, we could list the information and it's sort of in a passive way. Somebody can go in there and look at it, but to actually engage and encourage young you know, students and really engage them in a meaningful way, you need something active, something that they can really work on. And the question is, what is this? Maybe a hackathon would be type of activity, could be something else, a problem posted. And frankly, personally, I see the involvement of industry as a major component to that. Not only for getting philanthropic funding that I think is needed, but even beyond that, to connect really the academic world with the industrial world, because ultimately our graduates would be, you know, your employees in the long run and elsewhere, and they will contribute, you know, in terms of economic development in multiple sectors. So that's what we're trying to do with NEPI. We kind of restarted, built a new website. We're trying to get some automated systems in. It will take some time to get to that level. Hopefully at the NSBRI meeting in October, we'll be able to launch some of these new activities and invite participants. It would be a process, right? And at some point, I'd love to see an undergraduate degree here in Louisiana taught in 20 undergraduate institutions with listed in all their curricula, but really taught by multiple faculty that are really dispersed, you know, within the state. That could be replicated in West Virginia, could be replicated somewhere else. And who knows, maybe at some point it would be an idea undergraduate degree, you know, that's actually uniform across the network. But I mean, that's basically the long-term vision. And I think, and I've seen this because I've been for many years in academia, you see some young minds that get exposed and they blossom and they become extremely good. But you have to provide an opportunity, because if they don't see it, they don't get to interact, they don't get the right mentorship early on, they actually lost. They end up in some other field, you know, that really they could have done much better elsewhere. So really mentoring and reaching as early as possible becomes very important.
Just want to add to that, we had another speaker earlier today from the Pistoia Alliance. The Pistoia Alliance brings together all the major pharmaceutical companies, and they have somehow figured out a way to partner on pre-competitive challenges that all of them face. And I think that this is somewhat similar to what we're trying to, you know, discuss here, because everybody has their interests, everybody's got their objectives, but in some areas like training and like, you know, encouraging students to pursue certain topics, this is a field where everybody can agree to work together and find a meaningful way to get that. And I think the hackathon idea actually is really great. I'll use this opportunity to say that you mentioned that the NSBRI meeting is in October, right, Gus?
Right.
This meeting information will be posted on the NEPI website very soon. And once it is, and we'll of course send it out to everybody who's participating in this meeting as well, maybe this is the time to start planning, because you know, hackathons, even though they seem like just the time to get together and do something, you know, in reality, they take a lot of planning for people to be prepared, identifying topics, all of these things. So this is definitely a great idea. Jesus, do you wanted to add something to that?
Yeah, I have an idea for that. Let's get, for example, the state of Louisiana, right? You talk to your HHS department in the state of Louisiana, and you ask them, like, what is more important for you? They're going to tell you maybe tobacco research, maybe cancer research. we can do that the topic of the hackathon. Okay, we get a public data set of data Louisiana research. And we ask a question to the participants. And then all the schools agree that the students participating are going to go into that contest, and you are going to give them a grade and an assignment based on the position on the hackathon. I commit to give the credit so that everyone can run the tool for free. And we will have somebody we will share training on the tools in advance. And we can even try to have somebody on site to help them.
That would be a fantastic idea. So
State is happy.
Yeah.
State is happy. Schools start collaborating. Students are engaged. We give it a try. Let's see what happens.
Yeah, and if we can replicate that later, and if we could work with Elia and see what we could fit something in that. Right now in Louisiana, cancer is #1 target, obviously, because there's a lot of effort. And if you look at heat maps, of course, if you look at even at across the board on idea states, cancer, obesity, diabetes, poverty, these are common, difficulties. So cancer is #1. Interesting enough, because we're now cannabis producers, cannabis-related drugs and therapeutics are also a target for across the state. But I'm not sure what kind of a topic could be mined out of that. I've been told there are 180 drugs, chemicals that actually use all sorts of purposes. And the question is, how could it be applied, and what kind of data mining it really is available and needed after that? But just parenthetically, I'm mentioning these two targets. Of course, we are big on infectious diseases. We have, in essence, 3 primate centers. The Tulane National Primate Center, I'm chairing the external advisory committee there. We have the Chimp Sanctuary in Shreveport and the New Iberia Primate Center. And so infectious diseases, including obviously COVID-19 and everything else, from West Nile to dengue to chikungunya and other viruses, also become very important. But there are all sorts of interesting topics. I think cancer may be an overall topic that really will reverberate across all our idea states. And that's a big one. As you know, President Biden is pushing ARPA-H, which is the program, the federal program that may add $5 billion to NIH that would be targeted for cancer research. The state of Louisiana has set as priority 1 cancer research. Our new president, Tate, that came from USC, South Carolina, has cancer as priority #1. Interesting enough, data science and bioinformatics is a priority #2 for LSU right now. So I'm pleased to actually see that, directive from the president. So yeah, that would be fantastic. So we can work on that, and we could revisit this issue with, Google and, Pine, biotech, and see what can be done.
So briefly, before everyone else leaves, because we're kind of running over time, I did want to mention Laura here, who has been doing a wonderful job throughout the symposium to coordinate everybody's talks and introduce everyone. She's going to be around to be involved in this project. So hopefully over the course of the next few months, we'll be able to be in contact with all of the in-breed programs related to bioinformatics and data science. We've got the contacts at Google who have been participating here and presenting their tools. And we'll try to really involve as many people as possible. And maybe that's one of the first things, like you mentioned, what is that major topic that everybody can agree on, that students will be interested in, companies will be interested in, faculty will be interested in, maybe in terms of funding priorities also, what is the top funding priority to give them a motivation?
And I think on a heck of a thing, even NIH could be interested through supplemental funding, especially, Google or anybody else would be interested. So if we could do something in the context of NSBRI, it would be great, because, normally we get up to 1,000 participants. Because this is virtual, it's possible that we go to 1,500 or higher on the first week of October. There will be a lot of people, including administrative, political, and other entities listening in. So I think this is the time where our network could really push forward for the next sort of transformational steps. Because we kind of, if I may, we kind of wobble around. We're doing different things. We're doing a great job locally, but we don't have orders of magnitude, right? We haven't really harnessed the power of those 23 states in Puerto Rico, working together. We do represent half of the country, maybe even more than half of the population, and a lot of resources. So people have to listen to us, and we need all the assistance that we deserve, and to really be able to fulfill, participating, in our success, as we have demonstrated really by graduating nationally competitive investigators that come from the idea state. I'm only going to leave you the last note from the point of view of NEPI. The idea states are the net exporters of talent to everybody else, right? So our undergrad students, graduate students, postdocs, and faculty get extremely successful. And unfortunately for us, a lot of them moved to MIT, Harvard, Texas, and elsewhere, which is, I think, a success. But ultimately, I think we need to create circumstances where we can develop these resources where they originate.
All right. Well, thank you everyone again for participating. We have a plan of action. Great contribution from everyone who joined and from all the speakers. So thank you again. We will be following up and being in touch. Again, Laura, I wanted her to be on here and for everyone to connect the dots and really be in touch. So thank you again, Jesus, for joining and supporting this idea. Really good to have you with us.
Yeah, thank you for having us, and thank you for what you do for your community. It's awesome.
Yeah. And thank you, Eli and Laura. You've done a great job. That's wonderful to actually listening on and off throughout these couple of days. Great job.
Thank you. Thank you for coming and speaking as well to all of you.
All right. Thank you, everyone. And thank you, Luis, again, for joining and for sharing today. And Nicole Garrison, thank you. And Gus, it's always a pleasure to see and hear your insights about all of this.
Take care, everyone.
All right. Take care.